everyone and welcome to Miss Estruch Biology. In this video we're going through everything you need to know for AQA paper 3. So basically all of the theory for A-level biology, which you know that means this is going to be a very long video, so either watch it in chunks, check out the time codes, or ×2 speed for those of you who are here last minute revising before your exam. And if that is the case, best of luck to you, and I really hope the exam goes well. And if you do need any more extra help, don't forget I've got so many resources to help you improve. Check out the links in my description for some of my top items, such as my flashcards and my A-level notes. But for now, let's get into it. So monomers are smaller units which can create larger molecules, and polymers are made from lots of monomers which are bonded together. And the examples that you need to know are glucose, amino acids, and nucleotides, so RNA or DNA nucleotides for the monomers, and then the polymers which those monomers can create. So glucose can create starch, cellulose, glycogen. Amino acids can make proteins, DNA, and RNA polymers. Now, to create these polymers, it is a condensation reaction. And this would be a 2 or 3 mark definition. So joining two molecules together would be the first mark. Creating a chemical bond would be the second. And removing water would be the third. To hydrolyze, which means to break apart or to split the monomers, that would be a hydrolysis reaction. And it's still 3 marks, but the opposite 3. The breaking of a chemical bond between two molecules and involves the use of water. So carbohydrates is the first biological molecule that you need to know. And I've got an overview here of the 3 levels of size of the molecules you need to know. So monosaccharide, mono means 1, so 1 sugar unit. Disaccharide, di is 2, so that's when you have 2 sugar units joined together. And the polysaccharides is when you have many joined together. And for each of those carbohydrates, there are 3 examples that you need to know. Glucose, fructose, and galactose are the 3 monosaccharides that you need to know. The disaccharides are sucrose, maltose, and lactose. And the polysaccharides, starch, cellulose, and glycogen. Now for the monosaccharides, the main thing you need to know is the structure of alpha and beta glucose. So alpha glucose we can see here. This is the level of detail that you'd be expected to draw this for AQA biology. And you would also need to know the formula, so C6H12O6. Now I did say you need to know alpha and beta, and that's because glucose comes as 2 isomers, which is when you have the same molecular formula, but there is a different structure. And the key differences I'll just highlight here. So for alpha glucose, on the carbon 1, which is the carbon that would be in this position, you have the hydrogen atom on top and the hydroxyl group on the bottom. For beta glucose, the only difference is those swap around. So the hydroxyl group's on top and the hydrogen atom is on the bottom. The disaccharides then is made up of 2 monosaccharides. When those are joined together, the chemical bond that forms is a glycosidic bond, and they are created via a condensation reaction. Now I did name the 3 that you need to know, but in addition to that, for those 3, you would need to know the word equation and therefore which 2 monosaccharides they're made from. So maltose is glucose plus glucose. Lactose is glucose plus galactose. Easier one to remember because there's lactose in the name. And then sucrose is made up of glucose plus fructose. And because all 3 are condensation reactions, that is why one of the products is water, because water is released. Now for the polysaccharides, you need to know the structure and how that links to the function, as well as some general other facts. So here's a very basic summary. Starch and cellulose are both found in plants, but they have different functions. Starch is a store of glucose, so it can provide chemical energy. And the cellulose is also in plants, but the function is structural strength in the cell wall. Glycogen is the only one found in animals, and this is a store of glucose as well, mainly found in the liver and the muscle cells. So just to summarize everything you need to know about the carbohydrates polymer, which is the polysaccharides, you need to know which monomer they're made of. Now, yes, it is glucose for all 3, but which isomer is different. Starch and glycogen are both made from alpha glucose, but cellulose is beta glucose. They're all glycosidic bonds, but they're different types. And the thing that's different about them is the location. And that's what these numbers refer to. So a 1 to 4 glycosidic bond means the bond forms between carbon 1 in one of the molecules and carbon 4 in another. And those numbers just refer to the position of the carbon in the glucose ring. So starch is made up of 1 to 4 and 1 to 6. And amylose, which is one of the polysaccharides of starch, only has 1 to 4, whereas amylopectin has both. Cellulose only has 1 to 4, glycogen also has both. And it's the 1 to 6 glycosidic bonds that create a branched structure. The 1 to 4 forms polymers in a straight line. The function we've already said on the previous slide, as have we said the location, but a little bit more about the structure then. And this again links to what we were just saying about the bonds. So amylose is an unbranched polymer, and it actually coils up to make a helix. And that is really useful because if it coils up, it can then be compacted to fit a lot in a small space. Amylopectin is branched, and the advantage of that is the branches create a larger surface area, so more enzymes can attach to the end and hydrolyze to turn it back into glucose when the plant might need glucose. All three have one feature in common. They're polysaccharides, which means they're large. And because they are large, they're insoluble. That means they won't affect the water potential of the cell and therefore no impact on osmosis. Cellulose has got a very different structure, and this is because it only contains 1 to 4 glycosidic bonds. So the polymer forms long straight chains. Now those chains line up in parallel next to each other and hydrogen bonds join them together. And we call that structure a fibril. Because there are so many hydrogen bonds holding these chains together, collectively they provide a lot of strength. And that is why cellulose is a very strong molecule. It's the large quantity of hydrogen bonds. Glycogen, now this is actually very similar to starch, in particular the amylopectin in starch. The key difference is it has a higher proportion of 1 to 6 glycosidic bonds, and for that reason it's even more branched and it can be even more readily hydrolyzed back into glucose. And that's an advantage because we find in animals, and because animals move, they will need more glucose. The next molecule is the lipids, and there's two lipids you need to know, triglycerides and phospholipids. So here is the level of detail you need to know their structure. They both have a glycerol molecule, and for the triglyceride, there are 3 fatty acid chains that come off that. So tri for the 3, tri meaning 3. Phospholipids, the key difference is one of those fatty acid chains is lost and instead it has a phosphate group attached to the glycerol molecule. Now how they form is the same, but I'm actually just going to go through it with the triglycerides. So it is a condensation reaction, and it will be 3 condensation reactions because there are 3 fatty acids that are going to be joining to the glycerol. Now, although it's a condensation reaction, that does not mean this is a polymer, and it isn't a polymer because it's not many repeated units joining together. So what we have is 1 water molecule is lost between each of the fatty acids and the part of the glycerol it's attaching to. So in total, that means we have 3 water molecules lost and 3 condensation reactions occurred. And the bond that forms is called an ester bond, and we have 3 ester bonds. So that is the structure of a triglyceride. A little bit more about the structure is the fatty acids. You do need to know that they can be either saturated or unsaturated. And a saturated fatty acid means that there are no double bonds between any of these carbon atoms. So you only have single bonds between the carbon atoms, and therefore it's fully saturated, holding the maximum amount of hydrogen. But this here would be your 1 mark definition in an exam.
Unsaturated fatty acids have at least one double bond between the carbon atoms, but you do have to state between the carbon atoms in the exam to get the mark. The properties then of the triglycerides. So the function is as an energy store, and the reason it stores lots of energy is because of the large ratio of the energy storing carbon to hydrogen bonds. And that is compared to the number of carbon atoms. So you have lots of energy stored in those bonds. The second reason is due to the high ratio of hydrogen to oxygen atoms, and that can actually act as a metabolic water source. So triglycerides can release water if they are oxidized. And that's actually what camels have in their hump. They have lipids, triglycerides, they don't have water. Triglycerides also have a similar property to the polysaccharides in that they do not affect the water potential and therefore affect osmosis. And this is because they are large and also hydrophobic, which means they will repel water. The last property is lipids have a very low mass compared to other types of tissue in your body, for example, muscles. And that's an advantage because it means you can store a lot without increasing the mass as much as extra muscle would. So you can store a lot of this for an energy storage without increasing the mass as much as other tissues would. Next then, the phospholipids. So we've already talked through the difference in the structure. It's still made by condensation reactions, but it would only be 2 condensation reactions because there's only 2 fatty acid chains. Still an ester bond, but we'd only have 2 ester bonds. And we can see up here, that is where the phosphate group attaches to the glycerol. Now this phosphate group is what gives the phospholipids some very different properties to the triglycerides. The phosphate group has a negative charge, and due to that negative charge, that bit, so the hydrophilic head, will actually attract water, but it will repel lipids. The fatty acid chain, so the hydrophobic tails, they don't have any charges on them. So they're actually described as hydrophobic, meaning it repels water, but they are able to mix with other fats or other lipids. And that is why the phospholipids can form this bilayer. We have these 2 charged regions, and in water, these phospholipids would position themselves so that the heads are on the outside, exposed to the water because they are hydrophilic, meaning they're attracted to the water. But because the tails are hydrophobic, meaning they are repelled or they repel water, they would spin round and the tails would face each other so they're not in contact with the water. Proteins then is our next biological molecule, and they are another example of polymers, and the amino acids are the monomers that they're made up from. You do need to know how to draw this general structure of an amino acid, so it is one of the things you could be assessed on in the exam. Now, a way to help you to remember it is to box it into these key groups. You have a central carbon in the middle of the molecule. There's a hydrogen atom that comes off, and an R group that comes off the top. Now, those could actually be either way round, top or bottom. The R group represents the variable group. So that changes for all 20 different amino acids. The amine group or amino group, that will always be present, and that is NH2. And the carboxyl group, that will also always be present, C double bond O, OH. Now, to make a dipeptide, which means 2 amino acids bonded together, it would be a condensation reaction, so water would be removed. The bonds that would form would be a peptide bond. To make a polypeptide, that would be when you'd have multiple amino acids joined together and multiple condensation reactions, still all joined together by peptide bonds. So that would create your primary structure of a protein, but that primary structure gets modified into the secondary, that gets modified into the tertiary, or it could be a quaternary. So we're going to go through what all of these 4 levels of organisation or development of a protein look like and how they're held in place. So the first level is the primary structure, and this is what is made straight after translation in protein synthesis. And the definition for this would be a 1-mark question. It's the order, or you could say the sequence, of amino acids in a polypeptide chain. So that's your polymer. The secondary structure then is when that primary structure is folded, or could be modified by twisting, because we can see here the alpha helix. But that would be the key marking point, that we then have an alpha helix or a beta pleated sheet that's created, and those are held in place by hydrogen bonds. The secondary structure then gets modified further, so it's further folded to create a unique 3D shape. And that shape is held in place by ionic, hydrogen, and sometimes disulfide bonds. And it's actually the primary structure that determines the location of these bonds, the ionic, hydrogen, and disulfide bonds. And it's the location of the bonds which determine how it folds and the 3D shape. The final level of organisation is the quaternary structure. That's cut off slightly, but that says structure there. Now that is still this unique 3D shape with the same bonds, but the only difference is it's a protein that is made up of more than one polypeptide chain. But it is still basically the tertiary structure. You just have more than one chain, so we call it quaternary. Enzymes are an example of proteins that you need to know. So an enzyme is a protein in the tertiary structure, so that unique 3D shape, and their function is that they catalyze reactions, and they do this by lowering the activation energy of a reaction. Now every enzyme is specific, and what that means is it can only catalyze one particular reaction, and that is due to the unique shape of the active site, which, this is an application of what we just said in the slide before, that primary structure determines the locations of the bonds, that determines the folding and the unique shape. So this is why each enzyme can only catalyze one particular reaction, and you get that unique or specific active site. So in that way, the active site is complementary in shape to a particular substrate. Now there's actually different models which explain how enzymes work. And at GCSE you would have learnt the lock and key model, but the accepted model currently is the induced fit model. So that is what you'd be expected to talk about at A level. You wouldn't be expected to mention the lock and key method. So the induced fit model is one that states that the enzyme's active site is induced, or it slightly changes shape to mold around the substrate. So initially, the substrate and active site are not completely complementary, but as the substrate binds, that causes the enzyme's active site to slightly change shape and mold around. That moving around the substrate puts strain and tension on the bonds, and therefore less energy is needed to break the bonds. And that is how enzymes lower the activation energy, which is the amount of energy needed for a reaction to occur. There are 5 factors that you need to know that affect the rate of an enzyme-controlled reaction: temperature, pH, substrate concentration, enzyme concentration, and inhibitors. So let's have a look at each one. So for temperature, if there is a lower temperature, that would mean that the molecule, so the enzyme and the substrate, would have less kinetic energy. Therefore, they won't have as many successful collisions, and you'd have fewer enzyme-substrate complexes. That is why the rate is lower at colder temperatures. Above the optimum, though, there is now so much kinetic energy that it causes some of the bonds to break. So, for example, the hydrogen bonds might break, and that means that the protein loses its unique 3D shape. The active sites change shape, and therefore you won't have enzyme substrate complexes forming, and the rate decreases. For pH, either side of the optimum pH, which actually can vary depending on where the enzyme is found, either side we have a very rapid denaturing of the enzyme. And that's because either too high or too low a pH will interfere with the charges in the amino acids found at the active sites. That can cause the hydrogen and the ionic bonds to break, and again the loss of that tertiary structure, and the active site changes shape. So we describe that as the enzyme denaturing, and again there'd be fewer enzyme substrate complexes, and therefore the rate of reaction decreases. Substrate and enzyme concentration have a similar idea behind them. There's no enzyme denaturing, but if we have a look at this one first, if there's insufficient substrate, there will be fewer collisions between the substrate and the enzymes, and that's why the rate of reaction is lower. But if you add more and more substrate, but no extra enzyme, eventually you'll get to the point where the enzyme active sites are all in use, or they're saturated. So even if you add more substrate, there's no more free enzymes, so the reaction can't go any faster. So the rate remains constant. For the enzyme concentration, if there's insufficient enzymes, so at these low concentrations, then the active sites will become saturated with whatever substrate is there. And that's why if we don't add more enzyme, the rate will stay low, but as you add more enzyme, the rate will increase. However, you'll get to a point though where if you keep adding more and more enzymes, but don't add any more substrate, you'll just have a surplus of enzymes and there isn't any extra substrate for those enzymes to bind to, so the rate won't increase any further. The last one was the enzyme inhibitors. Now, both type of inhibitor, the competitive and the non-competitive, both bind to an enzyme. So in the exam, you have to be specific and say which part of an enzyme they attach to get the mark. A competitive inhibitor binds to the active site, so we can see that here. And if it can bind to the active site, that means this inhibitor must be the same shape or very similar in shape to the substrate. And if the inhibitor is bound, that will prevent enzyme substrate complexes forming. So if you add more substrate, for a competitive inhibitor, the substrate will actually eventually be able to knock out the inhibitor, take its place, and therefore with a very, very high concentration of substrate, the effect of the inhibitor is no longer seen. However, a non-competitive inhibitor binds to the allosteric site, and that is a part of the enzyme away from the active site. So it doesn't bind to the active site. But as it binds, it causes the active site to change shape. And for that reason, the inhibitor has made it impossible for enzyme substrate complexes to occur because the substrate is no longer complementary to that active site and it can't bind. So that is how these inhibitors lower the rate of reaction. And even if you add more substrate, that won't help because the active site's a different shape. Now you could be asked for the biochemical test for all of those molecules that I've just gone through, or some of them anyway, the ones that I'm going to go through. So the first one is the biochemical test for starch. This would be a 2-mark question to describe the method. You add iodine, and a positive test result would be the iodine goes from orangey brown to blue-black. The test for reducing sugars, you would add Benedict's reagent and you have to heat it. So you need both of those ideas to get the mark. The positive test result would be the original blue colour would turn either green, yellow, orange or brick red. And those different colours indicate the concentration of reducing sugar present. So the more red it is, the higher the concentration of reducing sugar. The test for a non-reducing sugar test then, you would first of all have to do your Benedict's test for reducing sugars, and if it remains blue, which means a negative test result, you then go on to these stages. You would add acid and boil. You have to say boil because it does need to be above or at 100 for this to work. You would then cool and neutralise the solution. Because you've cooled it, you would then have to say heat and add Benedict's reagent again. And this time the positive result would be the solution goes from blue to either orange or brick red. Now it would always be orange or brick red this time, because if it was a non-reducing sugar, that means it was probably sucrose, and sucrose is made up of glucose and fructose. So when you hydrolyze it, that means it goes to two sugars, so glucose and fructose, instead of just one sugar, sucrose. So you've now doubled the concentration of sugar present, so it will always be orange or brick red. The test for proteins is you add biuret, which is blue in colour, and if you have a protein present, it will go purple. For lipids, it would be a 3-mark question. You have to dissolve your sample in ethanol, and you would do this by adding ethanol and shaking. And sometimes there is a mark for saying you have to shake. Once you've done that, you then add distilled water, and a positive test result would be white in colour, and we describe it as an emulsion, this sort of thick, milky texture. So the next biological molecule is DNA, or deoxyribonucleic acid. And this function is it codes for the sequence of amino acids in the primary structure. So it's very, very essential. It contains the genetic code, and it can be passed on to make new cells, but also it can be passed on to the next generation. Now DNA is a polymer, and you get 2 polymer chains joining together to create a double helix. So the monomer, you would have to be able to draw this in this level of detail. We have our phosphate group attached to a pentose sugar and the nitrogenous base. But because this is a DNA nucleotide, you would have to say that the pentose sugar is deoxyribose, and the nitrogenous bases, or nitrogen-containing bases, are guanine, cytosine, adenine, and thymine. So to make the polynucleotide, which is the name for the polymer, it would be a series of condensation reactions where water is removed and a phosphodiester bond forms between the deoxyribose and the phosphate group of another nucleotide. And that makes this sugar phosphate backbone, which is very, very strong because a phosphodiester bond is a strong covalent bond. Between the 2 polymer chains, we get hydrogen bonds, and those form between the complementary base pairs, which are cytosine and guanine, and adenine and thymine. RNA is the other nucleic acid that you need to know about, and it's almost identical in shape or in structure. The key differences are that instead of having deoxyribose as the pentose sugar, it has ribose. It doesn't have the nitrogen-containing base thymine, it has uracil. Now RNA is also, in terms of the polymer, it's much shorter than the DNA polymer, and that's because mRNA is only a copy of one gene, whereas DNA is all of the genes, and also tRNA is relatively short also. And all of the polymers are single-stranded for RNA. Now the functions you actually learn in more detail in topic 4, but you do need to know about one particular RNA molecule called rRNA. That combines with proteins to make ribosomes. So DNA replication then, in order for new cells to be created, every new cell needs their own copy of the entire genome, which is all of the DNA. So the DNA must replicate before a new cell can be created. And the way DNA replicates is described as semi-conservative replication. And what that means is one of the original strands of DNA combines with one newly synthesized strand to create the new molecule. So how that happens? Step 1, the enzyme DNA helicase breaks hydrogen bonds between the complementary base pairs. And that causes the double helix to unwind and the 2 strands to separate. And both of those strands will act as a template. Free-floating nucleotides within the nucleus will then align opposite the complementary base pairs, and the enzyme DNA polymerase will join adjacent nucleotides together. So it's joining the nucleotides together to make phosphodiester bonds. So now we have our newly synthesized DNA combined with the template strand, and that is our new molecule of DNA, or the daughter DNA. The evidence for this you need to know a little bit about also. So it was Watson and Crick who discovered the structure of DNA, so that double helix, but they only managed to discover that because of Rosalind Franklin's research on X-ray diffraction. Michelson and Stile, they conducted experiments which proved that DNA replication was semi-conservative. Now, ATP is what we call a nucleotide derivative, and that's because it's very similar in structure to DNA and RNA. It still has a pentose sugar, it still has a nitrogen-containing base and a phosphate group, but it actually has 3 phosphate groups. It will always have ribose and it will always have adenine. So adenosine triphosphate is what ATP stands for. And its function is it's an immediate source of energy for biological processes. So in other words, it's used in metabolism, which is all of the chemical reactions within a cell. ATP is made during respiration by ADP and PI, which is inorganic phosphate, joining together in a condensation reaction using the enzyme ATP synthase. Now, ATP releases energy when it is hydrolyzed. So when the bonds between the phosphates are broken, and we then have ADP plus that inorganic phosphate, that releases energy. And the enzyme ATP hydrolase is what catalyzes that reaction. Now, ATP can also do something called phosphorylation. And that is when that inorganic phosphate is actually transferred and bonds to a different compound. And in doing that, the compound that it binds to or bonds to becomes more reactive. And that happens to glucose at the start of respiration in glycolysis. The next biological molecule is water. And water has 5 key properties that you need to know about. Now, water is very, very important because it makes up about 60 to 70% of your body. And the reason it's so important links to these properties. Now, most of the properties are due to the hydrogen bonds which form between water molecules. And those will form between the oxygen of one water molecule and the hydrogen in another. Water is also described as being polar or dipolar because the oxygen has a slight negative charge and the hydrogen has a slight positive charge in the water molecule. So the 5 properties: number 1, water is a metabolite, which means it's involved in chemical reactions. So we've already seen in this video how it's involved in condensation and hydrolysis reactions, but it's also involved in photosynthesis. Because of its dipolar nature, it's a very, very good solvent, and this is important because if it can dissolve solutes, then they can be easily transported around the body in the cytoplasm in a cell, or in the plasma of blood, or in the liquids in the phloem and the xylem in plants. Chemical reactions also happen more readily in liquids. It has a high heat capacity, which means it takes a lot of energy to raise the temperature. And in that way, it can buffer temperatures. So that means the fact that our body is mainly water, it takes a lot of energy to increase our body temperature. And that's good because we don't want enzymes to be denaturing. It also has a large latent heat of vaporization, and that provides a cooling effect because this property means a lot of energy is required to convert water from its liquid state into its gaseous state. So if water does evaporate, that means a lot of energy has been transferred in that process, and it provides a cooling effect when we sweat or when plants are going through transpiration with water evaporation. Water also has strong cohesion, and this is due to the hydrogen bonds between the different water molecules. And cohesion means the water molecules are sticking together. And in plants, this is particularly important because that means you get this continuous column of water moving up the xylem. It is also what provides surface tension to water, and that can actually provide a habitat for certain organisms. Inorganic ions is the last part of the specification for this topic. And what you need to know is that ions can occur in solution, so for example in the cytoplasm of cells, in other bodily fluids as well like the blood, sometimes in high concentration, sometimes in low. And there are a selection of inorganic ions that you need to know and what function they have. Now this actually links to other parts of the spec, but the hydrogen ions, you could look at the importance of them in terms of how they can alter the pH of a solution, and that could have an impact on enzymes, but also hemoglobin, if you think about the Bohr effect that comes up in another topic. Or you could look at the importance of hydrogen ions in chemiosmosis, in respiration and photosynthesis. Iron ions are a component of hemoglobin and are involved in the transport of oxygen. Sodium ions are involved in the co-transport of glucose and amino acids in absorption, or you could look at the role of sodium ions in action potentials as well. Phosphate ions are found in DNA, RNA and ATP. The importance in DNA and RNA is the phosphate group is where the phosphodiester bond forms. In ATP, the phosphate group can be added to other compounds to make them more reactive. So within the eukaryotic cells, the key organelles that you need to know the structure and function of are all the ones listed here. And we're going to go through each of them one at a time. The nucleus, the key structures within that are the nuclear envelope, nuclear pores, nucleoplasm, chromosomes and the nucleolus. And the overall function of the nucleus is where DNA replication occurs, and it's a site of transcription, which is where mRNA is made, and it contains the DNA, which is the genetic code for the cell. We also have within the nucleolus, that is the site of RNA production, and it's where ribosomes are made. The endoplasmic reticulum, you have the smooth and the rough. And we can see here the top diagram, we've got the rough endoplasmic reticulum, and we can tell because we've got the ribosomes on the outside. And the other side of this top diagram is the smooth endoplasmic reticulum, and that has no ribosomes on the outside. Now the RER, which is the rough endoplasmic reticulum, that is where protein synthesis occurs because of those ribosomes on the outside. The SER, the smooth endoplasmic reticulum, that is where you have the synthesis of lipids and carbohydrates, and they also get stored there as well. The Golgi apparatus is a folded membrane making this cystine shape, and we have vesicles that will pinch off from those cystinae once whatever it is that's going to be modified has been modified and packaged. And we've got the whole list here of the different things that would happen. So it could be that the carbohydrates are being added to proteins to make glycoproteins, which might be embedded within the cell membrane. You can also produce secretory enzymes, carbohydrates, transport and modify lipids. We get lysosomes. Molecules are also labelled with their destination, essentially meaning they're going to have a molecule added so they'll be able to bind to receptors on the target cell for where that molecule is needed. Lysosomes are bags of digestive enzymes, and this is often involved in phagocytosis. So the enzymes that are required to hydrolyze the bacteria or virus, whatever it might be, would be in a lysosome. There'll be exocytosis, which is where the products are going to be released to the outside after it's been destroyed. The mitochondria is the site of aerobic respiration. So you'll have lots of ATP production here. It's a double membrane organelle with the outer membrane, and the inner membrane is this folded part making up the cristae. And the inner membrane is where oxidative phosphorylation happens, which is one of the key stages in aerobic respiration. They have their own loop of DNA, which is very similar to prokaryotic DNA. And this is so that they can code for the enzymes that they need in respiration. Ribosomes, now these are found in prokaryotic and eukaryotic cells, and that is because they are not membrane bound. Ribosomes are just made up of rRNA and proteins. You do have different sizes though, so the ATS is the larger size ribosome in eukaryotic cells, 70S is a smaller sized ribosome found in prokaryotic cells, as well as in mitochondria and chloroplasts so that they can make their own enzymes. And it's the site of protein synthesis. The vacuole, which is only found in the plant cells, not in animal cells, this structure is filled with fluid and surrounded by a single membrane called a tonoplast, and this is what helps to make the cell turgid and provide support. You'll have temporary stores of sugars and amino acids within the fluid as well, and the pigments within it may colour the petals, and that's what helps to attract pollinators. Chloroplasts, again, only found in plants, and this is the site of photosynthesis. You have, again, it's a double membrane organelle, and we have the outer membrane, the inner membrane, and then on the inside we have even more membranes, and that is these thylakoid membranes which stack up to create what we call a grana if it's plural, or just a granum if we're just looking at one of those stacks. And the thylakoid membranes are embedded with proteins and pigments such as chlorophyll, and therefore it's the site of the light-dependent reactions in photosynthesis. The stroma, which is the fluid part surrounding the thylakoid membranes, you have lots of enzymes in this location which are needed for the light-independent stages of photosynthesis. Cell walls are not in animal cells, but they are in fungi and plant cells, and they help to provide structural strength to the cells and prevent them bursting if lots of water moves in by osmosis. And that's because they contain a particular molecule that provides strength, and in plants that's cellulose, and in fungi that would be chitin. The plasma membrane is found in all cells, and this is in the cell membrane, so the cell surface membrane, but also when we say it's a double bound or membrane bound organelle, it's also made up of this phospholipid bilayer with the different molecules embedded within it. And the function of the plasma membrane is it controls what can enter and exit the cell. Prokaryotic cells, for example bacteria, you need to be aware of the key differences. So it could come up as a comparison or a contrast question. So first of all, the cells are much, much smaller. They don't have any membrane-bound organelles. They do have ribosomes though, because ribosomes are not membrane-bound, but they have the 70S size ribosomes. They don't have a nucleus because that is a membrane-bound organelle. So instead, their DNA will just be as a single loop, which is free within the cytoplasm. They do have a cell wall, and that is made up of murin. Sometimes they might also contain plasmids, which are additional loops of DNA. They might have a capsule around the cell, which is to help prevent desiccation and to evade the immune system. And sometimes they'll have flagella. It might be one, it might be many, which is to help them to be able to move. But those aren't always found, those are only sometimes found in prokaryotic organisms. Viruses are acellular and non-living. The structure of virus particles consists of genetic material, capsid, and attachment proteins, which we can see over here. So we have the genetic material in the middle, we have the capsid, and then we have the attachment proteins, or here they're labelled envelope proteins. Viruses replicate inside of host cells, and that is why it's very difficult to destroy them, because you would have to destroy the host cell to be able to destroy the virus. Now you need to know methods of studying cells, and the reason this is so important is the way we've discovered these internal structures or organelles is because of these advances in the methods to study cells. So advances in microscopes, and then also cell fractionation and ultracentrifugation to be able to isolate particular organelles to examine. So we'll go through each of these methods of how to study cells. First of all, with the microscopes, there's a couple of key definitions you need to be aware of. You also need to know the three main types of microscope. There are optical microscopes, which are the light ones, which you'd be using in school. You then have electron microscopes, but you can have either a transmission electron microscope or a scanning electron microscope. The definition of magnification is how many times larger the image is compared to the object. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate. And the resolution of an optical microscope is determined by the wavelength of light, and the resolution of the electron microscopes is determined by the wavelength of the beam of electrons. And because that wavelength is much shorter, that is why they have a higher or a more powerful, that is why they have a much higher resolution, meaning that the distance between the two objects is smaller. So let's compare the two general types of microscopes in more detail. The optical microscope, we've already said it's the light, but it is a beam of light. It's a beam of light that is condensed to create the image. In contrast, an electron microscope, it's a beam of electrons which is condensed to create the image. And what is used to condense the light and the electrons is also different. The beam of light is condensed using a lens, whereas the beam of electrons is condensed using electromagnets. The optical microscope has a poorer resolution due to the fact that light has a longer wavelength. So that means that the electron microscopes, because electrons have a shorter wavelength, they have a much higher resolving power. There's also a lower magnification for the optical, higher for the electron microscopes. You can have colour images though, and you can view living samples with the optical microscopes. So those are two big advantages. The electron microscopes can only produce black and white images, and because electrons can be easily absorbed, you have to have your sample in a vacuum. And because it's in a vacuum, you can't actually observe any living samples using an electron microscope. Now, just to go back to this black and white image point, you can still get coloured images if colours are added on programs like Photoshop afterwards. Because the optical microscopes have a lower resolution, small organelles in a cell are not visible. So you can't see details of the mitochondria, chloroplasts, ribosome. You can just see some of the large organelles, like we can see here. You can see the nucleus, you can see the DNA, you can see the chromosomes. The electron microscopes, they have much higher resolving powers, as we said, and the difference between the transmission and the electron is that the transmission, you have extremely thin specimens, and the electron gun produces a beam of electrons that transmits or passes through the specimen. So some parts of the specimen will absorb the electrons and therefore appear dark, some parts won't and the electrons would have passed through and they appear lighter. And we get these 2D images where we have the darker and lighter depending on whether the electrons were absorbed or not. And because there's a higher resolution, that is why we can actually see the details internally of some of those small organelles. A scanning electron microscope creates 3D images in contrast. The specimen does not need to be thin, and that's because the electrons are not going to be passing through the specimen. Instead, the electrons are beamed onto the surface, and the electrons are scattered in different ways depending on the contours. And that is how we get these 3D images, which we can see here in these different blood cells in a sample from the blood. You could be asked to calculate magnification of an image, so the formula that you need to remember is IM, and that is the image size equals actual size times magnification. Or you can rearrange it to work out the actual size or the magnification. And the key thing that you need to be aware of when you are doing these calculations is the image size and the actual size have to be in the same units. So you need to be familiar with how you convert from millimeters to micrometers or micrometers back into millimeters. Those are the two most common conversions in a magnification question. But if you want to know any of the other conversions, here they are. You might also need to be able to measure the size of a specimen using the eyepiece graticule. And inside of the optical microscope, there is a scale on a glass disc, which is called the eyepiece graticule. So it'd be just here within that eyepiece. And this can be used to measure the size of objects you're viewing under the microscope. However, each time you change the objective lens and therefore the magnification, you have to recalibrate the eyepiece to work out what the distance between each of these different divisions is actually worth. Now you need to know how to do that, but I'm not going to go through that in the summary video, but I'll link the video up here of how to calibrate the eyepiece graticule. Cell fractionation is the final way that you can study cells, and this is used to isolate the different organelles so that they can be studied further. First thing that happens is the cells are broken open to release the contents and the organelles. The cells need to be prepared in a cold, isotonic and buffered solution, and you need to know why it has to be those three conditions. So it has to be cold to reduce enzyme activity, and that's because when you break open the cell, you'll be releasing enzymes which wouldn't typically be in contact with the organelles and they might damage them. It has to be isotonic so that you don't have excess water moving in or out of the organelles by osmosis, because we don't want the organelles to burst or shrivel, because then we won't be able to study them. And it has to be buffered because if it became too acidic or alkaline, again, it could damage the organelles. Now, cell fractionation is a two-step process: homogenization and ultracentrifugation. So first of all, homogenization is when you break open the cells, and that can be done in a blender. As long as you have, as we said, the solution that you're blending up your sample in, as long as the solution that you're blending your sample in is cold, isotonic and buffered. Then we'd need to filter to remove the large debris, and then the filtrate, we can then use ultracentrifugation to isolate the different organelles. So we'd put our sample into a centrifuge and we'd spin at different speeds. The organelles are going to separate according to their densities, and that is why we have to spin our sample at different speeds. And this is what differential centrifugation is. So when we spin our sample, the centrifugal forces cause pellets to form at the bottom with the most dense organelles. So we can see here in this first image, we've got all of the organelles equally distributed. And if we first spin at low speed, the most dense organelle will form a pellet at the bottom. We would then remove that pellet and spin the rest of the filtrate again at a slightly faster speed. We'd then get the next most dense organelle forming the pellet, and we repeat this process at increasingly faster speeds. So each time that liquid, which is the supernatant, is removed and the pellet is what the organelles will be in, and we can examine those. Now, just to let you know the order of the density, in that first centrifugation, the nuclei are the most dense. So those would be isolated in the first spin. The next most dense are the chloroplasts and the mitochondria, then the lysosomes, endoplasmic reticulum, and finally the ribosomes. So that would be the order in which you would collect your different organelles. We then move on to cell division, and eukaryotic cells enter the cell cycle and divide by mitosis or meiosis. But in topic 1, you only learn about mitosis. In comparison, prokaryotic cells replicate by binary fission, and viruses do not undergo cell division at all because they are non-living. They do still replicate though, but viruses replicate inside of a host cell. They will invade that host cell by injecting in their nucleic acid, so their genetic material, and then it will be the host cell that uses that genetic material to replicate the virus particle. The cell cycle that eukaryotic cells will be going through includes these key stages. Interphase is the longest stage of the cell cycle, and it includes G1, S, G2. G1 is when the cell is going to be increasing in size and the organelles will double. S phase is when DNA replication happens. G2, you'll have further growth, but also it says preparation for mitosis. In G2, you'll have this error check stage. So if there are any errors in the DNA replication, the cell would be destroyed at that stage. Nuclear division is either mitosis or meiosis, but in topic 2, we just focus on mitosis. The final stage of the cell cycle is cytokinesis, and this is when the cytoplasm divides to create the 2 new cells if it's mitosis. Mitosis is split into 4 key stages, which are our PMAT, prophase, metaphase, anaphase, and telophase. The key facts about mitosis are it's only one round of division, genetically identical cells are created, the cells are diploid, which means there are 2 copies of every chromosome, and this stage is used for growth and repair. So a specific example of that, which we'll see later in this video, is the clonal expansion of B cells. So that is growth in the sense of we're creating lots of new cells. So in prophase, the chromosomes will condense and at this point they become visible. And in the animal cells, the pairs of centrioles will move to opposite poles, which means opposite sides of the cells. The centrioles are going to be creating these spindle fibres, which are released from both poles to create the spindle apparatus. And these will attach to the centromere and the chromatids on the chromosomes in the later stages. Plants have a spindle apparatus, but they don't have the centrioles. In metaphase, we can see that the chromosomes will then line up in single file along the equator, and the spindle fibres are released from those centrioles at the poles, and they'll attach to the centromere and also the chromatids. In anaphase, the spindle fibres will start to retract and pull back towards the centrioles, and in doing that they'll pull on the centromere and chromatids, and this causes the centromere to divide in 2, and the individual chromatids are pulled to the opposite poles of the cell, and therefore it separates the chromatids, and once those chromatids are separated, we now actually call them chromosomes. Now this stage requires energy in the form of ATP, and that's provided by respiration from the mitochondria. In telophase, the chromosomes are now at each pole of the cell and become longer and thinner. The spindle fibres will then disintegrate and the nucleus starts to reform. The final stage in the cell cycle is when the cytoplasm splits in 2 to create the 2 new genetically identical cells, which the mitotic index can be calculated by counting first of all how many cells are visible in the field of view, and then counting the number of cells that are also visible but in a stage of mitosis. You can then do a percentage calculation. So you'd be doing the number of cells that you can see in a stage of mitosis divided by the total number of cells present and times it by 100. Prokaryotic cells don't go through mitosis, instead they go through binary fission. The first step of that would be the replication of the circular DNA and of the plasmids, and then the second stage would be division of the cytoplasm to produce 2 daughter cells, each with a single copy of the circular DNA and a variable number of plasmids. Viruses are non-living, so they don't undergo cell division. Instead, they inject their nucleic acid into the host, and the host cell replicates the virus particles. So now on to plasma membranes. All cells and organelle membranes have the same structure. It's this fluid mosaic model, which is named because of the fact that it does have some slight movement, and also it's composed of a range of different molecules, such as phospholipids, proteins, glycoproteins, and glycolipids. All of these molecules are arranged within the phospholipid bilayer, and they create the partially permeable membrane. To create the bilayer, the phospholipids align in this manner because of the fact that the phospholipid has a hydrophilic head due to that negative charge on the phosphate group, and that causes it to be attracted to water. Whereas the hydrophobic tails, those are repelled by water but can interact with lipids. So the phospholipids end up spinning round so that the tails are opposite each other, the heads are on the outside where they can interact with water. Cholesterol is present in some membranes too, and this will restrict the lateral movement of other molecules in the membrane. This is useful as it makes the membrane less fluid at high temperatures and prevents water and dissolved ions from leaking out of the cell. The other components of the membrane are mainly the proteins, and these are embedded across the cell surface membrane, either as peripheral, meaning they're just on the outside, sometimes called extrinsic, or they could be integral, sometimes called intrinsic, meaning it spans from one side of the bilayer to the other. The peripheral proteins provide mechanical support, or they can be connected to proteins or lipids to make glycoproteins and glycolipids. The peripheral proteins provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and also carbohydrates can bind to the lipids directly to make glycolipids. The function of these is cell recognition or as receptors. The integral proteins are protein carriers or protein channels involved in the transport of molecules across the membrane. Protein channels are tubes, and these fill with water, which will enable water-soluble ions to diffuse across the membrane, whereas the carrier proteins will enable molecules to bind with them, and therefore larger molecules like glucose and amino acids can cause those carrier proteins to change shape and therefore transport the molecule to the other side of the membrane. So when we say that the membrane is partially permeable, what we mean by that is only lipid-soluble substances and very small molecules can pass across the membrane by simple diffusion. Other molecules, such as water-soluble or polar substances and large molecules, can't simply diffuse across the membrane. Therefore, they have to be transported across by other means, which could be facilitated diffusion, active transport, or if it's water, osmosis. So simple diffusion, first of all, this is the net movement of molecules from an area of higher concentration to an area of lower concentration, and this will continue until an equilibrium is reached, and this process does not require any ATP. The molecules do still have energy to be able to move, but this is just due to the kinetic energy that they possess, and that is why they're able to move constantly as a fluid. For molecules to diffuse across the membrane, they have to be lipid soluble and small. If they're not lipid soluble or small, then it will be facilitated diffusion instead. And this is still a passive process because the molecules are moving from an area of higher concentration to lower, but because they can't simply diffuse through the phospholipids, instead they have to diffuse through proteins. Now this could be through protein channels, where we have those tubes filled with water, and therefore water-soluble ions can dissolve and then diffuse through. This is still selective though, as the channel proteins only open in the presence of certain ions when they bind to the protein. Carrier proteins will bind with a molecule such as glucose, which causes a change in the shape of the protein. This change enables the molecule to be released to the other side of the membrane. Osmosis is the movement of water, and that will be from an area of higher water potentials to an area of a lower water potential across a partially permeable membrane. The water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. Pure water or distilled water has a water potential of 0, so you cannot get one any higher than 0. It's always going to be 0 or negative. And that means when solutes are dissolved in water, the water potential will be negative. And the more negative the water potential, the more solutes there must be dissolved in it. Now, 3 key terms that you need to know linked to osmosis are isotonic, hypertonic, and hypertonic. An isotonic solution is when the water potential is the same in the solution and the cell. Hypotonic is when the water potential of the solution is more positive, so closer to 0 or closer to pure water compared to the cell. Hypertonic is when the water potential of the solution is more negative than the cell, so there'd be more solutes dissolved in it. And in an animal cell, we can see the impact that would have. In an isotonic solution, because the water potential is the same, there's not going to be any net gain or loss of water. In a hypertonic solution, there'll be more water moving out of the cells by osmosis, and that can cause animal cells to shrivel or crenate. If animal cells, such as red blood cells, are put into a hypotonic solution, that would mean water would be moving into the cell by osmosis, and if enough water moves in, it can cause the cell to burst. Active transport is the movement of substances from a lower concentration to a higher concentration, and this requires metabolic energy and a carrier protein. So the process by which this happens is, first of all, it's always through carrier proteins only. It is not channel proteins for active transport. The molecule will bind to the carrier protein, and it can only bind to proteins which have a complementary receptor shape, so it is still selective. The ATP binds to the carrier protein as well from the inside of the cell, and it is hydrolyzed into ADP and PI. This causes the carrier protein to change shape, and therefore it releases the molecule to the other side of the membrane. The phosphate ion is then released, and the protein can return to its original shape, and the process can then happen again as long as there is plenty of ATP present. Co-transport is a type of active transport, and you learn about it across the course, but one of the key examples you learn about is the co-transport of glucose, or it could be amino acid, with sodium ions in the ileum. And to absorb glucose from the lumen of the gut, there has to be a higher concentration of glucose in the lumen compared to the epithelial cell. But there's usually more glucose in the epithelial cells, and that is why active transport and co-transport are required. So the first thing that has to happen is the sodium ions, which are shown in blue here, are actively transported out of the epithelial cell into the capillaries. And that will result in a lower concentration of sodium ions within the epithelial cells. And that creates this concentration gradient from the ileum to the epithelial cell. And therefore the sodium ions can diffuse down their concentration gradient into the epithelial cells. The proteins the sodium ions diffuse through, though, is a co-transporter protein. So either glucose or amino acids will also attach and are transported into the epithelial cell with the sodium ions, except the glucose is going in against its concentration gradient. The glucose, once in the epithelial cell, can then move by facilitated diffusion from the epithelial cell into the blood. Now, you also need to be aware that cells may be adapted for rapid transport across their internal or external membranes. So it could be that cells have an increased surface area, so things like microvilli, for example. Or there could be an increase in the number of protein channels and carrier molecules within the membrane as another way to increase the rate of transport. The last section of topic 2 is all about immunity, and we begin by looking at identifying self and non-self cells. So your body's immune system has cells to be able to identify the presence of pathogens and potential harmful foreign substances compared to your own body cells. And that's so it can destroy the foreign items and not your own cells. And those cells are called lymphocytes. But how can lymphocytes then actually distinguish between a pathogen and your own body cells? Well, each type of cell has specific molecules on its surface that identify it. And these molecules are usually proteins, so they'll have this unique 3D tertiary structure. And this is how they can identify whether it is an antigen from a pathogen or whether it is one of your self-antigens. If a non-self cell is detected, a response will be triggered to destroy that cell. And these different surface molecules enable them to identify pathogens, which we've mentioned, so bacteria, fungi, viruses, But it could also be cells from other organisms of the same species, and that can be harmful for individuals who have had organ transplants. It could also be abnormal body cells, for example, cancer cells. And finally, toxins, because some pathogens release toxins into the blood, such as the bacteria cholera. Now, an antigen is a molecule that generates an immune response, and this is because it triggers the lymphocytes. They are usually proteins, and they are located on the surface of the cells. Now, antigen variability is this concept that pathogens' DNA can mutate frequently. If a mutation occurs in the gene which codes for the antigen, then that means the shape of the antigen on the outside of the virus, the fungus, the bacteria will change. And if that happens, any previous immunity that you may have had to the pathogen is going to be lost. So it will no longer be effective, as all of the memory cells in the blood that you have will have a memory of the old antigen shape and not the new one. And this is known as antigen variability. The influenza virus mutates and changes its antigens very quickly, and this is why a new flu vaccine is created every year to account for that change. So looking at your immune response then, if a pathogen gets past the chemical and physical barriers, so for example your skin is a physical barrier and stomach acid is a chemical barrier, then your white blood cells are the next line of defense. Your white blood cells have a specific and also non-specific response. The phagocytes are a non-specific response, meaning that they will destroy any foreign item that they come across. Lymphocytes have a specific response though, and this is where the idea of detecting using antigens will come into it. So let's look at phagocytosis first of all. A phagocyte is a macrophage cell, which is a type of white blood cell, and they are found in the blood but also tissues. Phagocytosis, as we said, is a non-specific response. So any non-self cell that is detected will trigger the same response every time to cause the destruction of that object. So the stages of phagocytosis are, first of all, the phagocytes are in the blood and the tissues, and any chemicals or debris released by the pathogens or abnormal cells attract the phagocytes, and that will cause those white blood cells to move towards the cell. There are many receptor binding points on the surface of the phagocyte, which we can see here. They will attach to the chemicals or the antigen on the pathogen via the receptors. The phagocyte then changes shape to move around the pathogen and engulf it, and once engulfed, the pathogen is contained within a vesicle, which we call a phagosome. A lysosome within the phagocyte will then fuse with the phagosome and it will release its contents. And the contents is a lytic enzyme called lysozyme. And lytic enzymes are able to hydrolyze. So the result is that enzyme will hydrolyze and it breaks down the pathogen. Any soluble products are absorbed and used by the phagocyte, and anything else is released as waste and debris. So that is our non-specific response, but now if we look at the specific responses, which are by the lymphocytes, first lymphocyte we're going to look at are the T lymphocytes, or the T cells. So all lymphocytes are made in the bone marrow, but the T cells mature in the thymus, which is why they're called T cells. And this is classed as the cell-mediated response. So the first thing to be aware of is the cell-mediated response is specific to T cells responding to antigens on the surface of cells. So it involves antigen-presenting cells, or APC. An antigen-presenting cell is any cell that presents a non-self antigen on their surface. Infected body cells will present the viral antigens on their surface if they are infected. A macrophage, which has engulfed and destroyed a pathogen, will also present the antigens on their surface. Cells of a transplanted organ will have different shaped antigens on their surface compared to your own self cells. And cancer cells will have abnormal shaped self antigens as well. So all of these are presenting antigens on the cells, so they are antigen presenting cells and can therefore trigger an immune response. So the cell mediated response then, once a pathogen has been engulfed and destroyed by a phagocyte, the antigens are positioned on the cell surface. And that is why we'd now call that phagocyte an antigen presenting cell. Helper T cells, which are a type of T lymphocytes, have receptors on their surface that can attach and bind to the antigen on antigen presenting cells. And once those two cells are bound, it activates the helper T cell to start to divide by mitosis. And that then means we get a very large number of cloned cells. So essentially, this stage is to make sure you end up with lots of helper T cells which have the correct shape to bind on to that particular antigen. So you'll have lots of helper T cells within your blood that have different shaped receptors to be able to bind onto different shaped antigens. But if you have one collide, that will then activate that particular T cell with that particular shape receptor to divide by mitosis to make large cloned copies. Those cloned helper T cells then differentiate into particular types of T cells. Some remain as helper T cells, which then go on to activate the B lymphocytes. Some can stimulate macrophages to perform more phagocytosis. Some become memory cells for that particular shaped antigen. And some become cytotoxic killer cells or cytotoxic T cells. And what those cytotoxic T cells do is destroy the abnormal or infected cells that have the antigen on their surface. And they do this by releasing a protein called perforin, which embeds in the cell surface membrane to make a hole or a pore, so that any substance can now enter or leave the cell. Now this causes death because you can have lots of water move in and therefore the cell will burst, or you could have lots of water moving out and therefore the cell will actually shrivel up and die. This is most common in viral infections because viruses infect body cells of the host. Body cells therefore have to be sacrificed to prevent further viral replication. And this is actually why you get a really sore throat when you have a cold, because those cytotoxic T cells are destroying the infected body cells in your throat to try and prevent that virus dividing and spreading further in your body. Next then we move on to the B lymphocytes or the B cells. As we already said, all lymphocytes are made in the bone marrow, but B cells mature in the bone marrow as well, which is why they're called B cells. This is now called the humoral response, and this is the one that involves antibodies. Antibodies are soluble and transport in bodily fluids, and humour is an old term for body fluids, and that's why this is called the humoral response. There are approximately 10 million different B cells which have antibodies on their surface, complementary to 10 million different antigens. And antigens in the blood will collide with their complementary antibody on a B cell, and the B cell takes in the antigen by endocytosis and then presents it on its cell surface membrane. When that B cell collides with a helper T cell, that can then activate the B cell to go through clonal expansion and differentiation or clonal selection. So this is the step where there's that link between the cell-mediated response with the helper T cells binding via the receptor to the antigens on the B cells to activate them. Once activated, the B cells undergo mitosis to make large numbers of the cells which have that particular antigen on their outside. And these can then differentiate into plasma cells or memory B cells. The plasma cells will go on to make antibodies complementary in shape to that particular antigen. And the memory cells can divide rapidly into plasma cells if you are reinfected with the same pathogen and therefore antigen later on. And that means that you can make large numbers of the correct shaped antibody so rapidly that you should be able to destroy the pathogen before it causes any damage and symptoms. So the memory B cells can live for decades in your body, whereas the plasma cells are only short-lived. Memory B cells, though, cannot make antibodies, but they can divide by mitosis and then differentiate into plasma cells. So this is what results in the large numbers of antibodies being produced so rapidly that the pathogen is destroyed before any symptoms occur. And that's what it means when we say you are immune to a particular disease. It's because you have these memory B cells, so you can destroy the pathogen before it causes any symptoms. And that would be an example of active immunity. Primary and secondary response is referring to the number and the speed at which antibodies are produced when you are first exposed to an antigen compared to your second exposure. So the first time you're exposed to a new pathogen and therefore a new antigen, it takes a little bit longer for the antibodies to be produced because you have to go through that initial step of those 10 million different B cells colliding until you have the correct shape antigen antibody binding. So that can take a couple of days, and that's why you have a slower response in producing the antibodies, and also you don't actually make as many. But because memory B cells are made, if you are exposed for a second time, you will be able to create those antibodies very, very rapidly and in much larger numbers. And this is what is meant by the primary response and the secondary response. So if we look a bit more at antibodies, antibodies are an example of quaternary structured proteins because they're made up of 4 polypeptide chains. The part shown in purple is described as the variable region, and that is where you would have the part of your antibody that binds onto particular shaped antigens. The part in red is constant, so it would be the same for different antibodies. We can then see we have one longer chain, which is heavier, so we call it the heavy chain, and we have a shorter chain, which is called the light chain. And this bit here where it is variable, as we said, that is the antigen binding site. Now those antibodies are actually flexible, and that enables an antibody to bind to multiple antigens. So we can see we've got binding to 2 antigens here, and then this antibody is binding to 2 as well, and as a result they all end up clumping together, and we call this agglutination. The advantage of agglutination is if you've got a big clump of antibodies and antigens which are going to be attached to the pathogen, it makes it much easier for the phagocytes to locate and therefore destroy the pathogens more rapidly. So we've talked about active immunity. Passive immunity, though, is when the antibodies are introduced to the body. So you haven't made the antibodies yourself, you are just gaining the antibodies. So that means that the pathogen didn't cause the creation and differentiation of plasma cells or any memory cells, so you won't have any long-term immunity. Examples of passive immunity could be antibodies passed to a fetus through the placenta or through breast milk from the mother to their baby. Active immunity, as we said, is when the immunity is created by your own immune system following the exposure to the pathogen. But this can be either natural or artificial. And natural is following an infection where you've actually had that pathogen within your body. Artificial is following the introduction of a weakened version of the pathogen or antigens via a vaccine. So if we go through then how vaccines work. Small amounts of weakened or dead pathogen, or it could just be the antigens, are introduced either to the mouth or injected into the body. And exposure to those antigens activates the B cells to go through clonal expansion and differentiation. So that was the clonal selection part where we have mitosis occurring to make large numbers of those B cells. Those then differentiate into plasma cells and memory B cells. As we said earlier, the plasma cells will make antibodies, But the key bit of importance for a vaccine is the fact that B memory cells will also be produced, and those can stay in the blood for years. So that means that if you are infected with the actual pathogen, those memory B cells, if they collide with the antigen, will divide rapidly into plasma cells when you are reinfected, and therefore you create large numbers of antibodies so rapidly that you shouldn't get the symptoms of the disease. Or if you do, they'll be very, very mild, and you should be able to overcome the disease more quickly. Now, not everyone is able to take vaccines for various reasons, but the concept of herd immunity is if enough of the population are vaccinated, the pathogen cannot spread easily amongst the population anymore. So this provides protection for those who aren't vaccinated. For example, if you already have another illness, which means it'd be too dangerous for you to have the vaccine, if you have a lowered immunity, if you're too young, and sometimes if you're pregnant, you're not able to take vaccines as well. Now, the structure of HIV is made up of 4 key components. We have the core, and that is the genetic material, which is RNA, and the enzyme reverse transcriptase. And this is needed for the viral replication when it's in the host. You also have a capsid, which is an outer protein, which you can see just here. You then have the envelope, which is an extra outer layer made out of the membrane taken from the host cell membrane. And then we have protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cells. So the way that HIV replicates in helper T cells is that first of all, it's transported in the blood until it attaches onto a CD4 protein, which is one of the attachment proteins on the outside of helper T cells, which we can see just here. The HIV protein capsule then fuses with the helper T cell membrane, enabling that RNA and enzyme to be injected or enter into the host's cell. The HIV enzyme reverse transcriptase copies the viral RNA that's been injected into a DNA copy and moves to the helper T cell nucleus, and this is why it's called a retrovirus. Here, the mRNA is transcribed and the helper T cell starts to create viral proteins to make the new particles. Someone's described as HIV positive when they are infected with HIV. AIDS is when the replicating viruses in the helper T cells interfere with the normal functioning of the immune system. So someone who is HIV positive doesn't necessarily have AIDS. With the helper T cells being destroyed by the virus, the host is unable to produce an adequate immune response to other pathogens and is left vulnerable to infections and cancer. It is the destruction of the immune system that leads to death rather than the HIV directly. So the very final thing is this concept of monoclonal antibodies. Mono meaning one and clonal meaning identical. Antibodies are proteins, which we looked at, and they have these binding sites complementary in shape to a particular antigen. And this has been manipulated to create monoclonal antibodies. And that can be for medical treatments, medical diagnosis, it's used in pregnancy tests, it's also used in tests such as drug testing and testing for other viral diseases. So targeted medication, we're going to go through that example first. This is an example of direct monoclonal antibody therapy. So what we mean by that is some cancer can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of the cancer cells. The antibodies are given to the cancer patient and they will then attach onto the cancer cells only. While the antibodies are bound to the cancer cells, this prevents chemicals binding to the cancer cell, which will enable uncontrolled cell division. Therefore, the binding of these monoclonal antibodies prevent the cancer cells from growing. And as they're designed to only attach to the cancer cells, they're not going to harm any other normal cells. Indirect monoclonal antibody therapy can also be used to treat some cancers. The monoclonal antibody is created exactly the same, except there will be a drug attached to them. Indirect monoclonal antibody therapy starts with the same process of creating a monoclonal antibody which has a binding site complementary in shape to the antigens on the outside of cancer cells. But this time, a drug is also attached to the antibody. The cancer drugs are therefore delivered directly to the cancer cells and can destroy those cells only. And this reduces the harmful side effects that traditional chemotherapy and radiotherapy can produce. And this is sometimes called a bullet drug because it's being directed to just one particular site. Medical diagnosis is another use, and as we said, this could be in pregnancy tests, viral tests, so influenza, could be in bacterial tests as well, cancer tests. Most recently, we've seen it in COVID-19 lateral flow tests as well. And these use the ELISA or the ELISA test. So the ELISA test, which stands for Enzyme-Linked Immunosorbent Assay, or ELISA, uses two antibodies. So first of all, the first mobile antibody, which is complementary to the antigen being tested for and has a coloured dye attached to it, we can see just here. Part C, we have a second antibody, which is complementary in shape to the antigen as well, but this one is immobile, so it's attached to this fixed position. There is then a third antibody, which is immobilized at point D, and it is complementary in shape to the first antibody. So what this means is, if you are pregnant, if we think about this in terms of a pregnancy test, you contain the hormone HCG within your urine. That will then bind to the antibody, and any antibodies with the HCG hormone bound to it will then attach to the immobilized antigen at point C. And therefore, you get a coloured blue line at this point because we have that blue bead held in that fixed position. You should always have a second blue line, whether you're pregnant or not, at position D though, because this second immobilized antibody is just complementary to the shape of the first one. So it's basically just a control to check that you do definitely have that first antibody in your sample and it is able to move along the test kit. Now another test we can look at is this one here for the ELISA test, where you would add the test sample from a patient to the base of a beaker, wash to remove any unbound test sample. Then we add an antibody complementary in shape to the antigen you are testing the presence of in the test sample. Again, we then wash to remove any unbound antibody. Add a second antibody that is complementary in shape to the first antibody and binds to the first. The second antibody we can see here has an enzyme attached to it. Again, we have to rinse just to make sure if there's any unbound antibody, those are taken off. And then the substrate for the enzyme, which is colourless, is added. And this substrate produces a coloured product when that enzyme is present. So the presence of colour indicates the presence of the antigen in the test sample, and the intensity of the colour indicates the quantity present. Now there are some ethical issues surrounding monoclonal antibodies, and that is because within the creation of monoclonal antibodies, you do require animals such as mice to produce the antibodies and tumour cells. And that can lead to ethical debates as to whether the use of animals is justified to enable the better treatment of cancer in humans and to detect diseases, because this will cause discomfort, harm and death to the animals that are being used.
So we begin with surface area to volume ratio. And this is where we have this particular formula of whatever the surface area of an organism is, you divide it by its volume. And the relationship between how large an organism is, or it could be the different shapes it has, are linked to the surface area to volume ratio.
So this concept has a big role within this topic, where we look at how different cells or even organ systems have adaptations to maximize gas exchange and other types of transport across surfaces. So here we just have an example where we can see this pattern of the larger an organism is. Now in this case we've just got cubes to demonstrate, but the larger an organism is, the surface area divided by the volume, or the surface area to volume ratio, will decrease. So what that means in terms of biology is small organisms, like single-celled organisms like amoeba, have very large surface areas compared to their volume. And that means that they don't have to have any additional special adaptations to meet their needs in terms of transport across their surface. They can just diffuse gases across their surface, and because they've got such a large surface area compared to their volume, they'll be able to get enough gases in and out to meet their metabolic needs. However, larger organisms, because the surface area is so much smaller compared to the volume, that means that if we were just to get gases into the organism through simple diffusion, the cells right in the centre of the organism are so far away that the gases wouldn't be able to reach there in sufficient time for their metabolic needs. Now, on top of that, larger organisms typically have higher metabolic needs as well. And what that means is the chemical reactions within a cell are happening faster. So that is another reason why large organisms have to have adaptations for mass transport or just for transport across cells. And that's what most of this topic is looking at, the adaptations that organisms have for their exchange surfaces to make exchange more efficient. So some of the key adaptations are the villi and the microvilli. So within the small intestines, those two particular types of tissue and then cells to maximize the absorption of digested food. The alveoli and bronchioles, so they're involved in gas exchange. Spiracles and tracheoles involved in gas exchange, but that's for terrestrial insects, whereas alveoli and bronchioles are for mammals. Gill filaments and lamellae is the gas exchange adaptation surface for fish. And then we'll be looking at plants as well. They exchange gases through the stomata on the leaves, but we'll be looking at some of those adaptations as well. Now, in addition to that, when we get to the idea of the mass transport as well, we'll see a link to gas exchange, whereby most of these gas exchange surfaces have a capillary network nearby as well, with the exception of plants because they don't have blood. So some of the key terms linked to the gas exchange and ventilation topic that you need to be familiar with are breathing. So this is the movement of air in and out of the lungs. This is different to respiration because respiration is a chemical reaction that results in the release of energy in the form of ATP. Ventilation is the scientific term for breathing, so we'll be using the term ventilate quite a lot in this video. And gas exchange is the diffusion of typically oxygen and carbon dioxide in and out of cells. Now, it's not just the alveoli, that is just for mammals. So those are the key terms just to be familiar with, and these are the key structures that you need to know for the human gas exchange system. So the alveoli, bronchioles, bronchi, trachea and lungs, which are all labelled here. So if you need to pause just to note that down, then pause that just here. So we have a look at how humans ventilate. It involves the muscle, which is the diaphragm. It also involves antagonistic muscles, which are surrounding the ribs. And this is where we have the external intercostal muscles, but we also have internal intercostal muscles. Now, antagonistic means that as one muscle is contracting, the other relaxes. And we'll see that in our intercostal muscles, how the internal and external will always be doing the opposite. And that is what controls the movement of the rib cage in and out to help with ventilation. So a bit more about these internal and external intercostal muscles. Antagonistic muscles always occur as pairs, and the external intercostal muscle on the outside layer of the ribs and they will contract and cause the rib cage to move out and air to flow in, or inspiration. The internal intercostal muscles, you can kind of see as it moves around 3D, they'd be on the inside layer around the ribs. And when these ones contract, that will pull the rib cage closer inwards, and therefore the air will flow out, or you'll be expiring. But just to have a summary then of what is happening to all of the muscles that are controlling the movement of the rib cage, and therefore whether you're inhaling or exhaling, or inspiring or expiring. So when you are breathing in, that is when your external intercostal muscles will be contracting, and that pulls the rib cage up and outwards. That means that the internal intercostal muscles are doing the opposite, they're relaxing. The diaphragm muscle contracts, and when that contracts, it causes it to move downwards and it becomes more flat. Now, as a result, that means that the volume of the thorax will be larger, so the volume increases. And by that, we don't mean the volume of gases, we mean the volume as in the cavity space, so the available volume, the actual space available. And because the lung volume, the thorax space, is larger, that causes the pressure to drop, and because you now have a lower pressure within the lungs compared to outside, air is going to flow in. So that is what happens when you breathe in. Now when you exhale, it's exactly the opposite. So this time the external intercostal muscles relax, the internal will contract, and that pulls the rib cage down and back in. The diaphragm relaxes, which causes it to dome upwards. And as a result, the actual volume in the thorax, that space, is smaller. And because we have a smaller space or a smaller volume, that causes the pressure to increase. And that is why the air is forced out of the lungs. Now, the pulmonary ventilation calculation is one that you do need to know. And it's the total volume of air that's moved into the lungs during 1 minute. And the units would be decimeters cubed for volume and per minute. So to work this out, you would be doing the tidal volume times the ventilation rate. So tidal volume is the total volume of air, and the ventilation rate is how many times you breathe per minute. So then if we go on to the gas exchange and look at the alveolar epithelium. Once the gases are in the alveoli, so as a result of ventilation, this is where gas exchange occurs, and it's between the epithelium and the blood. Alveoli are tiny air sacs, and you have around 300 million in each lung. And that's actually why lung tissue floats compared to, let's say, muscular heart tissue, which would sink. So all of those alveoli air sacs make it float because it's so full of air. Now, the alveoli epithelium cells are also very, very thin. And that is to minimize the diffusion distance. So we can see here there's just one layer of cells and they're a thin layer. Each alveolus is surrounded by a network of capillaries, and this helps to maintain the concentration gradient, because as soon as that oxygen diffuses into the blood, it is then carried away in the capillaries and replaced by deoxygenated blood. Terrestrial insects, they do have a large surface area to volume because they're small. However, the issue they have to overcome is the fact that they have a waterproof exoskeleton. And that's an advantage to prevent water loss, but because it's made up of this hard fibrous material, it also means gases can't exchange across it. Now, insects don't have lungs, but they do have a tracheal system, which is where ventilation and gas exchange occurs. So their tracheal system then, we have a look at how this differs to the human lungs. First of all, it includes a trachea, tracheoles and spiracles. The spiracles we can see here, they're the tiny holes, they're round valve-like openings that run along the abdomen, and this is where oxygen and carbon dioxide are going to be entering and leaving. So that is instead of like the nose or the mouth for humans. And those spiracles are going to be attached to trachea. So that's the next structure. And the trachea are tubes. They have rings within them to strengthen the tubes to stop them from collapsing, and that keeps the tubes permanently open. And then the trachea branch into smaller tubes, which are deeper in the abdomen of the insect, and those are called the tracheoles. And those extend throughout all of the tissue in the insect to deliver oxygen to all of the respiring cells. So here we have the spiracles connected to the trachea, and then the tracheal systems branching all the way through into the centre where the abdomen is and to all of the respiring tissues. Now there's three methods of moving gases into that tracheal system. The first method is gases can just move in by diffusion. And that is because when the cells within the insect are respiring, they're using up oxygen, producing carbon dioxide, and that creates a concentration gradient from the tracheoles compared to the atmosphere. So some of the gases will simply diffuse in and out. The second method of gas exchange is mass transport. And this is where it's slightly similar to ventilation in humans, in that the abdominal muscles can contract and relax. And that is a way to ventilate, to move gases en masse in and out of the tracheal system. The final method is when the insects are in flight, the muscle cells will actually start to respire anaerobically. And when they do that, they produce lactate or lactic acid. And this lowers the water potential of the cells, and as a result, it draws water into the cells from the tracheoles. That decreases the volume in the tracheoles, and as a result, more air from the atmosphere is drawn in. So overall, the adaptations that terrestrial insects have for gas exchange are the fact that they have a large number of these fine tracheoles providing a large surface area. The walls of the tracheoles are very thin, and there's a short diffusion distance between the spiracles and the tracheoles, so that will speed up the rate of diffusion. And lastly, they use oxygen and they produce carbon dioxide, and that is how the steep concentration gradient is maintained. Now, the next challenge that the terrestrial insects have to overcome is limiting the amount of water that is lost at these gas exchange surfaces, because a gas exchange surface is also ideal for evaporation, and they do not want to be losing excess water. So here are the ways that water loss is prevented. Number one is the surface area where gas exchange actually happens is very small. So it's not the entire insect, we're just looking at the spiracle itself and then the tracheal system. That is a very small area compared to the whole volume of the insect, and therefore it's going to limit evaporation. The rest of the insect has this waterproof exoskeleton, and the spiracles can actually open and close, a bit like stomata on a plant, to reduce water loss also. So next is gas exchange in fish. Now fish are waterproof and they have a small surface area to volume ratio. So those are the two reasons why fish have to have a gas exchange surface with adaptations. And for fish, it's gills. Now fish obtain their oxygen from the water, but there is 30 times less oxygen dissolved in water than there is available in the atmosphere, in the air. So they have to have an additional adaptation to help maintain the diffusion and concentration gradient across their gills. So just as a reminder, for all of these gas exchange surfaces, they have to have these three features: a large surface area compared to the volume, a short diffusion distance, and they have to have a mechanism to maintain the concentration gradient. Now those features can be used to work out the rate of diffusion using Fick's law. And this is just here. So diffusion is proportional to the surface area times the difference in concentration divided by the length of diffusion pathway. So you could get questions linked to this, testing that math skill. But let's have a look then at the fish gills to see how they have these three features maximised in their adaptations. So we're going to have a look at the fish gill anatomy. So there are 4 layers of gills on both sides of the head, and the gills are made up of stacks of gill filament. Each gill filament is covered in gill lamellae, and these are positioned at right angles to the filament. So this here is the lamellae, and this part is the filament. Now, because there are so many, that creates the large surface area. So that's the first adaptation ticked off. Because there's so many gill lamellae and gill filaments, it creates that large surface area. Now, when a fish opens its mouth while it is swimming, water rushes in and then actually comes out the side of their head, which means that the water rushes over the gills. So the water enters here and then it goes over the gills on both sides of the head, where they have these 4 layers of gills with the gill filaments and the gill lamellae. So the next thing is looking at the additional adaptations. We've talked about the large surface area to volume ratio, but the next thing is the fact that there is a short diffusion distance, and that is because of the network of capillaries in every gill lamellae. So we're zooming in here on the gill lamellae, and there is our network of capillaries within each one, and that provides a short diffusion distance. How the concentration gradient is maintained is by something called the counter-current flow mechanism. So the counter-current flow mechanism is where water flows over the gills in the opposite direction to which the blood is flowing through the capillaries. The reason this is an advantage is it makes sure that the equilibrium of the concentration of oxygen is never reached. And because we don't have equilibrium being reached, that means that oxygen is able to continue diffusing from the water into the capillaries in the gill lamellae across the entire gill lamellae. And that's in bold and underlined because that is the key marking point. Now that's demonstrated in these 2 diagrams here where we look at concurrent flow in comparison to countercurrent flow. So in concurrent, not co-current, you'd have the water and the blood flowing in the same direction. So about halfway across the lamellae, you'd be reaching equilibrium, and therefore you wouldn't have any more diffusion of gases. Whereas here we have the water and the blood flowing in opposite directions, which means you should never actually have equilibrium, and there will always be a higher concentration of oxygen in the water compared to the blood, and that is why we maintain the concentration or the diffusion gradient across the entire gill lamellae. So lastly is looking at gas exchange in leaves. So you could be asked to look at some of the structures in the leaf. So we have the tissue layer palisade mesophyll, which is where photosynthesis mainly happens. And then we have the spongy mesophyll, where there's lots of air spaces. And we have the stomata, which is where the gases actually diffuse in and out of. So oxygen will actually diffuse out of the stomata if it's not being used in respiration. And carbon dioxide diffuses in because it's needed for photosynthesis. To reduce water loss by evaporation, stomata close at night when it's dark, and they'll open in the daytime when it's bright. And this is linked to the idea of photosynthesis because it's a light-dependent reaction. Now there are compromises though, and in particular linked to xerophytic plants, which are plants that are adapted to survive in environments with very limited water. So they have adaptations to minimise water loss because there's such little water available already. And over here, looking at this marram grass under the microscope, it demonstrates some of those adaptations really well. So the leaves aren't actually flat, they roll up, and we have stomata which are really deep and sunken in, and we have lots of tiny hairs sticking out. Now, all of those adaptations are to reduce water loss, because any water that is evaporating out is going to get trapped on these hairs, and the fact it's curled in, it gets trapped in this space. So it makes this area very, very humid, and if it's very humid, that will reduce further evaporation. Now, in addition to that, they also have a thicker cuticle to reduce water loss, and we can't see it on this particular image, but they do have a longer root network to be able to reach further distances to try and find any available water. So we now move on to digestion and absorption. And during digestion, large biological molecules are hydrolyzed into smaller soluble molecules which can be absorbed across the cell membranes. Now there's 3 biological molecules that you need to know the digestion of, and that's carbohydrates, lipids and proteins. So with the carbohydrates, there's actually more than one enzyme that's needed to hydrolyse them from the polysaccharides to the monosaccharides. We have amylases and membrane-bound disaccharidases. So digestion begins in the mouth. It will then continue in the duodenum, and then it's completed in the ileum. And the first thing is the amylase that is produced by the salivary gland is going to be doing the digestion in the mouth. But you also have amylase produced in the pancreas, and that is going to be releasing the amylase into the duodenum. And this hydrolyzes the polysaccharides into the disaccharide maltose, and that is by breaking the glycosidic bonds between the molecules in the polysaccharide. Then we have sucrase, and it could be lactase as well, which are membrane-bound enzymes that hydrolyze sucrose and lactose into monosaccharides. You could also have maltase, which would be hydrolyzing maltose into glucose. For proteins, these are large biological molecules, they're polymers, and they are hydrolyzed by three enzymes. We have the endopeptidases, which hydrolyze the peptide bonds between amino acids in the middle of a polymer chain. We have the exopeptidases, which are going to do hydrolysis and break the peptide bonds between amino acids at the end of the chains. And then the membrane-bound dipeptidases will be breaking the peptide bonds between two amino acids. So it hydrolyzes those dipeptides. Now, where this is actually happening is firstly in the stomach. So that's where it begins. It continues in the duodenum, and then it's fully digested within the ileum. Lipids are digested by lipase or lipase, and also through the action of bile salts. Now, lipase is produced in the pancreas, and it can hydrolyze the ester bonds in triglycerides to form monoglycerides and fatty acids. What the bile salts do after they've been produced by the liver is emulsify lipids to form tiny droplets called micelles, and these increase the surface area available for the lipase to bind onto and therefore act on. So they're going to be making this digestion more efficient. So we can see here the physical and the chemical digestion. Physical is the emulsification and that micelle formation. Chemical digestion is the action of the lipase. So the physical, this is where the lipids are coated in bile salts to create an emulsion. Many of those small droplets of lipids provide a larger surface area to make hydrolysis faster. And as we said, the chemical is just the action of lipase. Now, a micelle is a vesicle formed of fatty acids, glycerol, monoglycerides and bile salts, but we're going to have a look at how that aids in the absorption of lipids. So lipids are digested into monoglycerides and fatty acids by the action of lipase and the bile salts, and that is how we create these tiny structures, which are the micelles. When the micelles encounter the ileum epithelial cells, due to that non-polar nature of the fatty acids and monoglycerides, they can simply diffuse across the cell surface membrane to enter the cells of the epithelium. Once in the cell, these will then be modified back into triglycerides inside of the endoplasmic reticulum and the Golgi body. And then they can form vesicles and be released from the cell into the lacteal and be transported around the body. So absorption in mammals is taking place in the ileum. And this is where it links to the villi and the microvilli and the adaptations that these structures have. So the ileum wall is covered in villi, and they have thin walls surrounded by a network of capillaries. And they also are covered in epithelial cells that have even smaller microvilli on them. And that is creating that large surface area. The fact that the walls are so thin is providing a short diffusion distance, and that network of capillaries is maintaining the concentration gradients. Now, this actually links to as well topic 2 when you learnt about co-transport. And that is because the monosaccharides like glucose and amino acids can only be absorbed by active transport in the form of co-transport. And that is because there's usually a higher concentration of either amino acids or glucose already in the epithelial cell. So in order to be able to move even more from the ileum into the epithelial cell, we require this co-transport. But that theory is covered in topic 2. Now, hemoglobin is involved in the mass transport of oxygen around the body. And it's an example of a quaternary structure protein because it's made up of 4 polypeptide chains. You also have a range of different types of hemoglobins. One that we'll be looking at is myoglobin, which is found in the muscle tissue in vertebrates and also in fetuses. The oxyhemoglobin dissociation curve is a way to look at how hemoglobin behaves in different conditions. Oxygen is loaded in regions with a high partial pressure of oxygen. So what that means is when hemoglobin is in areas with a lot of oxygen available, such as the alveoli, it will be able to pick up lots of oxygen. In regions with a low partial pressure of oxygen, for example respiring tissues, hemoglobin unloads the oxygen. And that is how we get the shape of this oxyhemoglobin dissociation curve. We can see at the different partial pressures how saturated hemoglobin is with oxygen. And the more saturated it is, that means it must have loaded up with more oxygen. The less saturated it is, it must have unloaded oxygen, and that's why it's not holding very much anymore. So we can see here we've got our respiring tissues compared to the alveoli. Now this graph also demonstrates cooperative binding. And this cooperative nature of oxygen binding to hemoglobin is due to the hemoglobin changing shape when the first oxygen binds. It then makes it easier for the subsequent oxygens to bind. Now you can also see dissociation curves to demonstrate the Bohr effect. And the Bohr effect is when a high carbon dioxide concentration or partial pressure causes the oxyhemoglobin curve to shift to the right. The affinity for oxygen decreases, and that is because the acidic carbon dioxide changes the shape of the hemoglobin slightly, and therefore it means the hemoglobin behaves differently and it's more likely to unload the hemoglobin even at the same partial pressures. So we can see here we've got 3 curves at different pHs, and the lower the pH, the more carbon dioxide there would be present. So we can see here at pH 7.6 compared to, let's say, 7.2, the curve has shifted to the right, and even at the same partial pressure, so we'll pick 20, we can see that the saturation is only about 25%, but for this one, it's just under 60%, and that is because of this Bohr effect. So low partial pressure of carbon dioxide would typically be in the alveoli because you are exhaling that carbon dioxide. A high partial pressure of carbon dioxide would be at respiring tissues because carbon dioxide is produced in respiration. Now that would be an advantage because it means that hemoglobin will behave differently, it will unload oxygen more readily, and therefore it's unloading the oxygen at the respiring tissues. Now, different animals have hemoglobin adapted to their particular needs and environments also. And that is one thing that you could get application questions on. So a fetus, and this is a human fetus, they will have myoglobin or fetal hemoglobin, and the fetal hemoglobin has an even higher affinity for oxygen even at the same partial pressures compared to adult hemoglobin. And that's an advantage because it means that as the blood is circulating through the umbilical cord, the fetus's hemoglobin is able to load the oxygen off of the mother's adult hemoglobin. Llamas are found at high altitudes where we have very, very low partial pressures of oxygen. So for the llama, we can see that they also have hemoglobin that has a higher affinity for oxygen, even at lower partial pressures. So we can see that would mean that if there isn't very much oxygen available, which there wouldn't be at a high altitude, then the hemoglobin is still able to load up. Animals like doves, for example, their needs are that they need more oxygen to match their faster metabolism because they're flying so much, they need oxygen for the muscle contraction. So the hemoglobin of a dove, the curve actually is shifted to the right compared to human hemoglobin. And that means that the hemoglobin has a lower affinity for oxygen, and therefore it will more readily unload the oxygen which is needed for respiration. Earthworms are a different example, so they will be underground a lot where there's very low partial pressures of oxygen. So they have hemoglobin that has very high affinities even at low partial pressures, so that their hemoglobin can load up with whatever oxygen is available. In mammals, the circulatory system is described as closed and double. Closed meaning that the blood remains within the blood vessels the entire time, and double referring to the fact that the blood passes through the heart twice in each circuit. So there is one circuit that delivers blood from the heart to the lungs, and the other circuit delivers the blood from the heart to the rest of the body. Mammals are required to have a double circulatory system to manage the pressure of blood flow. The blood flows through the lungs at a lower pressure, and this is to prevent damage to the capillaries in the alveoli, but it also means that the blood will flow at a slower speed, so there is more time for gas exchange. The oxygenated blood from the lungs then goes back to the heart and it's pumped out at a higher pressure to the rest of the body. And this is important to make sure that the blood is able to reach all of those respiring cells in the body. Now, the key blood vessels that you need to know about are, first of all, the coronary arteries, and these are the arteries that cover the heart itself to supply the heart muscle or cardiac muscle with oxygenated blood. You also need to know the 4 blood vessels that are delivering blood into and out of the heart: the vena cava, aorta, pulmonary artery, and the pulmonary vein. You need to know the blood vessels that deliver blood to the lungs and carry it away, so the pulmonary artery and the pulmonary vein. So any time that you see pulmonary, that is referring to the lungs. And you can see here that the pulmonary artery is carrying blood away from the heart to the lungs. The lungs will then oxygenate the blood, and the pulmonary vein is delivering that blood back into the heart. The kidneys, we have the renal artery and the renal vein. So where you see the word renal, that is referring to a blood vessel attached to the kidneys. Those major blood vessels are connected within this double circulatory system via arteries, arterioles, capillaries, and veins. The cardiac muscle has a range of special features. Now, the walls of the heart have a very, very thick muscular layer so that it can contract with high force to deliver high pressure blood to all of the body cells. Now, the unique properties that the cardiac muscle has is, first of all, it's myogenic, and that means it can contract and relax without nervous or hormonal stimulation. It also never fatigues. So as long as it has a constant supply of oxygen and glucose, it will be able to respire aerobically. The coronary arteries we can see here, they are what supply the cardiac muscle with this oxygen and glucose so that it never fatigues. They branch off from the aorta, which we can see here. And if one of those coronary arteries was to become blocked, that would then mean that the heart muscle or the cardiac muscle wouldn't be receiving the oxygen or glucose, therefore the cardiac muscle wouldn't be able to respire and it would stop contracting. And that would cause a myocardial infarction, or in other words, a heart attack. So you need to know some of the key structures of the heart. First of all, there are 4 chambers. We have 2 atria at the top, and we have the 2 ventricles at the bottom. The atria have thinner muscular walls, and that's because they don't need to contract with as much force because they're only delivering the blood from the atria into the ventricles. They also have elastic walls so that they can stretch when the blood is entering. The ventricles have much thicker muscular walls, and that is so they can contract with more force and pump the blood out at higher pressure because they are carrying the blood further distances, either to the lungs or to the rest of the body. Now the right ventricle is pumping the blood to the lungs, and that is at a lower pressure, as we said, to prevent damage to the capillaries and go at a slower speed. So comparatively, the right ventricle wall has a thinner muscular layer. The left ventricle has a much thicker muscular layer in its wall because it has to contract with more force to pump the blood at high pressure around the body. Highlighted here we have the 4 key blood vessels. We've got the aorta and the pulmonary arteries, the vena cava and the pulmonary veins. But for some of them there's actually multiple. So we can see we have the superior vena cava and the inferior vena cava. We also have the pulmonary artery, which is carrying the blood away from the heart to the lungs, but we have one coming out of the left and one out of the right. Same with the pulmonary veins, which is carrying blood into the left atrium from the lungs. We have one on the left side, one on the right side. And the reason for both of those is we have a right lung and a left lung. The aorta is carrying blood from the left ventricle to the rest of the body. Now, some of the ways just to try and remember what each of these are doing are, first of all, if you see veins, veins are carrying blood into the heart. And vena cava means vein, and cava means body. So that is carrying the deoxygenated blood from the body into the right atrium. Pulmonary, we said, means lungs, and again it's a vein. So it's carrying oxygenated blood from the lungs into the left atrium. Arteries, think A away, it's carrying blood away from the heart. So pulmonary artery is carrying deoxygenated blood from the right ventricle to the lungs. And the aorta is the major artery, which is carrying oxygenated blood from the left ventricle to the rest of the body. Now the valves that you need to know about are the semilunar valves, which are in the aorta and the pulmonary artery, as well as the atrioventricular valves, which are between the atria and the ventricles. Sometimes you'll see those called the bicuspid and the tricuspid valves. And we can see these labelled here on the diagram of the heart. Now valves are to prevent the backflow of blood, and they do this by only opening when the pressure is higher behind the valve compared to in front. So if the pressure is higher in front, it causes the valve to shut, and that is how it stops the blood from flowing backwards. The septum is running through the middle of the heart to separate the blood on the deoxygenated and the oxygenated side, and that's to help to maintain a high concentration of oxygen in the oxygenated blood to make sure that these diffusion gradients are maintained so that diffusion can occur at respiring cells. Looking at the blood vessels that are connecting all of the major blood vessels together, we have the arteries, arterioles, capillaries and veins. Arteries, as we said, think A away, they carry blood away from the heart towards the arterioles. The arterioles are smaller than arteries and connect to the capillaries. The capillaries connect the arterioles to the veins, and the veins will then carry blood back into the heart. So we can see here our arteries connected to our arterioles. We then have this capillary bed or network of capillaries connecting to the venules and then the vein. So the structures of the arteries and vein are quite different because of the fact that one is carrying blood away from the heart and one's carrying it into, so the pressure of the blood they're carrying will vary. So arteries have a much thicker muscular layer compared to the veins, and that is so that constriction and dilation can occur to control the volume of blood flowing through them. They also have a thicker elastic layer than the veins, and that's to help maintain the blood pressure. So the walls can actually stretch and recoil in response to the heartbeat. So overall, the thickness of the wall is thicker because of those two thick layers, but also it stops the blood vessels from bursting at that high pressure. So we can see here how thick those walls are. They also don't have any valves. So in comparison, veins have a much thinner muscular layer, thinner elastic layer and thinner walls in general, and that is because the blood is at lower pressure, so they're not at risk of bursting. However, because the blood is at lower pressure, they do require valves to help prevent the backflow of the blood, to make sure that the blood is going to be pumped back into the heart. The capillaries are very, very narrow in diameter, and this is to make sure that the blood speed is going to slow right down as it's passing through the capillaries. And that is to allow time for gas exchange and tissue fluid formation in the capillaries. So we've already talked about the arteries and the veins, but if we now add in the arterioles and capillaries, the arterioles are thicker than in the arteries, the muscular layer, and that is to help restrict the blood flow to the capillaries. The elastic layer is thinner than the arteries, and that is because the pressure is now slightly lower. And overall, the wall is thinner compared to the arteries because the pressure is slightly lower, but they still don't have valves. Capillaries do not have any muscular or elastic tissue layer. They are only one cell thick, and that is to make sure there's a really short diffusion distance, because the function of the capillaries is that is where exchange of materials between the blood and the cells occur. The cardiac cycle is split into 3 stages. Now people pronounce this differently, so I'm going to say both diastole or dystole, atrial systole or atrial systole, and ventricular systole or systole. And we're going to go through what happens at each stage. So in diastole, the atria and ventricle muscles are relaxed. And this is when blood will enter the atria through the vena cava and the pulmonary vein. And the blood flowing into the atria then causes an increase in the pressure because we've now got a larger volume of liquid there. Then we get atrial systole occurring, and that is when the atria muscles contract, and that increases the pressure even more in the atria. And because we now have this high pressure, that causes the atrioventricular valves to open, and blood moves from the atria into the ventricles. And at this stage, the ventricle muscles are relaxed. We have ventricular diastole. The last stage then we have is ventricular systole, which is when the ventricle muscles contract. And that happens after a short delay, and it will increase the pressure beyond that of the atria, which causes these atrioventricular valves to shut. But when they've contracted high enough to increase the pressure above that of the atria and pulmonary artery, the semilunar valves will open, and that causes blood to be pumped out of those 2 blood vessels. You could be asked to calculate the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute, and it can be calculated by doing the heart rate times the stroke volume. And the heart rate is beats of the heart in 1 minute. Stroke volume is the volume of blood that leaves the heart each beat, and that is in decimeters cubed. Tissue fluid is a fluid that contains water, glucose, amino acids, fatty acids, ions and oxygen, and it's the liquid that is forced out of the capillaries to bathe the cells, to make sure the cells are getting all of the minerals, nutrients that they require from the blood. Now it's formed due to the fact that capillaries have very, very small gaps between each of the cells that make up the capillary walls. And as the blood enters the capillaries from the arterioles, the smaller diameter results in a very, very high hydrostatic pressure of the blood. And because we have this high hydrostatic pressure and tiny gaps between the cells that make up the capillary walls, water and small molecules are forced out. So glucose, amino acids, fatty acids, ions, oxygen are all forced out of the capillary to the surrounding cells. And that is called ultrafiltration. So the molecules that can be forced out, we just went through all of those, but anything that is too big cannot get out of those tiny gaps between the capillary cells, and that would be things like the red blood cells, platelets, and large proteins. So those will remain in the blood. And the fact that they remain in the blood is actually what enables the liquid from the tissue fluid to be reabsorbed again. So towards the venial end of the capillary, which is over here, the hydrostatic pressure has decreased, and that's because so much liquid has been forced out. The pressure within the capillary has now dropped by the time we've got to the venial end of the capillary. However, we don't have as much liquid, but we have lots of those large proteins that have remained behind, which has lowered the water potential of the blood in the capillary compared to the water potential in the liquid surrounding the cells, which is the tissue fluid. And as a result, that liquid is going to, or the water is going to re-enter by osmosis, and it will carry with it waste that has dissolved in that tissue fluid that was released from the cells. So things like carbon dioxide and urea. So that gets absorbed into the capillaries by osmosis, and then it'll be transported around the body to be removed as waste. Now, not all of the liquid will be reabsorbed by osmosis, because eventually equilibrium will be reached and the water potential inside of the capillary and of the tissue fluid will be the same. So any of that water that doesn't get reabsorbed by osmosis will enter the lymphatic system instead, and then when we have it transported around the lymphatic system, it will eventually get to one of the lymph vessels near the heart where that liquid is drained into the blood again, and that is how we don't run out of liquid in the blood. So the last bit of topic 3 is mass transport in plants. And we start with the mass transport of water, looking at, first of all, transpiration. So transpiration is the loss of water vapour from the stomata. So water evaporates out of stomata on the leaves. And there are 4 key factors that affect the rate of transpiration. First of all, light intensity. The more light there is present, the more stomata will open, and therefore there's a larger surface area for evaporation. If it's hotter, that means that the water molecules will have more kinetic energy, and therefore they'll be moving faster and evaporate at a faster rate. With humidity, the more water vapour that is in the air, that will actually make the water potential more positive outside of the leaf, and it reduces the water potential gradient. So what that means is the more humid it is, the less transpiration there will be occurring. With wind, because the wind is carrying away air that contains water vapour, the wind will actually be maintaining the water potential gradient. So the more windy it is, the faster the rate of transpiration. So how water actually moves from the roots all the way up the xylem to transpire out of the stomata links to this idea of the cohesion tension theory. So water has to move up the plant from the roots against gravity, and that can be a very, very large distance if it's a huge tree. And this is possible because of the cohesion, the capillarity or adhesion, and root pressure. So if we look at cohesion first, this links to the theory of water. Water is dipolar, meaning we've got 2 slight charges, 2 different charges, a slight negative on the oxygen and a slight positive on the hydrogen atoms. And that means hydrogen bonds can form between the hydrogen and oxygen of different water molecules. And it creates this cohesion or sticking together of the water molecules. Now that's an advantage because as the water moves up the xylem, that means it's all bonded together with these hydrogen bonds, and it moves up as a continuous column of water, and it's much easier to pull up a column than it is individual water molecules. The next idea was this idea of adhesion, whereby the water molecules will actually also stick to the walls of the xylem. And if you have a really narrow xylem, that will increase this capillarity effect, and therefore the liquid will be moving up more just from this sticking, this adhesion effect. So the narrower the xylem, the easier it's going to be to transport the liquid up the xylem against gravity. Root pressure is the final concept, and this is as the water moves into the roots by osmosis, it increases the volume of liquid inside the roots, and therefore the pressure inside the root increases as well. And we call that root pressure. And that creates this positive pressure, which means it's a pushing force. Because we have lots of water in the roots, it pushes all of the water above it upwards, so it helps push the water up the xylem. So those 3 concepts all work together for the cohesion tension theory, and that's how water moves up against gravity. So just have a look at this as a whole. The first thing that happens is the water evaporates out of the stomata, and as that water vapour is leaving, that is then leaving behind this lower pressure because liquid has been lost. And that creates this negative pressure, or in other words, a pulling force. And that pulls the water column up the xylem. And because of cohesion, we have that water column whereby all the water molecules are stuck together. The water molecules are also adhering or sticking to the walls of the xylem to help pull the column upwards. And as the column of water is pulling up, it also creates tension in the xylem, which actually pulls the xylem inwards, making it narrower, which increases that impact of the capillarity or the adhesion, and it helps to move the water up even more. Now, the second type of mass transport in plants is looking at how the organic molecules like glucose produced in photosynthesis are transported around. And this now happens in the phloem. Now, the phloem tissue contains two key cells. We have the sieve tube elements and the companion cells. And the sieve tube elements, which are here, are living cells, but they don't contain any nuclei, and they're very few organelles. And that is so it's pretty much hollow to make it easy for the solutions to transport through the tube. The companion cells are on the outside, and they provide the ATP required for active transport of the organic substances. So they have all the organelles that the sieve tube elements don't, so they can provide the resources needed. Now, the transport of these organic molecules within solution is often explained using this source to sink model. So we have here the xylem next to the phloem, and the source in this model would be a photosynthesizing cell. The sink, which is where the sugars are going to be delivered, is a respiring cell. So at the source, we have photosynthesizing cells, and the sucrose or glucose that will be made in photosynthesis is going to lower the water potential of those cells. And therefore, any surrounding water in the plant or from the xylem is going to be entering those cells by osmosis. At the other end, where we have our respiring sink cells, because they'll be using up those sugars in respiration, there will be a more positive water potential inside of the cell compared to outside. And therefore, water is going to leave the respiring cells by osmosis to other cells in the plant or even the xylem. So the effect that has is there'll be an increase in the hydrostatic pressure in the source cell, but a decrease in the sink cell. And because of those pressure changes, the liquids that are within that source cell will be forced by that high hydrostatic pressure through the xylem all the way to the sink cell. Now, that is part of the story, but also you need to know the translocation steps. So how those sugars within the leaf cell actually make it into the phloem for that pressure then to actually move the liquid along. So photosynthesis is occurring in the chloroplasts, in the leaves, and we're calling those the source cells. That, we said, creates a high concentration of sucrose. Now, as well as that affecting the water potential, that also means that the sucrose can diffuse down its concentration gradient into the companion cell by facilitated diffusion. We then get active transport of protons or hydrogen ions from the companion cell into the space within the cell walls, and that uses energy because it's active transport. Now that creates a concentration gradient, and therefore the protons move down their gradient via carrier proteins into the sieve tube elements. And the co-transport of sucrose with those hydrogen ions occurs via a protein co-transporter, and that is how the sucrose goes from being in the companion cell into the phloem, even though there is a high concentration often of sugar or sucrose in the phloem already. So the next step then is we're looking at the movement of that sucrose within the phloem, sieve tube element. So the increase of sucrose in the sieve tube element is going to lower that water potential, and therefore water enters those sieve tube elements from the xylem vessels, which are tightly compacted next to the phloem. The increase in water volume in the sieve tube element at this position will increase the hydrostatic pressure, and this is where we have the idea of source to sink. It causes the liquid to be forced from the source area down to the sink cells. Lastly, the sucrose is then used in respiration at the sink, or it might be stored away as starch if it's not currently required. But that means more sucrose is actively transported into the sink cells, and that is going to cause the water potential to decrease. And as a result, we'll have water moving by osmosis from the sieve tube elements into the sink cell. Some water will also be returning from the sieve tube element into the xylem. The removal of that water is decreasing the volume in the sieve tube element, and therefore the hydrostatic pressure decreases. So the movement of soluble organic substances is due to the difference in the hydrostatic pressure between the source and the sink end of the sieve tube element. Now you could be asked about two particular investigations that prove translocation. The first one is called tracers, and this is where we have tracing involving radioactively labelling carbon. Plants are provided with only radioactively labelled carbon dioxide, and over time they'll be absorbing that in through the stomata, using it in photosynthesis, and the organic substances like the sugars created will all contain that radioactively labelled carbon. Thin slices from the stems are then cut and placed on X-ray film that will turn black when exposed to radioactive material. When the stems are placed on the X-ray film, the section of the stem containing the sugars turn black, and this highlights where the phloem are, and it can show the sugars are transported in the phloem, and it also means you can track the route that is taken. Ringing experiments is when a ring of bark and phloem are peeled and removed off the trunk, like we can see here. The result of removing the phloem is that the trunk swells above the removed section, and analysis of the liquid in this swelling shows it contains sugar. So this shows that when the phloem is removed, the sugars cannot be transported, and it therefore proves the phloem transports sugars. So we start topic 4 by comparing the DNA in eukaryotic cells and prokaryotic cells, and comparisons mean similarities and differences. So our two key similarities are the fact that the DNA is made up of DNA nucleotides for both, meaning it contains deoxyribose, a phosphate group, and a nitrogenous base. The nucleotides are also joined together by phosphodiester bonds to make the polymer chain. The three key differences are that eukaryotic DNA is longer, eukaryotic DNA is linear, so it occurs as straight lines in the chromosomes, whereas prokaryotic DNA you get circular loops of DNA. The DNA in eukaryotes is associated with histones, but for prokaryotic DNA it is not associated with proteins, those histones. So those are your key differences in the DNA. And this is just showing you here the organisation of the DNA in a eukaryotic cell. So we have our chromosomes within the nucleus that is tightly coiled to make the chromosome. But if we unwind that, you can then see the nucleosomes, which is actually the DNA wrapped around the histone proteins, and that is how we tightly coil to fit it all in the nucleus. Now you also need to be able to compare the DNA that is found in the mitochondria and chloroplasts of cells to the DNA that you find in prokaryotic cells, because they actually have quite a few similarities. So we can see here our mitochondria, and you have just a loop of DNA, and the same within your chloroplast. You just have this circular loop of DNA, and they have their own DNA so that they can translate their enzymes that they need for photosynthesis in the chloroplast and respiration in the mitochondria. So the similarities are they are both short sequences of DNA, they are both circular, and neither of them are wrapped around histone proteins. So that then takes us on to looking at what a gene is. And a gene is a sequence of DNA, and it codes for the amino acid sequence for a particular polypeptide, and also a functional RNA. So it codes for an mRNA molecule. And we can see here one of the extra key terms as well, and that is locus. And locus is the exact position that one particular gene is found on a chromosome. So locus is location. So that's the way to remember it. Locus, location of the gene. Okay, so the genetic code, you need to know a few features of the genetic code. One thing, first of all, is knowing that a sequence of 3 bases on DNA is called a triplet, and those 3 bases will code for a particular amino acid. So the 3 features are, it is a degenerate code, it's universal, and it's non-overlapping. So if we ever think about this idea of degenerates, there are 20 amino acids that we said exist, and we have 4 possible DNA bases. And the way they actually worked out that it was 3 DNA bases that codes for 1 amino acid was actually mathematically. They looked at all the possible code options you could get if you only had 1 base coding for 1 amino acid, and that would only give you 4 possible codes, G, C, T or A. That's not enough to code for 20 different amino acids. So then if you think about, could it be 2 bases? If you had only 2 bases, that would give you 16 possible different codes, and that's still insufficient. So they realised it must be 3 bases that code for 1 amino acid, because that actually gives you 64 possible different options for what those triplets of bases could be. And that is more than enough to code for the 20 amino acids. And that is why the genetic code is degenerate. And what we literally mean by this definition is there are more than 1 triplet of bases that codes for the same amino acid. That would be your key definition. And this table here is just showing you an example of that. So you can have a look, for example, at, so let's look at glycine here, GLY, GGG, GGA, GGC, GGU. All 4 of those triplets of bases code for glycine. And this is an advantage of the genetic code because if there is a mutation and one of the bases in a triplet is changed, you might still have the new triplet coding for the same amino acid, and therefore it has no effect on the overall polypeptide chain. Universal means that the same triplet of bases codes for the same amino acid in all organisms. Non-overlapping is the fact that each base is only involved in 1 triplet. So if we just draw boxes around this to show you what I mean, this base A is only in this 1 triplet, and this C is only in the triplet, this G is only in this triplet. We don't have this G also making up a second triplet of bases. So every codon or triplet of bases is read as a discrete unit. This is an advantage as if a point mutation occurs, it will only affect 1 codon and therefore 1 amino acid, so it will minimize any potential harm. Now in your DNA, you have sections of base sequences, which are introns, and you have sequences of DNA that are exons. Introns are sequences of DNA bases that do not code for polypeptides. And you actually have a lot of introns making up your nuclear DNA. The exons are sequences of DNA bases that do code for the amino acids. So the exons are the coding regions. And when we say codon, a codon is 3 bases on mRNA that codes for a specific amino acid. A start codon is 3 bases that you find at the start of every gene. And that is what initiates translation to occur. A stop codon is the final 3 bases that you have at the end of every gene. And those 3 bases will cause the ribosome to detach during translation, and therefore it stops the translation of the polypeptide chain. A genome is what we call an organism's complete set of genes in a cell. So that is your definition of a genome, whereas the proteome is the full range of proteins that a cell is able to produce. The genome should never change unless there are mutations, whereas the proteome of a cell can constantly change depending on which proteins are needed in a specialized cell, because you'll have some genes have been switched off or on, and that's what makes it specialized. The genome of an organism will really differ between different species. So for example, a bacteria contain on average 600,000 DNA base pairs within their genome, whereas humans, we have 3 billion DNA base pairs. So this then starts to take us on to RNA before we get into protein synthesis. So messenger RNA is what mRNA stands for, and we can see it here on the picture. It is short compared to DNA because it's only a copy of one gene, whereas the DNA is the entire genome. It's single-stranded, and it's found in both the cytoplasm and the nucleus. So it is made during transcription, and that happens in the nucleus. But then once it's been modified, it leaves the nucleus, enters the cytoplasm to attach to a ribosome. It's 3 bases on mRNA that are codons, so 3 bases which can code for a particular amino acid. tRNA is transfer RNA, and this is found in the cytoplasm. It has an amino acid binding site, which we can see up here at the top, and each tRNA molecule will have a particular or specific amino acid attached to that binding site. The tRNA molecule also has 3 bases on it at the bottom here, and we call those 3 bases the anticodon, and they will be complementary to a particular codon on mRNA. And when those align, they're held in place so that amino acids can start to bond together during translation. So tRNA is involved in translation, the second stage of protein synthesis. A ribosome will be holding it in place to enable the joining of amino acids. It has this clover leaf shape, that's what we call this shape, and we can see here these lines are representing hydrogen bonds. So it's still single-stranded, it's just folded to create this shape, and that shape is held in place by hydrogen bonds. So that then takes us on to protein synthesis, and it's split into 2 steps: transcription, which is where one gene at a time from DNA is copied into mRNA. Then we have translation, where the mRNA will join with a ribosome, and corresponding tRNA molecules will then bring specific amino acids. So first of all, we have transcription. So a complementary mRNA copy of one gene on the DNA is created in the nucleus. mRNA is much shorter, we've already said, and that is because it's only copying one particular gene, and therefore is able to leave the nucleus because it is smaller. Your key steps are all here. So if you did have a long answer question, describe the process of transcription, these would be your key marking points, with the key marks put in bold. So first of all, the DNA helix unwinds to expose bases, and you have one strand acting as a template, and that's our second mark. Like with DNA replication, that is caused by DNA helicase breaking the hydrogen bonds. In the nucleus, you then have free-floating mRNA nucleotides, and they will align opposite their complementary DNA base pairs on the template strand. The enzyme RNA polymerase will then join together those RNA nucleotides to create the mRNA polymer chain. Once it's copied, it then has to be modified, and then it leaves the nucleus via a nuclear pore. The modification that happens is this here, splicing. In eukaryotes, after transcription, we actually call the molecule pre-mRNA, and that is because it still contains the introns, which are those non-coding sequences of bases. And that's because the DNA, the gene that was copied, there will be introns within it, which are the non-coding sequences. So the RNA that is copied will still contain those introns. So the introns need to be removed, and we call that splicing. They're spliced out, so cut out, they're spliced out by a protein called a spliceosome. And now we have finished mRNA that is ready to leave the nucleus. That stage doesn't happen in prokaryotes because they don't have introns. Translation is the next stage in the creation of the polypeptide chain, and it involves both mRNA and tRNA. If you were asked to describe that whole process, again, these are your 6 key marking points, and in bold are the key terms you would have to include. Once the modified mRNA has left the nucleus, it will then bind to or bind with a ribosome in the cytoplasm. The ribosome will attach at the start codon of the mRNA molecule. The tRNA molecules with complementary anticodons to the start codon will then align opposite, and they're held in place by the ribosome, which we can see here in the picture. The ribosome holds together 2 tRNA molecules at a time. The 2 amino acids that have been delivered by the tRNA molecule are joined by a peptide bond. And that reaction does require energy in the form of ATP and an enzyme, but you don't need to know the name of it. Once that happens, the tRNA molecule will be released and the ribosome moves along one codon, so the next tRNA molecule can then align its anticodons to its codons. So this continues until the ribosome reaches the stop codon at the end of the mRNA molecule, and when it does, that causes the ribosome to detach and therefore translation ends. Now the modifications, because we now just have a polypeptide chain, the modifications will occur in the Golgi body for folding to create that secondary, tertiary or quaternary structure. We then move on to how variation is introduced, and gene mutations is one way. A change in the base sequence of DNA is what a gene mutation is, and they randomly occur during DNA replication, so within the interphase part of the cell cycle. These random mutations are more likely to occur if you're exposed to mutagenic agents, which can interfere with the DNA replication. That includes high energy radiation like UV light, ionizing radiation like gamma rays and X-rays, and also some chemicals which we call carcinogens, for example mustard gas and cigarette smoke. A gene mutation can result in either a base being deleted or swapped, so substituted for a different base. So here are our examples. We have our original DNA sequence. This is showing a substitution. So instead of cytosine, that has been swapped or substituted for adenine. This one is showing a deletion because that base C has now been deleted. And that's actually caused what we call a frame shift. Everything downstream of the mutation has shifted to the left. A base mutation, though, might have no impact at all because the new codon may still code for the same amino acid, and that's because the genetic code is degenerate. Chromosome mutations can also occur, and chromosome mutations are changes in the number of chromosomes, and this spontaneously occurs during meiosis in a process called non-disjunction. So non-disjunction is when the chromosomes, or it could be the chromatids, do not equally split during anaphase of either meiosis 1 or meiosis 2. So that's what we can see here, non-disjunction occurring because the chromatids didn't separate and instead all of them are being pulled to the same pole of the cell. Now this can occur in 2 forms. Either a change in the whole set of chromosomes, which we call polyploidy, or changes in the number of individual chromosomes, which is aneuploidy. So we'll go through polyploidy first, which we said is a change in the whole sets of chromosomes. So you could end up, instead of being diploid, having 2 copies of every chromosome, which we have in humans, you could have 3 copies or 4 copies of every chromosome, which would be called triploid or tetraploid. Now in humans that would be fatal. You don't see triploid or tetraploid humans, but it's actually quite common in plants. So how this would occur then? Each homologous pair is doubled in replication, and that happens in interphase. In this example, we have non-disjunction in meiosis 1. For some reason, the spindle fibers haven't attached to the chromosome on this side, and they have attached to all of the chromosomes on the other side. So when the spindle fibers retract, it's going to pull all of the chromosomes to one side of the cell, and therefore they're all going to be in this cell and there'll be no chromosomes in the next one. In meiosis 2, that would mean that these two gametes will contain no chromosomes at all, so those gametes will not function. These gametes though, meiosis 2 is happening normally and we do have complete separation of all of the chromatids, but we now have 2 copies of every chromosome in the gamete. So instead of having a haploid gamete, we have a diploid gamete. And if a diploid gamete fuses with a haploid gamete, that is how we then end up with 3 copies. So we get 2 chromosomes from this gamete instead of 1, and we get just 1 chromosome from the haploid gamete. So that is polyploidy, changes in the whole sets of chromosomes. That could also happen if you have non-disjunction in meiosis 2. In this example, we can see the chromosomes in meiosis 1 did separate equally. And then we had normal meiosis 2 in this example, so we have 2 haploid gametes. But for this cell, in meiosis 2, there was non-disjunction. So the spindle fibres didn't form on this side, so the chromatids aren't separated equally, and instead they're all pulled to this cell. So again, we end up with a 2N gamete, a diploid gamete, and this gamete has no chromosomes in. Aneuploidy, this is different. This is when you have changes in the number of individual chromosomes. So sometimes individual homologous pairs of chromosomes fail to separate during meiosis. It's still called non-disjunction, but instead of it being affecting every single chromosome or chromatid, it's just one. And this is how Down syndrome occurs. You have non-disjunction on chromosome 21, so you end up with 3 copies of that chromosome instead of 2. So let's see how that might occur. We can see in this one, we have non-disjunction occurring at meiosis 1 because these spindle fibres for just that one chromosome or that one homologous pair of chromosomes is attaching and it pulls them both to this cell. And this cell does not get a copy of that red chromosome. If meiosis 2 occurs normally, so no non-disjunction, all of the chromatids are separated equally. However, because of the non-disjunction in meiosis 1, this gamete is haploid. It has 1 copy of all the chromosomes except for the red. So we describe that as haploid plus 1 extra chromosome. So N plus 1. These 2 are still haploid, but they're missing a chromosome. So we describe it as N for haploid minus 1. Now if an N plus 1, so a haploid with an additional chromosome, is to fuse with a haploid chromosome, that is how you can get trisomy, which means 3 copies, so trisomy is 3 copies of one particular chromosome. And that is how Down syndrome occurs, 3 copies of chromosome 21. Now you could also have non-disjunction occurring in meiosis 2. So we can see that normal cell division occurred in the first round of meiosis, but now we have non-disjunction in meiosis 2 because the chromatids are not separated equally for the red chromosome. They're all pulled to this one gamete, so that would be N plus 1. And this one is missing the red chromosome, so it's N minus 1. Now another way that variation can be introduced is in meiosis. And meiosis creates gametes, and it creates 4 genetically different haploid gametes by 2 nuclear divisions. So meiosis is how variation can be introduced as well, and that's through 2 mechanisms: independent segregation of homologous chromosomes and crossing over between homologous chromosomes. And both of these occur within the first round of division in meiosis. Independent segregation is when the homologous pairs of chromosomes line up opposite each other at the equator to form bivalence. It is random which side of the equator the paternal and maternal chromosomes from each homologous pair align. So we can see on this side by chance 2 purples, 2 reds, but equally it could have been a purple and a red and a purple. In meiosis, those homologous pairs of chromosomes are separated in meiosis 1, so one of each homologous pair ends up in the daughter cells eventually. This creates a large number of possible combinations of chromosomes in the daughter cells produced. And you can actually calculate this by doing 2 to the power of n. 2 because it is homologous pairs, so you have pairs of chromosomes, and n you would substitute in for how many homologous pairs of chromosomes that species has. So for humans, that would be 2 to the power of 23. We have 23 pairs of chromosomes, which means we can make over 8 million different possible gametes just from independent segregation. Now, crossing over also occurs sometimes. It's actually quite rare, but it can occur. And again, it occurs when the homologous pairs of chromosomes line up at the equator and form a bivalent, which is what we call it when you have both of them next to each other. And you have chromatids from each of those chromosomes cross over and they can get twisted around each other. That puts tension on the chromatids, causing part of the chromatid to break and swap. And in doing that, we create new combinations of alleles, which is represented by the letters here. So originally, this chromosome only had a dominant allele, but now we've got a dominant and recessive, so when that's pulled apart, we've got new combinations of alleles on that chromosome. Now, comparing meiosis and mitosis, meiosis is 2 nuclear divisions, whereas mitosis is only 1. That's why meiosis results in haploid cells, whereas mitosis is diploid cells. Meiosis introduces genetic variation through crossing over and independent segregation. Mitosis creates genetically identical cells. Now, you could be asked to identify meiosis in an unfamiliar life cycle. And what you need to do here is look for where you have cells that were diploid or 2n dividing to then create cells that are haploid, because that's what happens in meiosis. You go from 2n to n. It won't always be gametes because not all organisms have life cycles like humans where it is the creation of the gametes that is meiosis. So for example, we can see here we have the zygote, which is 2n, and then it makes something called zoospores, which are n. So that would be the meiosis stage. So that's what you're looking for, 2n moving to n. So again, that bit there would be meiosis. Genetic diversity, this is the number of different alleles of genes in a population. And this is what enables natural selection. Natural selection can only occur if there is genetic diversity. Natural selection is the process that leads to evolution. And our definition of evolution is the change in allele frequency over many generations. Natural selection is really important to the survival of the whole species because it results in the species becoming better adapted to their environment. And these adaptations might be anatomical, physiological or behavioural. So the key marking points that you'd need to describe for this process are, first of all, you would have new alleles for a gene being created by random mutations. If those new alleles increase the chances of the individual surviving in that particular environment, then they're more likely to survive and therefore more likely to reproduce and pass on that new advantageous allele to the next generation. Over many generations of that occurring, that new allele will now become more common in the gene pool, or in other words, we've increased the allele frequency. Now, the types of selection that occur are directional selection and stabilizing. Directional selection is when the advantageous allele is coding for an extreme trait. So this links to your antibiotic resistance example. And if we think about the traits being low resistance, medium, high resistance, the extreme traits would be either very low resistance or very high resistance. And in the case of antibiotic resistance, when there was a change in the environment, which is introduction of antibiotics, the bacteria which had the alleles for high resistance to antibiotics were more likely to survive and pass on that allele for antibiotic resistance. And that's why we saw this shift, and antibiotic resistance allele became far more common in the species. Stabilizing selection is when whatever is the middling trait remains the selective advantage. And that would be the case if there's no change in the environment. And this is exemplified by human birth weights. So being a middling birth weight increases your likelihood of surviving, because if you're very, very small, then you might be very premature, might have underdeveloped lungs, difficulties regulating your temperature, and more vulnerable to infection. If you're very, very heavy, then it's going to be a more complicated birth, and that could result in difficulties. So that would mean that the modal trait remains a selective advantage, and we can see that the range of traits or alleles decreases over many, many generations. The range and the standard deviation decrease. Next then, it's thinking about species and taxonomy. So a species is when two organisms are able to produce fertile offspring. And a species must reproduce and pass on advantageous alleles in order for the whole species to be able to survive. And this is where courtship behaviour comes in. This behaviour is essential for successful mating, meaning mating and creating fertile offspring, in order for species to survive. So courtship behaviour is a sequence of actions which is unique to every species. So it is genetically coded for, this behaviour, and it is how animals are able to identify members of their own species to make sure they are reproducing with members of their own species to make sure that they can create fertile offspring. The behaviour, the sequence of actions, is normally carried out by the male, and then the female picks whether they are worthy of mating with. Now, this sequence could include dance moves, creation of sounds, release of pheromones, display of feathers, fighting. Whatever it is always a unique sequence to that species. The female then observes the ritual and decides if they look like they have a good enough set of alleles based on their fitness, their performance, to mate with. So the reason this is important is, we said, to ensure successful reproduction. And the way it does that is, first of all, because the behaviour is unique to every species, it allows them to recognise members of their own species and also the opposite sex. That is because to make fertile offspring, you'd need sperm and egg to fuse. It also synchronises mating behaviour. And what this means is it makes sure that the male and the female are mating when they are sexually mature. So the female is releasing eggs and the male is able to produce and release sperm. It can actually also help the survival of the offspring once it's born in some animals. And that's because this behaviour, this courtship ritual, can help form a really strong bond, which we call a pair bond, between the parents. So they're more likely to stay together. And if they are together, for some animals, like penguins, it increases their likelihood of survival, because you need one penguin to look after the chick, one penguin to go and find food. It also enables that strong and healthy mates are selected for, and therefore the advantageous alleles are being passed on to the next generations to ensure the survival of the entire species. So this could be used as a way for us to identify how closely related different species are as well. So for example, we've been shown 3 ducks here and the sequence of their courtship behaviour. And we can see that ducks 1 and 2 must be more closely related because their sequence of behaviours in the courtship ritual are more similar than duck 1 and 3 or ducks 2 and 3. And because this behaviour is genetically coded for, because the sequence of behaviours is more similar, their DNA-based sequence is likely to be more similar too. Phylogenetic classification is another way to look at how closely related different species are, and also how recent their shared common ancestors were. Phylogenetic classification is arranging groups according to evolutionary origins and relationships. So humans and chimpanzees, we can see, are most closely related to each other because they branch in the tree most recently compared to the others. So that means they evolved from a shared common ancestor more recently than they did compared to any of the other species. So that means they have had less time to accumulate different mutations in the human and chimpanzee populations compared to, let's say, the human and the horse population, because humans and horses, their recent common ancestor is back here. And there's no time scale on this, but this is normally going back, like this would probably be at the very start here. This could be looking at maybe 13, 20 million years ago, and so on. So it is millions of years of accumulating mutations in these separate species. Now you can also classify and group using a hierarchy. And a hierarchy is when you have smaller groups arranged within larger groups, and there's no overlap between those groups. So this is one particular hierarchy you need to know off by heart. Domain, kingdom, phylum, class, order, family, genus and species. And we can see here that within one genus you can have multiple different species. So that is our smaller groups within larger groups. But there's no overlap in those species. They're still distinct groups. The binomial system is a universal way of identifying organisms, and it's using two names. That's what binomial means. The first name is the genus, and the second name is the species. So for humans, our genus is Homo, and our species is Sapiens. So our binomial name is Homo sapiens. And we can see an example here of common names, which would be unique to every language, versus the binomial system, which is universally used. That gives you more information on how closely related they are, because calling them both robins is misleading, that they are closely related. But actually, we can see they're not the same species, and they're not even the same genus. So the final thing is looking at biodiversity. And there's different ways that you can classify and measure biodiversity. It could be looking at the range of different habitats that exist. It could be the variety of genes amongst all the individuals of a population of one species. Or it could be looking at the number of different species and individuals within each species in a community. Species diversity as well, if we go into that further, that takes into account species richness. And species richness just means the number of different species in a community. Biodiversity can be used to describe a range of habitats. You could be looking at the biodiversity of a very small local habitat like a forest, or it could be talking about the entire Earth. Having a low biodiversity isn't actually a cause for concern. For example, in the Arctic or deserts, you would expect that to be low. But if you have a decrease in biodiversity, that is a cause for concern because that could be caused by human activity causing destruction. And farming techniques is an example of that. Farming techniques can reduce biodiversity, but it is needed to provide food for humans. So we have to come up with some kind of compromise between the two. You could get application questions where you have to suggest different farming techniques that could reduce the biodiversity. So I've got some of those listed here. And if you want more detail on that, then go to my actual biodiversity video to hear all of the examples and how it reduces the biodiversity and what the compromise would be. The way that we measure biodiversity is using the index of diversity. And that describes the relationship between the number of species in a community, which is the species richness, and the number of individuals within each species. So this is the formula to calculate it, and they would give you that in the exam. But they won't give you this. That capital N is the total number of organisms of all species. Lowercase n is the population size of one species. 1 is the lowest value you can get, and the larger the value, the greater that index of diversity, the greater the biodiversity. So here's an example. If you want to have a go, pause the video now and write it down and then have a look at the answer. If not, let's go through. So the first bit of the formula we're going to look at is the lowercase n times n minus 1. So that would be 6 as our lowercase, the population size of species A. And n minus 1, that would be 5. 6 times 5 would be 30. So we then do that for all of the others. And we need the sum of that. So the sum of that column is 180. Capital M we said was the total number of all individuals. And in this example, it's 25. So 25 times 24 divided by 180 is 3.3. Genetic diversity within or between species can also be measured by comparing different factors. So you can compare observable characteristics, but that can actually be quite inaccurate because members of different species that aren't even closely related might look similar because they live in similar environments. So more accurate ways to compare how closely related species are is through comparing the DNA-based sequence, the mRNA-based sequence, or the amino acid sequence for proteins. And the more similar those sequences, the more closely related they must be. So the first thing to do with photosynthesis is knowing where it occurs and just a mini recap on the structure of a chloroplast because you need to know where each stage of photosynthesis happens. So here on our chloroplast we can see the thylakoid stacks, and the thylakoid membrane is on the outside of that. And that's where you have these folded membranes where you have all the proteins embedded for the electron carrier chain, and also ATP synthase. And that is where the light-dependent reactions occur. A stack of them is called a granum. The fluid part in the centre is called the stroma, and that's where the light-independent reactions happen, and you have lots of enzymes there for those. And then we have the inner and outer membranes as well, which are controlling what can enter and exit. Photosynthesis is a two-stage reaction. We have the light-dependent and the light-independent reactions. And the light-dependent reactions happen first, and as the name suggests, it requires light. This happens on the thylakoid membrane, or you could call that the granum. In this light-dependent reaction stage, light energy is used and water to create ATP and reduced NADP. And those two products are required for the light-independent stage, which comes second. So there's 4 key steps here. We have photolysis, or sometimes it's pronounced photolysis to remember the stage, photoionization of chlorophyll, and chemiosmosis. Now those are actually the 3 processes. Production of ATP and reduced NADP is the outcome, but AQA states these are the 4 key stages that you need to know. So starting with photolysis or photolysis, photo refers to light, lysis means splitting. So photolysis of water means light energy is absorbed and then that splits the water. And it splits it into oxygen, electrons and protons, which are hydrogen ions. What then happens is the protons are picked up by NADP to form reduced NADP or NADPH, and that is then used in the light-independent reaction. The electrons are passed along a chain of electron carriers, and the oxygen is either used in respiration, but it's not used in photosynthesis. So if it isn't needed for respiration, it will diffuse out through the stomata. We'll go through in a bit more detail what happens to the protons and electrons when we get to chemiosmosis. Photoionization of chlorophyll, again, is to do with light, because we've got photo. So light energy is absorbed by the chlorophyll, and the ionization part is referring to the fact that the light energy results in the electrons gaining energy. And we describe that as them becoming excited and raising up an energy level. And that causes the electrons to leave the chlorophyll, and therefore the chlorophyll has been ionized. Some of the energy from the released electrons is used to make ATP and reduced NADP in chemiosmosis. So let's have a look at chemiosmosis then, because this then links it all together for the light-dependent reactions. So we've said that the electrons gain energy from the chlorophyll, will be released from the chlorophyll. What happens is those electrons move along a series of proteins embedded within the thylakoid membrane. And as those electrons move along the proteins, they release energy. And some of that energy is used to transport or pump those protons from the stroma across a protein and into the thylakoid lumen. This then results in a concentration gradient being built up, but we actually call it an electrochemical gradient because we have a charged molecule, so we have ions there. So now we have this electrochemical gradient that enables the protons to move by facilitated diffusion back down their concentration gradient to the stroma. But the only protein that they can attach to is ATP synthase. And as the protons attach and move through ATP synthase, that enables the enzyme, which the protons move through, to phosphorylate the ADP into ATP. So that's how the ATP is produced. We then have the protons back on the stroma side, and this is how we create NADP that is reduced, or reduced NADP. So the NADP coenzyme picks up the electrons from the end of the electron transport chain. It picks up the protons after they've passed through ATP synthase, and that reduces NADP, which makes it NADPH. So now we've created ATP and reduced NADP. Those two molecules are used in the light-independent reactions, also known as a Calvin cycle. This occurs in the stroma. It requires enzymes, and the enzyme Ribisco is the enzyme that you need to know. And because of that, this stage is temperature sensitive. But it doesn't require light energy for the Calvin cycle to occur. Now, the Calvin cycle uses carbon dioxide as well as the two products from the light-dependent reactions, and we create a hexose sugar. The ATP that is needed is hydrolyzed to provide energy for the reaction, and the reduced NADP is used to donate the hydrogen to reduce the molecule GP within the cycle. So let's go through this cycle then. The first part of the cycle we'll look at is how the carbon dioxide enters the cycle. So carbon dioxide reacts with RuBP, and that is a 5-carbon compound. So all of these yellow circles are representing carbon molecules. So we can see we've got a 5-carbon compound reacting with carbon dioxide, which only has 1 carbon. That would result in a 6-carbon compound, but actually it's unstable, so it splits into 2 3-carbon compounds, and that's our GP. So we have 2 lots of GP. This is then where one of the ATP molecules is used, and it's where the reduced NADP is used. GP picks up the hydrogen, and therefore NADP is reformed, and the GP is now reduced to form TP. That reduction requires energy from ATP. The TP is then used for 2 things. One of the carbons is removed from these 2 times 3 carbon compounds, and it goes towards creating a hexose sugar. That will leave behind 5 carbons, but as a 3-carbon compound and a 2-carbon compound. So ATP is needed to form those back together, or in other words, regenerate RuBP. Now, that is just one round of the cycle, and that only created 1 carbon, which isn't enough for a hexose sugar. Hexose means 6. So the Calvin cycle has to happen 6 times before a hexose sugar is made. And that could be glucose, it could be sucrose, and then the plant can use that glucose or sucrose and convert it into whatever organic compound is needed. So that could be another carbohydrate, so cellulose for the cell walls. It could be a store of glucose in the form of starch, or it could convert the hexose sugar into glycerol and then combine with fatty acids to make lipids, or it could combine it with nitrates to form the amino acids. Now you need to know a little bit about limiting factors as well, and the limiting factor is anything that reduces the rate of photosynthesis, and that could be light intensity, carbon dioxide concentration, or temperature. And the way you can look at a graph to tell what is currently limiting the rate of reaction is wherever you have a positive correlation on the graph, that tells you that whatever is on the x-axis is currently limiting the rate. So that means up until this point, or probably a bit further up to here, light intensity is limiting the rate of reaction. And we know that because as we increase the light intensity, the rate increased. But that then starts to plateau and level off. So even though you add more light, the rate doesn't increase. And what that means is there must be another factor that is now limiting the rate. And that must be either carbon dioxide concentration or temperature. So it's the same concept with carbon dioxide concentration, that as the rate is increasing and the carbon dioxide concentration is increasing, we know that carbon dioxide is a limiting factor until it plateaus. Temperature, this is linked to the idea of enzymes because it's an enzyme controlled reaction. So at low temperatures, the rate of reaction is lower because we don't have enough successful collisions. There's not enough kinetic energy. We don't get enough enzyme substrate complexes. But if it gets too hot, the enzyme denatures. Now, the carbon dioxide, if you had to say why that limits, that is because it is a reactant in the Calvin cycle. Light intensity can limit the rate of reaction because light energy is needed in the light-dependent reactions for photolysis and photoionization. So for maximum photosynthesis, in agriculture, when they're growing crops, they need to consider how they can remove those limiting factors to maximise their profits. So sometimes you get application questions linked to this. So the idea of adding in artificial lighting, heating a greenhouse, and that could be through burning fuels because that will also produce carbon dioxide. But all of those additions cost. So it has to be considered whether the cost of the additional light, heat or fuel is offset by the additional profit. So they have to make sure it's cost effective in order for it to be worthwhile to pay for those additional conditions. Next then we have a look at respiration, starting with aerobic respiration. And there are 4 key stages: glycolysis, the link reaction, Krebs cycle and oxidative phosphorylation. And for each stage, you need to know exactly where in the cell it occurs. And that's what we have in brackets here. So we'll start with glycolysis. This does produce some ATP, but not a lot. And it also happens in anaerobic respiration because oxygen is not required for glycolysis. It happens in the cytoplasm, not actually inside of the mitochondria. And there are 3 key stages that AQA say you need to know. First thing that happens is that glucose is phosphorylated to glucose phosphate, and that uses 2 molecules of ATP. That glucose phosphate very quickly is converted into triose phosphate, and then triose phosphate is oxidated to form pyruvate, and that produces 4 ATP and reduced NAD, which is the coenzyme used in respiration. So let's just have a look at what that would be on a diagram. Again, the yellow circles are representing carbon. So glucose is a 6-carbon compound. We said the first thing that happens is glucose is phosphorylated, and what that means is phosphate groups are added to it. Now we're going to add 2 phosphate groups, so we need 2 ATP molecules, and that creates glucose phosphate. That is an unstable compound, so it splits into 2 3-carbon compounds called triose phosphate. And then the triose phosphate, or TP, both of those molecules are oxidized. Both TP molecules will result in the gain of 2 ATP when pyruvate is made, and also as each is oxidized, there'll be a reduced coenzyme created. So NAD picks up a hydrogen to form NADH, or reduced NAD. So at the end we have 2 molecules of pyruvate, a net gain of 2 ATP, and what we mean by that is, although we've made 4 ATP molecules in this stage, we had to use 2 at the beginning. So overall, we've created 2. We've also made 2 reduced NAD. So that pyruvate that has been created and the reduced NAD, They are needed in the next stages, which is within the mitochondrial matrix. So they have to be actively transported into the mitochondrial matrix, ready for the link reaction. So the pyruvate is going to be oxidized further to make acetates. We're going to have an NAD picking up a hydrogen to create yet another reduced NAD molecule. So that's the first step we can see here. Pyruvate, our 3-carbon compound, it's going to be oxidized further, and we can tell it's been oxidized because it's lost the hydrogen, which has been picked up by the coenzyme NAD. So that has created acetate. Acetate is only a 2-carbon compound, and that is because carbon dioxide is produced at this stage of the link reaction. Now that's not the end, because the acetate then combines with another coenzyme called coenzyme A, and that creates acetyl-CoA. And acetyl-CoA is what can then enter the Krebs cycle. So overall, for every glucose molecule, there are 2 pyruvates created. So for every glucose molecule, the link reaction happens twice. So for one glucose molecule, we'd create 2 acetyl-CoA, 2 carbon dioxides, and 2 reduced NAD. So that acetyl-CoA can then enter the Krebs cycle, and it combines with a 4-carbon molecule. And that will then create a 6-carbon molecule. Now the coenzyme, its only job is to deliver the acetate to that 4-carbon compound. So once that happens, the coenzyme is released and it can be reused in the link reaction. We then have a series of redox reactions, and the Krebs cycle is going to be generating even more reduced coenzymes, a small amount of ATP, and some carbon dioxide. And that's what we can see in this stage here. The 6-carbon to the 4-carbon molecule, which is then regenerated. To go from 6 to 4, we must have lost 2 carbons, and that's in the form of 2 carbon dioxides. We only have 1 molecule of ATP being produced, but we have quite a few reduced coenzymes. We create 3 reduced NAD and 1 reduced FAD, which is our final coenzyme involved in respiration. So overall, the products per cycle are 3 reduced NAD, 1 reduced FAD, 1 ATP, and 2 carbon dioxides. But for every glucose, the cycle happens twice, because we'd have 2 acetyl-CoA produced, so 2 rounds of the cycle. So we actually double all of those values. So look carefully at an exam question, whether they're asking for the products per cycle or per glucose molecule. Oxidative phosphorylation is the final step, and this is very similar to what we looked at in chemiosmosis in the light-dependent reaction of photosynthesis. It's when most of the ATP is produced. So the first thing that happens is, in the mitochondrial matrix, all of those reduced coenzymes that were produced are going to release their hydrogen, and that splits into protons and electrons. Those electrons are going to be transported along the electron transfer chain. That releases energy, and that energy is used to transport the protons from the mitochondrial matrix into the intermembrane space. That creates this electrochemical gradient, and therefore the protons can move by facilitated diffusion down their concentration gradient through ATP synthase. And the ATP synthase will then phosphorylate ADP to create ATP. So that is how we create lots of ATP in this stage, and you actually create 34 ATP molecules. Now at the end of the electron transfer chain, those electrons are picked up by oxygen. And oxygen also picks up the protons once they've passed through. And that is how we create water. So we say that oxygen is the final electron acceptor, and it will form water when it also picks up those protons. Anaerobic respiration is without oxygen, and it only occurs in the cytoplasm. In plants and microbes, anaerobic respiration produces ethanol and carbon dioxide. In animals, it produces lactate or lactic acid. And this stage is all to do with reoxidizing NAD so that glycolysis can continue to occur. So what we have happening is glycolysis occurs as usual because no oxygen is needed for that stage. But instead of the pyruvate being transported into the mitochondrial matrix for the link reaction, it remains in the cytoplasm, and the pyruvate is reduced to form lactate. We know it's reduced because the reduced coenzymes that were just produced in glycolysis are immediately reoxidized to NAD, and that is because the pyruvate gained the hydrogen. Now that is important because that NAD can then be reused in glycolysis so that glycolysis can continue to occur. And that ensures that at least some ATP is continually made. So the downside is lactic acid is acidic, so it can denature enzymes and other proteins, but that is the price to pay so that at least some respiration can still occur. But you can't respire anaerobically for long because that buildup of lactic acid will eventually denature the enzymes involved in glycolysis. Same idea for microbes, except we have ethanol and carbon dioxide produced. Again, ethanol is going to be harmful, but it's to ensure that glycolysis can still continue, so at least some ATP is being produced for metabolic reactions to continue. The next bit in this topic is looking at how energy is transferred in an ecosystem. So it's looking at food webs. And all food webs begin with producers, which are green plants because they can photosynthesize to use the light energy to make their own organic compounds. So they don't need to eat, they produce their own food. Between each trophic level, which is the term of what we call each stage in a food web, the majority of energy is lost, and that is due to respiration and excretion. The remaining energy is used to form the biomass. And the biomass is the mass of carbon within the organism, or you can describe that as the dry mass. Now that is a key term, so definitely learn that definition. And we give biomass per given area, so it can be a fair comparison between organisms. MPP and GPP are two key terms that you need to know. So how productive an ecosystem is depends on the abiotic and biotic factors that are within that ecosystem. If there's plenty of water, light, it's warm, there's lots of green plants, then you'll have a really high rate of photosynthesis, and that results in more carbohydrates being produced by the plant. And we can quantify this using GPP and MPP. So GPP is gross primary production, and it is the chemical energy stored in the plant biomass in a given area, or it could be volume, and it's the total energy resulting from photosynthesis. NPP is the net primary production, and that is the GPP minus the energy that will be lost by respiration. So basically MPP is the energy you have left to go towards making the biomass.
Another formula that you need to be aware of is this one here, and this is looking at the net production of consumers. So to work this out, which is represented by the letter N, for organisms such as animals, you'd use the formula below, where N is the net production of the consumers, I is the chemical energy stored in ingested food, F is the chemical energy lost to the environment in feces and urine, and R are the respiratory losses. So here's the formula, and it's come up a couple of times as just a multiple choice question, just ticking which is the correct formula. So that's a good one just to learn, maybe put on a flashcard. So here's just a visual representation. We're basically looking at the amount of energy going in minus the energy losses coming out, which is the energy lost in respiration and the energy lost in feces and urine.
The rates of productivity are recorded using the units kilojoules per hectare per year. So there's 3 parts to those units, and it can be asked as a 2-mark question to give the units or to say why you have 3 parts to the units. So the kilojoules is the unit of energy, but these units also include per unit area and per year. So it's recorded per unit area to standardize the results to enable environments to be compared. So it's taken into account that different environments will vary in size. The units are also per year to take into account the impacts of the seasons. So when you have rain, when it's light or when it's less light, the temperature changes. That's going to affect the productivity because it affects photosynthesis. So we do it per year, so you have an annual average, so it's a fair comparison. The last thing within this topic is the nutrient cycles. And we're going to start with the nitrogen cycle. The nitrogen cycle is really important because nitrogen is within proteins, ATP and nucleic acids. So that's the DNA and RNA. So it's essential that animals, plants, microbes have nitrogen within them to create these molecules. And we need this cycle because you can't actually gain nitrogen gas through gas exchange like you can with oxygen or carbon dioxide for plants for photosynthesis. And the reason for that is nitrogen gas has this triple bond and we don't have the enzymes to be able to break those bonds and then use the nitrogen in compounds. Microorganisms do though, so they have the enzyme to be able to do that, or some of them do. And therefore we need this cycle involving microorganisms to make the nitrogen accessible for nitrogen containing substances, which plants and animals can then use. The nitrogen cycle involves these key processes: ammonification, nitrification, nitrogen fixation and denitrification. But you also need to be aware that saprobiotic nutrition and microbes are essential for all of those stages highlighted in blue. So saprobiotic nutrition is referring to when microbes are feeding on dead plant matter or waste, and it's the microbes that are going to be doing that. So if we have a look at the cycle, we have nitrogen gas in the atmosphere. There'll be nitrogen fixing bacteria that can convert that nitrogen into ammonia. And that will either be done directly by nitrogen fixing bacteria in the root nodules of leguminous plants, which are types of plants like clover and beans, which have these lumps or nodules on their roots that contain bacteria. So for them, the bacteria will directly convert the nitrogen in the atmosphere into ammonium into their roots. Plants that don't have those root nodules, there is free living nitrogen fixing bacteria within the soil, which can also fix nitrogen into ammonium. Then we have nitrification occurring, which is when nitrifying bacteria can oxidize ammonium into nitrites and then oxidize it further into nitrates. And that can then be absorbed by active transport into the plant roots. And then animals can absorb that through digestion and absorption. Now, the decomposers will be breaking down the nitrogen containing compounds in the dead plant and animal material and in the waste produced by animals. But for A level, it's not enough to just say the dead material or waste. You have to name the nitrogen containing compounds. So the waste, you could name urea. And then in the dead plant and animal material, it would be proteins. It could be DNA as well. So you have to name the compound that the decomposers would be breaking down. And this would be our saprobiotic nutrition. So the decomposers will break down the proteins to release the nitrogen, and it converts it back into ammonium, and then the cycle can go round again. If though there is a lack of oxygen within the soil, we then have anaerobic denitrifying bacteria taking over, and they can convert the nitrates back into nitrogen gas. So that's how we get the nitrogen going back into the atmosphere. So other than the denitrifying bacteria stage, all of the other stages are oxidation, and they would require oxygen to be present. The other cycle you need to know is the phosphorus cycle. And phosphorus is really important because it's used in the phosphate groups of DNA, RNA, ATP, and in the phospholipids within the phospholipid bilayer. So it is an essential element for life for all organisms. The phosphorus cycle is different to the carbon and nitrogen cycle. Not that you actually learn the carbon cycle at A level. And that is because you do not find phosphorus as a gas in the atmosphere. So instead, the main store or source is as phosphate ions in the mineral form in sedimentary rock. Now you need to know about the mycorrhizae, which are fungal associations which occur between the plant roots and beneficial fungi. Now, although we're talking about this here in the phosphorus cycle, it would also be occurring in the nitrogen cycle as well. The mycorrhizae are going to provide benefits for plant growth. And that's because the mycorrhizae fungal associations, we can see here you get these really long networks of fungi, and they can spread out in the soil to create a larger surface area for the absorption of water and mineral ions going into the roots eventually. The plants provide an advantage to the fungus because the plants through photosynthesis will create carbohydrates, and it can share those carbohydrates with the fungus. So this is a mutualistic relationship because they both provide a mutual benefit to each other for survival. So the phosphorus cycle, if we start in the middle looking at the phosphate ions in the oceans, or it could be within the soil, plants can absorb those phosphate ions through their root hair cells by active transport. Animals will then be eating the plants so they can consume and absorb the material to get the phosphate ions. When animals excrete, that will release phosphorus back into the oceans or the soil in the form of phosphate ions. As well as returning it to the soil, we also have decomposition. And phosphate ions from waste and remains are going to be broken down. So animals which have a shell, but also bones, contain phosphate ions. And those can be eroded to release the phosphate ions back into the ocean or the soil. The other thing you can have, which we can see in this image, is guano. And guano is from bird feces, and it's this white layer you can see on the rocks. So it's a type of excrement which is really rich in phosphate ions. Deposition is when we get phosphate ions building up to create the rocks, and that also links to sedimentation over here. So over time, from high pressure as well, the phosphate ions within the oceans, within the soil, will compact through sedimentation to create the sedimentary rocks. And then the last link back is the fact that those sedimentary rocks can be eroded to release the phosphate ions back into the soil or oceans. Now the extra one added here is the idea of fertilizers. Fertilizers added to soils can also run off from the soil or leach into water sources, so it would return it back into oceans, or it could just be within the soil itself, but that will be adding phosphorus into the cycle. If we just look at fertilizers a little bit further, we said that they can be added to the soil. The reason for that is when crops are harvested, you're removing the plants, and that is therefore removing one of the stores of nitrate or phosphate ions. So it's going to be taken out of the nutrient cycle. And if you continue to do that, eventually you'll have plants and soil which are mineral deficient. So you have to add either natural or artificial fertilizers to replace the minerals and the nutrients that are being removed when you harvest crops. So natural fertilizer would be animal manure. Artificial is soluble chemicals which contain exactly the proportion and quantities of the mineral ions required. Now, the natural fertilizers are cheaper because if you have your own animals on a farm already, it's completely free. It is just the manure from the animals. However, it doesn't contain necessarily the exact proportions of the minerals you need, and you have no control over that. Artificial fertilizers, because they are synthetically created, they are created to have the exact proportion of minerals that plants would require. However, the inorganic substances are very, very soluble, which has the downside of that they can dissolve in the rainwater and leach, which means they're washed away. So those soluble chemicals such as nitrate, phosphorus, dissolve in the water and then they leach, which means they run off from the rainwater into nearby water sources, such as rivers or ponds. If that nitrogen fertiliser leaches into waterways, it causes eutrophication. And eutrophication is when nitrates are going to stimulate the growth of algae in a pond or a river. It's more likely to happen in a pond though, because in a river the movement of the water can prevent the algae from growing or staying in one place. So the excessive growth of algae creates this blanket on the top of the water. That blocks the light. As a result, plants below that blanket aren't going to be receiving light for photosynthesis and they end up dying. Bacteria within the water will then feed and respire on the dead plant matter, and in doing that they're going to be using up the oxygen within the water, and you get more and more bacteria growing and thriving because of all that dead plant material to feed on, so even more oxygen is used up, and eventually there won't be enough dissolved oxygen within the water for animals, and animals such as fish and other aquatic organisms will therefore die because there's no oxygen for respiration. We begin with one of the key definitions, and that is of the term stimulus. So a stimulus is a detectable change in the environment, and the cells that can detect these changes are called receptors. So organisms increase their chance of being able to survive if they can respond to these changes in the environment. And we look at in this topic the different mechanisms for how organisms do this. So we begin with the responses in flowering plants. And tropism is the term given to when a plant responds to its surroundings via growth. Now, tropisms can be described as either positive or negative. Positive meaning the plant grows towards the stimulus, negative meaning it grows away. And the two stimuli that you need to know are light and gravity. Now, tropisms are actually controlled by specific growth factors, and one key example is IAA. Now, IAA is a type of auxin and it can control cell elongation, meaning making the cells get longer. And this is in shoots, it elongates, but having IAA in the roots actually inhibits the growth of cells. IAA is made in the tip of both the roots and the shoots, but it can move through by diffusion to other cells. Phototropism is the term given to the tropisms where the plant is responding to light. And for shoots, the reason that it improves their survival chance if they respond to light is because light is needed for the light-dependent reactions in photosynthesis. So plants' shoots will grow and bend towards the light source, and this is positive phototropism. Now the shoot tip cells, as we already said, those will be producing IAA, and that causes cell elongation, causing the shoots to grow upwards towards the light source. Now the IAA diffuses to the shady side. So in the first images we can see that the IAA is diffusing downwards away from the light source. However, in the other images we can see we've got a unilateral light source, meaning it's coming from one side. And picture 3 shows you the light to the right hand side. And the IAA will still diffuse to the shady side, that just happens to be on the left. And that will cause the cells on that side to elongate, and therefore the shaded side cells will be stretching and that side will be longer, and that causes the plant to end up bending towards the light source. In the roots we have the opposite happening. The IAA will still diffuse away from a light source, but this time we said that in the roots IAA inhibits the growth. So as a result, the top layer of cells will be growing or elongating more, and that is why the root ends up bending downwards away from the light. And this helps survival because that will help anchor the plant further into the ground and potentially reach more water sources. So that's an example of negative phototropisms. Gravitropism is the term given for the responses to gravity. And in the shoots, the IAA will diffuse from the upper side to the lower side of a shoot. So it's diffusing down towards gravity. And if a plant shoot was vertical, then that would mean the IAA would be diffusing down, causing the lower cells to elongate, and the plant would be growing upwards. Now the image we can see down here is if a shoot was on its side, and that will cause the shoot to bend upwards, and that's because the IAA diffuses down, the cells on the lower side will elongate, and that is why it's bending up. And this is against gravity, so that would be an example of a negative gravitropism. Now in the roots, IAA moves to the lower side of the roots also, and that would cause the upper side to elongate and the root will bend down towards gravity. And that would be a positive gravitropism. And that increases survival because again it helps to anchor in the plant. Now a reflex is an example of a rapid automatic response to protect animals from danger. A reflex arc is made up of 3 neurons, and that would be the sensory neuron, we'd then have the relay neuron and the motor neuron. So it's only 3 neurons, that means you're only going to have 2 synapses, and that is one of the reasons why it's such a rapid response. Now some more simple responses are taxis and kinesis, and these keep organisms within their favourable conditions within their environment. In particular, we're thinking about light, moisture and potentially chemicals as well. Taxis is one of these types of responses, and this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So the example we have here is an earthworm, and earthworms don't want to be in the light, that would dry them out. So if it is light, they would move their entire body away towards a darker light source. So when an organism moves towards a stimulus, this is known as positive taxis. When an organism moves away, it's described as negative taxis. So the earthworm moving away from the light would be negative phototaxis. Kinesis is slightly different. This is when an organism changes the speed of movement and the rate that it changes direction. So if an organism moves from an area where there are beneficial stimuli in the area within the harmful stimuli, its kinesis response will be to increase the rate it changes direction, and that's to increase the likelihood that it will return itself to the favourable conditions. If an organism, though, is surrounded by the negative stimuli, the rate of turning decreases to keep it moving in a relatively straight line, and this will actually increase the chances of it moving to find a new location with the favourable conditions. Receptors we talked about at the start, so these are cells which can detect a change in the environment or stimuli, and each receptor responds only to one specific stimuli, and this stimulus of a receptor can lead to an action potential or establishing a generator potential. And that is what results in an organism being able to respond. Now there are 3 receptors that you need to know: the Pacinian corpuscle, rods and cones, which are both light receptors or photoreceptors found in the eye. So if we start with the Pacinian corpuscles, these detect changes in pressure, so they're pressure receptors, and you mainly find them deep in the skin of your fingers and feet. And this is what we're seeing here. This is a cross section through your skin, and the Pacinian corpuscles are about the same level, slightly lower than your hair follicles. Now, the Pacinian corpuscle is actually a sensory neuron wrapped in lots and lots of layers of plasma membrane, and that has special channel proteins embedded within those plasma membranes. So that's what we're seeing here. We've got our sensory neuron with a nerve ending. We've got lots of connective tissue with gel in between each of these layers. Now, the membranes of the Pacinian corpuscle have stretch-mediated sodium channels. So where we said it's special channel proteins, that's what we're referring to. And what this means is when these proteins have pressure applied to them, they are stretched and deformed and it opens the channel. And in this case, it opens these sodium ion channels. So if pressure is applied to the finger, for example, the Pacinian corpuscles deep in the skin of your finger will be stretched and deformed. That will widen the sodium ion channels, and therefore sodium ions will be diffusing in, which leads to the establishment of this generator potential. So it's diffusing into the axon within the sensory neuron, and if enough sodium ions diffuse in to exceed that threshold, then you have an action potential and a response will occur. The next two receptors are both found in the human retina, and these were the photoreceptors, rods and cones. Now, rod cells are named because of their shape. They are more rod-like in shape, and they can only process images in black and white. Now, to create the generator potential, the pigment in the rod cells, which is rhodopsin, must be broken down by light energy. Now, they can detect light at very, very low intensities. And the reason for this is many rod cells connect to one sensory neuron, which we call retinal convergence. Now, although this gives the benefit that you're able to see in black and white, even if it's very dark, because all of those connecting rod cells contribute towards reaching that threshold to generate an action potential, the downside is it provides low visual acuity. And what that means is it's difficult for the brain to distinguish between the separate light sources. So you don't have very good clarity in your vision. So that's why at night time, if you do happen to wake up, you'll be able to see, but it'll only be black and white, and you won't be able to see particularly clearly, which is why you might end up bumping into things if you try and walk. Now, the other type of cell that is a photoreceptor is the cone cells. And there are three types of cone cells, and each one contains a different type of iodopsin pigment. There's one that is for red, green, and the last one blue. And they all absorb slightly different wavelengths of light. And we don't just see red, green, and blue in our vision, but depending on the proportion of red cone cells or green or blue cone cells that are detecting a stimulus, that will give the different colours, not just red, green, and blue. Now, iodopsin is only broken down if there is a high light intensity. So you can only see in colour if there is enough light present. And the reason for that is we only have one cone cell connecting to one sensory cell. And therefore there has to be a high enough light intensity to break down enough of the iodopsin pigment to be able to trigger an action potential. Now that gives the property of high visual acuity. So because only one cone cell is connected to one sensory neuron, that means the brain can distinguish where the separate source of light was detected, and therefore you have a lot more clarity and precision in your colour vision. Now, the distribution of the rods and cone cells is not equal across the retina. You actually have most of the cone cells located in this part here called the fovea. And that is where you have the highest light intensity reaching the retina, because that is where the light is focused by the lens. Now that is an advantage because the cone cells will only be able to detect the colour lights in high light intensities. And that's why you mainly find them at the fovea. The rod cells can still detect light even at low light intensities. And that is why they are found further away, where there'll be slightly less light being able to reach that part of the retina. Now, the next thing you need to know about is how your heartbeat is controlled. Cardiac muscle is myogenic, and what that means is it will contract and relax on its own accord without any nervous system input. However, the speed at which it contracts is controlled by the nervous system. And that is what you need to know about in this section of the A-level. Now, the parts that are involved in controlling the heart rate are, first of all, the sinoatrial node, or the SAN. And this is located in the right atrium. This group of cells or tissue is known as a pacemaker, and that's because they set the pace at which your heart will beat. You then have the AVN, or the atrioventricular node, which is located between the right atrium and the left ventricle. The bundle of His is what we call the tissues running down the septum. And then you have the Purkinje fibres, which branch through the ventricle walls. Now you can see it on the outside here, but it'll be branching all the way around the ventricle walls. Now, how those all work together to control the heart rate is linked to what you would have learnt in the topic 3 when you did the cardiac cycle. So when you learnt about atrial systole and atrial dystole, or diastole and systole, people say it differently. So the SAN starts off this process by releasing this wave of depolarization, or electricity, and that will spread across the two atria. And when the wave of depolarization hits the muscle, it causes it to contract. So that is why the atria contract first. When that wave of depolarization reaches the AVN, that causes the AVN to release another wave of depolarization. Now, there's actually a non-conductive layer of tissue that separates the atria and ventricles, and that prevents the wave of depolarization travelling straight down to the ventricles. So instead, what happens is that wave of depolarization that was released by the AVN travels down the bundle of His and then up through the Purkinje fibres. Now, because of that, it causes a very slight delay in the time it takes for the apex of the heart, so the base of the ventricles and the ventricle walls, to contract. And that is actually an advantage because that means it gives the atria enough time to contract and force all the blood into the ventricles before the ventricles will then contract. And the ventricles would contract from the apex first and then move upwards. And that's an advantage because it's a bit like squeezing toothpaste out of your toothpaste tube. If you squeeze from the bottom upwards, you're going to get all the toothpaste out. And it's the same here. Squeezing and contracting that cardiac muscle from the bottom, then upwards, forces all of the blood out of the heart. Finally, all of the cells repolarize, and that causes the cardiac muscle to relax, and then the cycle happens again when the SAN releases another wave of depolarization. Now, the involvement of the nervous system is what triggers the rate at which the SAN will release those waves of depolarization. And the parts involved are the medulla oblongata in the brain. That is what controls the heart rate, but this is through the autonomic nervous system, meaning it's automatic and there's no conscious control. So you can't think and control your heart rate. There are two key parts, and that is the sympathetic nervous system and the parasympathetic nervous system. And that's what we're being shown here, these two key routes that the impulses can take connecting the medulla oblongata to the SAN in the heart. Now, the sympathetic nervous system, if impulses travel via the sympathetic nervous system, that will cause the SAN to release the waves of depolarization more frequently and therefore increase the heart rate. If more impulses are sent down the parasympathetic nervous system to the SAN, that will cause the SAN to release these waves of depolarization less frequently, and therefore it decreases the heart rate. One of the stimuli that will change the heart rate is pH, and the other one is blood pressure. There are different receptors that detect these different stimuli. The changes in pH are detected by the chemoreceptors, and the blood pressure changes are detected by pressure receptors. but they are both found in the same location. You find them in the walls of the aorta and also the carotid artery, and this is the next blood vessel that attaches onto the aorta. Now, your blood pH does change, and during times of high respiratory rate, say for example maybe if you're doing lots of exercise, the blood pH will decrease, and that is because of the excess carbon dioxide from aerobic respiration or lactic acid from anaerobic respiration. And these acidic compounds have to be removed because if you have excess acid in your blood, it can denature enzymes. So this is achieved by increasing your heart rate so that the dissolved carbon dioxide in your blood can reach your lungs faster and therefore diffuse out into the alveoli and be exhaled. So that would mean you need more impulses going down the sympathetic nervous system to the SAN to increase the heart rate. Now, for blood pressure, if it's too high, it can cause damage to the walls of the artery. So it's very important to put mechanisms in place to bring down your blood pressure. And to do that, you would have more impulses sent via the parasympathetic nervous system to cause the SAN to fire less frequently and therefore decrease the heart rate. If your blood pressure is too low, though, that might result in insufficient oxygenated blood being supplied to respiring cells. So that would mean that your heart rate needs to be increased to try and counteract this. So therefore, the pressure receptors would detect this and result in more impulses being sent via the sympathetic nervous system to the SAN to cause the SAN to fire more frequently and therefore increase the heart rate. Now, if we have a look at a myelinated motor neuron structure, the key parts are, first of all, the cell body. And this cell body contains all the organelles you'd have in a typical plant cell. So, for example, we can see the nucleus. It also has proteins and neurotransmitter chemicals which are made in that part of the cell. The dendrites we can see here branching carry action potentials to surrounding cells. The axon is the conductive long fibre that is making up this whole part of the cell, and that carries nerve impulses all the way along this particular motor neuron. Schwann cells are what we call these layers wrapping around the axon, and they form the myelin sheath, which is a lipid, and therefore it insulates. It does not allow the charged ions to pass through to the axon. There are gaps though, and these gaps are called the nodes of Ranvier, and that is where they are not insulated. A resting potential is what we would have happening inside of the axon when there is no stimulus. So when a neuron is not conducting an impulse because there's no stimulus, there's no change in the environment, there is still a difference between the electrical charge inside the neuron, so inside the axon, and outside. And we call that difference the resting potential. There are more positive ions, the sodium ions and potassium ions, outside compared to inside of the axon. And therefore the inside of the neuron is comparatively more negative, and that is why the resting potential is minus 70 millivolts. Now you need to know how that resting potential is established in the first place after a response to a stimulus, and how it's also maintained if there is no stimulus. Now the key way it's maintained is by a sodium-potassium active transport pump. So this is an example of co-transport, but it is also active transport because ATP is involved. And at this pump you have 2 potassium ions being pumped into the axon, and 3 sodium ions are being actively transported out of the axon. This creates an electrochemical gradient, and therefore the potassium ions, because there's a higher concentration of them inside the cell, they will start to diffuse out of the cell. The sodium ions, there's a higher concentration outside, so they will start to diffuse into the cell. However, the membrane is more permeable to potassium ions because there are more potassium ion channels and because some of those ion channels remain permanently open. So that means more potassium ions can diffuse out than sodium ions diffusing in. And as a result, there are more positive ions outside of the cell compared to inside of the cell, and that's how we get the minus 70 millivolts. Now, if there is a stimulus, and if it's a large enough stimulus, it can generate an action potential. An action potential is when the neuron's voltage increases beyond a set point from the resting potential. And this is what generates the nerve's impulse to then travel along the neuron. So an increase in voltage, also known as depolarization, is due to the neuron potential becoming more permeable to sodium ions and therefore becoming more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So let's have a look at then how this action potential is generated. A stimulus provides the energy that can cause the sodium ion voltage-gated channels in the axon membrane to open, and this will cause the sodium ions to start diffusing in. And that's what we can see here. One of those channels has now opened, and therefore sodium ions will start to diffuse into the axon. The potassium ion channel here is permanently open, but because we now have some sodium ions moving in, we start to see this increase in the voltage. Now, if the stimulus is large enough to cause enough of these sodium ion channels to open, then you'll have enough sodium ions diffusing in to cause an increase beyond the threshold potential. And if you do have an increase beyond that minus 55 millivolts, then you will always have the rest of this curve happening. You'll always have an action potential generated. Now, there are even more voltage-gated sodium ion channels. So as that voltage will start to increase, that will cause more sodium ion channels to open. Then we have this positive 35 to 40 millivolts. What then happens though is we see this decrease, and that's because when it does reach this particular voltage, it then causes the voltage-gated sodium ion channels to close. So no more positive ions are entering, but we still have those potassium ions moving out. So we start to get this decrease, which we call the repolarization. That will then cause more potassium ion channels to open, so even more potassium is diffusing out. And eventually, so many positive ions have diffused out, we get to that resting state, but actually it overshoots and goes to minus 80 millivolts, which we can see here. We're now back to that original state of potassium ions would be able to move out, but all of the other gates are closed. So that is the generation of an action potential. And this happens along the axon multiple times. This graph is not a change over distance. So this isn't happening just once along the axon. It happens right at the start of the axon, and then at every node of Ramvir, if it is a myelinated neuron, you'll then have another action potential being generated until you get to the end of that neuron and then a synapse. So it's a bit like the idea of a Mexican wave moving along. If it was unmyelinated, then same idea, but it has to happen at every single position on the axon because you wouldn't have the myelin sheaths. So that would mean it would take longer for all of those action potentials to finally reach the end. Now you need to know about this all or nothing principle, and it links to the threshold. So if the axon isn't depolarized to at least minus 55 millivolts, which is the threshold to cause the rest of the action potential to be generated, then the impulse will not be produced. So what we mean by the nothing part of the principle is, If the stimulus isn't large enough to cause a voltage increase to at least minus 55 millivolts, then nothing more will happen. And that's what this bit is showing you here, some failed initiations of an action potential. The all part of the principle is the fact that if you do exceed that threshold, then you will always have an action potential and it will always peak at exactly the same point. So even if the stimulus is bigger, so brighter light compared to dimmer lights, you will still have the same peak. But what changes is the frequency of those action potentials being generated. So that is the all or nothing principle, and the reason it's important is to make sure that animals are only responding to stimuli which are large enough rather than responding to every single slight change in the environment. Because if we did that, it would actually overwhelm your senses and it wouldn't necessarily be protecting you from stimuli which might be causing you danger. Now, the refractory period we mentioned is when it goes into this minus 80 millivolts. But the importance of this is, once we have this stage, we have a period when another action potential can't be generated straight away. And that's because these sodium ion channels, we describe as they're recovering. But essentially what we mean is, because we've now gone even more negative, you would have to have even more sodium ions to be able to reach that threshold. The importance of this is, first of all, it ensures that discrete impulses are produced. So an action potential cannot be generated immediately after another, and that makes sure each one is separate. So that helps with you being able to identify the source of the particular stimulus. It also makes sure that the action potentials will only be travelling in one direction along the axon. So it stops the action potential from spreading out in both directions, which would actually result in the impulse not travelling along. It limits the number of impulses as well, and this is important to prevent you overreacting to a particular stimulus or over-responding. Now, the factors that will affect the speed of conduction are myelination and saltatory conduction, the axon diameter, and temperature. So the first one was myelination, and that results in saltatory conduction. And what that term means is the action potentials will jump from node to node, meaning the node of Ranvier. And that is because this myelin sheath created by the Schwann cells is insulating, so the ions cannot diffuse in and out during these sections, so it jumps to the parts where there is no insulation. Now, as a result, that means that the impulse will travel much faster along the axon because you don't have to generate as many action potentials. You're only generating them at those nodes. Next then, the axon diameter. The wider the diameter, that is going to increase the speed. And the reason for that is basically like the idea of physics and wires and electricity. So the wider the diameter, that means we're going to have less leakage of the ions, and therefore the action potential can move faster. Temperature, there's two reasons why this has an effect. Number one, because if you have a higher temperature, the ions will have more kinetic energy. That means they'll be able to diffuse faster. The second reason is the enzymes involved in respiration to provide ATP for the sodium-potassium pump will be able to work faster as well if there is a higher temperature. Now, if it does exceed the optimum of around 37, then that could actually decrease the speed. So synapses are the gaps that we have between the neurons. So once you have your action potential reaching the end of a neuron, it has to get to the next one. And in order to do that, we have to have neurotransmitters, which are chemicals, diffusing across the gap, which will then generate the action potential in the next neuron. Now, the way that this happens is, first of all, your action potential, as I said, it will arrive at the end of the neuron, and we describe that as the synaptic knob. Depolarization of the synaptic knob leads to calcium ion channels opening, and therefore calcium ions will diffuse into the synaptic knob. Those calcium ions will cause the vesicles that contain the neurotransmitter to move towards the membrane, fuse, and release the neurotransmitter into the synaptic cleft. Because we have a high concentration of the neurotransmitter where they've just been released, they'll then diffuse across to the postsynaptic membrane. And on the postsynaptic membrane, there are receptors, complementary in shape, for those neurotransmitters to bind to. When those neurotransmitters bind to the receptors, it causes the sodium ion channels on the postsynaptic membrane to open. Therefore, sodium ions will diffuse in, and if enough sodium ions diffuse in to exceed that minus 55 millivolt threshold, then the action potential is generated. So that will then cause that action potential to be generated in the postsynaptic membrane, and then it can continue along the axon of the next neuron. Now, this is unidirectional, and you could be asked, how is it ensured that it's unidirectional? And there's a couple of ideas. Number one, the vesicles containing those neurotransmitters are only in the presynaptic neuron, so they can only be released from this side. Because of that concentration gradient, diffusion happens from the pre to the postsynaptic neuron, and you only find the receptors complementary in shape to those neurotransmitters on the membrane of the postsynaptic neuron. Now, one example of a synapse that you need to know is the cholinergic synapse. So it's exactly the process that we just went through, but you need to know the name of the neurotransmitter. So the neurotransmitter is acetylcholine. It will bind to its receptor, and then it will go through the whole stages that we said. But those neurotransmitters don't remain permanently bound. If they did, that would mean the sodium ion channels would be continually open, and you'd continually be triggering this action potential and a response, even though the stimulus wasn't present anymore. So to prevent that happening, there is an enzyme that will break down the acetylcholine into choline and acetate, which can then be reabsorbed into the presynaptic neuron and be reused. And that neuron is acetylcholine esterase. Now, I said that in order for the action potential to be generated in the postsynaptic neuron, you needed to have enough sodium ion channels opening. And that is caused by the neurotransmitters binding to the receptor. And there's two different mechanisms of summation to ensure that happens. Summation is referring to the fact that you have this rapid buildup of neurotransmitter to generate the action potential. And you can either have spatial or temporal. Spatial is when you have multiple neurons connecting to the same postsynaptic neuron. And collectively, these three neurons in this example should release enough neurotransmitter to cause enough of those sodium ion channels to open on the postsynaptic neuron. The alternative is temporal, and this is where you only have one presynaptic neuron connected to one postsynaptic neuron, but instead the way that this will cause enough neurotransmitter to be released is you have repeated releasing of that neurotransmitter over a short period of time until eventually you have enough neurotransmitter bound to enough of the receptors on the postsynaptic neuron's membrane to open enough of those sodium ion channels. You also have inhibitory synapses, and in this case you would have chloride ions would be moving into the postsynaptic membrane. So you'd have different neurotransmitters binding to their complementary receptor, but this time it would cause chloride ion channels to open. And because you'd then have negative ions moving in, this would cause the membrane potential to decrease to at least minus 80 millivolts, and therefore the postsynaptic membrane would go into this hyperpolarization, making it very unlikely that an action potential could be generated. Neuromuscular junctions are what you have right at the end of your response arc. So you have your motor neuron, you then have a gap, and then you have your muscle. and your muscle is one of the effectors that would generate the response. And what you need to know is similarities and differences between the synapse that you find here at the neuromuscular junction compared to your typical synapse such as the cholinergic. So what they have in common is both of them will work in one direction only, so unidirectional. And that is because the receptors for the neurotransmitters are only found on the postsynaptic membrane, or in this case, the muscular membrane. A neuromuscular junction will only be excitatory, meaning it will only generate that response. Whereas a cholinergic, we looked at, could be excitatory, generating an action potential, or it could be inhibitory, which prevents the action potential from being generated. The neuromuscular junction will have a neuron connecting to a muscle, and it's always the motor neuron. The cholinergic is just a synapse between any two neurons. The neuromuscular junction is always at the end point for the responses, and the cholinergic synapse is always going to be generating a new action potential. For the neuromuscular junction, the acetylcholine will bind to receptors on muscle fibre, whereas the acetylcholine binds to receptors on the postsynaptic membrane in the cholinergic synapse. So that could be a long answer question, it could be a comparison table, so definitely learn those comparisons for those two. So that leads us on to muscles, which is an example of an effector, meaning it carries out the response. And muscles always occur in antagonistic pairs against the skeleton. And what that means is you have two different muscles that will be doing the opposite thing at the same time. So as one of them contracts, the other relaxes. Now this could be automatic or it could be conscious control for this movement. Now you need to know the structure of these muscles. So that's what we're looking at here. And we have the myofibrils labelled, and a myofibril is made up of lots of fused cells, and because they're fused together they will share organelles. So they share the nucleus, they'll be sharing the cytoplasm, and this cytoplasm, because it's all shared, is known as sarcoplasm. So there'll be lots of nuclei within one of these myofibrils, but there's also a high number of mitochondria, and that is because we'll be needing ATP from respiration for the muscle contraction. Now these myofibrils are all bound together to make up this muscle fibre. But what we're actually going to focus on is the myofibril in more detail, having a look at the sarcomere. Now the muscle fibres are made up of millions of myofibrils, which collectively bring about that force to cause the movement of the skeleton. And the myofibrils are made up of two key proteins, myosin and actin, and that is what forms the sarcomere. Sarcomere you can actually look at in terms of different sections or bands, depending on whether you're looking at a part which is just made up of the actin protein, the myosin protein, or where the two proteins overlap. And that's what this diagram is representing. The actin is shown in red, the myosin is shown in blue, and what you can actually see from the myosin is the position. You have these chains of myosin with heads sticking off, that's what these lines at right angles are showing, but also the myosin molecules are all bound together, and that keeps them in a fixed position. The actin you just have as a thinner filament, and they can slide closer together towards this M line. Now you could be asked to describe what happens to the length of the different bands when a muscle contracts. So the A band is the length of the myosin, so that should never change. The A band is constant because the myosin itself, apart from the heads, the actual length of the myosin is never going to move. The H zone is where you just have myosin with no actin overlapping. Now that will change when a muscle contracts, because when a muscle contracts, the actin is sliding in, so therefore the H band or H zone is going to decrease because that is where you have myosin by itself. The I band is also going to change in size because the I band is where you have just actin by itself. So when the muscle is relaxed, you have a bigger I band. When it contracts, the I band decreases because the actin has slid closer together, so you have more of an overlap with the myosin. You could be asked about what happens to the Z lines, and the Z lines are indicating the start and the end of one sarcomere. And because the actin slides in, the Z lines become closer together as well. Now, my tip for learning this is, it isn't really intuitive, the letters representing these bands. So I'd say put them on a flashcard. So A band on one side, on the other side, just say it's the length of the myosin and it remains constant. The H zone is where you just have myosin by itself, decreases when muscles contract. So create a set of flashcards for this and just learn that content. This is just showing you the image as well of this happening under the microscope compared to the diagram. Now, the sliding filament theory is how the muscles actually contract. So when an action potential reaches the muscle at that neuromuscular junction, it's going to stimulate the muscles to contract. It does this by causing calcium ions to release from the sarcoplasm. And what this does is it causes the tropomyosin, which is the protein that covers the actin binding sites. It causes it to move out of the way, and therefore these binding sites are now exposed. Whilst ADP is attached to the myosin heads, which we can see here, the myosin head is able to bind to these exposed actin binding sites, and it forms what we call a cross bridge. The angle that this creates with the myosin head binding is going to result in some tension, and that causes this pulling or sliding action of the actin along the myosin. And we call that the power stroke, which we can see just here, when that head causes the actin to slide along. Now, in that power stroke action, the ADP and PI are released. ATP is the molecule that then binds to the myosin head and causes it to change shape slightly, and as a result, it detaches from the actin molecule. Within the sarcoplasm, there's an enzyme, ATPase, and that is activated by the calcium ions that were released in the sarcoplasm. So as long as you still have the stimulus causing these calcium ions to be released, then the next step will happen to trigger even more contraction. So that enzyme ATPase is going to hydrolyze the ATP that was attached to the head into ADP and PI. And that now releases energy for the myosin head to return back to its original position. And then we're back to the start again. That process repeats as long as the calcium ions remain high, the tropomyosin is moved out the way, and the myosin head combined. So this will repeatedly happen to slide the actin as close together so you have this full muscle contraction. So that continues to happen while the stimulus is causing that release of calcium ions. So you need to have ATP for this process to happen, but also a molecule called phosphocreatine. Now, if the muscles have been very active, we just saw there, it requires ATP. So the muscles require a high concentration of ATP to be able to contract. And the chemical phosphocreatine, which is stored in muscles, assists this process because it provides the phosphate. So in order to regenerate ATP, you need that inorganic phosphate to bind with ADP, and that's provided by the phosphocreatine. The last thing you need to know about muscles is two different types of muscles, slow twitch and fast twitch fibres, and you need to be able to compare their structure, location and general properties. So the slow twitch contain a large store of myoglobin, which is a type of hemoglobin which has a very high affinity for oxygen, and therefore it can hold on and store oxygen until the partial pressure is really low, and then it unloads. It also has a rich supply of blood, and that is so it can constantly provide oxygen, and many mitochondria, so you can have lots of aerobic respiration. So all of those structures link to the fact that these slow twitch fibres do contract slower, but they respire aerobically and for longer periods of time because of those structural adaptations. and that is for exercises which are going to be endurance, or it could be just regular exercises as well, just regular movements, nothing intensive. So for example, calf muscles when you're walking. Fast twitch fibre muscles are much thicker and there's more myosin filaments. They have a large glycogen store and a phosphocreatine store to help make the ATP. They also have a high concentration of enzymes needed for anaerobic respiration. Now, all of those structural features link to the properties of it contracts faster, short bursts of powerful contractions as well. So this is for intense exercise, and as we said, it'd be anaerobic respiration. And an example would be your biceps when you are contracting your biceps. So the last bit in this topic is homeostasis, and this involves physiological control systems to maintain your internal environment within set restricted limits. And the examples that you need to know are maintaining the core body temperature and maintaining the blood pH as well in relation to enzyme activity. Now for those two, you don't really need to know much of the details. It's just knowing that those two conditions are controlled, and you'd need to know why in terms of the effect it has on enzyme activity. Homeostasis involves negative feedback, and this is when any deviation from the normal values, so if it goes too high or too low, there are mechanisms put in place to bring the conditions back to their original level. And it involves the nervous system and often hormones as well. Two key examples you need to know are blood glucose concentration and water potential. So we'll start with the blood glucose. Now your blood glucose concentrations will increase after you have eaten food containing carbohydrates or drinks containing carbohydrates, and they'll decrease if you haven't eaten food containing carbohydrate for a while or if you have been exercising. The pancreas is the organ that detects these changes in blood glucose levels, and within the pancreas there are cells called the islets of Langerhans, which are responsible for releasing the insulin and the glucagon, which are the two hormones that bring blood glucose levels back to normal. Insulin is going to be released when blood glucose levels are too high, and that will bring blood glucose levels down to normal. Glucagon is released when the blood glucose levels are too low. So think of glucose gone, glucagon. And this will cause an increase in the blood glucose levels. Adrenaline, which is released by the adrenal glands, which are found just above the kidneys, this hormone is released when your body anticipates danger, and this results in more glucose being released from the hydrolysis of glycogen in the liver. Now, just to look at this as an overview, so your normal blood glucose levels, after you've eaten, they will increase. And this will be detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will release insulin. That will cause the liver cells to become more permeable to glucose, and there'll be enzymes activated to convert the glucose that's been absorbed into the liver cells into glycogen. That glucose then has now been removed from the blood and stored as insoluble glycogen, and therefore the blood glucose levels have been restored to normal. Now, if you haven't eaten food containing carbohydrates or you've done a lot of exercise, your blood glucose concentration will decrease. This is detected by the alpha cells in the islets of Langerhans in the pancreas. And it will cause the alpha cells to release glucagon, and also the adrenal glands will be releasing adrenaline. The second messenger model occurs to activate enzymes to hydrolyze the glycogen into glucose. That glucose is released back into the blood, and it brings those blood glucose levels back to the normal range.
So let's have a look at the action of insulin. First of all, it's the beta cells in the islets of Langerhans that detect when blood glucose levels are too high outside of the normal limits. And this then results in the beta cells secreting insulin in response to this. And these are the three key effects that insulin will have to decrease the amount of glucose in the blood. First of all, when that insulin binds to receptors on the surface of target cells, so on the cell surface membrane of the cells in the liver and also in the muscles, it causes changes to the tertiary structure of the channel proteins, which are the channel proteins responsible for transporting glucose. And this change in structure of them results in more glucose being absorbed from the blood into the cells by facilitated diffusion. So that's one way that the blood glucose levels will decrease. Not only that, but when the insulin binds to those receptors on the cell surface membrane of the liver cells, it causes more protein channels to be incorporated into the cell surface membrane. So both points 1 and 2 result in more glucose being absorbed from the blood into the cells. The final step is converting that glucose that is now in the cells instead of the blood into glycogen. And this step has to happen because if we don't convert glucose to glycogen, glucose is soluble, so it will lower the water potential of the cytoplasm in the cells, resulting in more water entering the cell by osmosis, and the cell might burst, or we might have lysis. So this conversion of glucose to insoluble glycogen is glycogenesis, and when the insulin binds, it also causes that to happen in the liver cells. So this is just a visual representation of some of those stages that we talked about. So we've got here the insulin hormone is bound to the complementary shaped insulin receptor on the cell surface membranes of liver cells. When it binds, what happens is actually a cascade of reactions that you don't need to know about, but it causes this release of chemicals, and those chemicals cause vesicles that are within the liver cells, which is what we're looking at here, these vesicles, which is just a sphere of phospholipid, and we've got the channel proteins embedded within that. And when these chemicals are released, it causes these vesicles, which contain those glucose protein channels, to move towards the cell surface membrane, and they then with that cell surface membrane. And as a result, you would then have all of these extra protein channels embedded within the cell surface membrane, so there's a larger surface area for that facilitated diffusion of glucose from the blood into the liver cell.
The action of glucagon, again, there's 3 different actions. It will attach to the receptors on the target cells, and that would be the liver cells or the muscle cells. And when it binds, it causes the protein to be activated into adenylate cyclase. And this causes ATP to be converted into cyclic or cyclic AMP. That activates another enzyme, protein kinase, and that can then hydrolyze glycogen into glucose. So that is your second messenger model, how you have this 2 stages of activation of enzymes until we eventually get the hydrolysis of glycogen. It will also activate other enzymes to convert glycerol from lipids and amino acids from proteins into glucose. So this is the second messenger model. The glucagon binds to the glucagon receptors on the target cell, so for example the liver. That, once it is bound, causes this protein to change shape. And that change in shape to the enzyme adenyl cyclase or adenylate cyclase makes it activated. And the activated form can catalyze the reaction of converting ATP into cyclic AMP. And cyclic or cyclic AMP is the second messenger. So the cyclic AMP, which is this second messenger, is triggering the final stage to happen. It can convert the inactive protein kinase into an active form, and that is the enzyme which can then hydrolyze glycogen into glucose. Now this second messenger model happens for glucagon binding, but also adrenaline. So it's exactly the same process when adrenaline binds.
Now adrenaline is the hormone that is secreted from the adrenal glands in response to anxiety, fear, so it's that flight or fight hormone, that's often how it's described. And the adrenaline will increase blood glucose levels. So if you did have to suddenly fight to survive or run away to survive, that would mean you'd need a lot of muscle contractions, which requires a lot of respiration, which requires glucose. So that's why adrenaline results in this increase in glucose, or that's why it's an advantage. The way it does that is, again, this second messenger model, much like we just saw for glucagon. So all of this is exactly the same as it was for the second messenger model for glucagon. It's just this time it would be adrenaline is the hormone that binds, and it binds to complementary shaped adrenaline receptors on the cell surface membrane of those liver cells, which are the target cells. But the cascades of responses resulting in the second messenger model is the same.
The liver is involved in all three of these processes. Glycogenesis is when glucose is converted to glycogen, and this occurs in the liver and is catalyzed by the enzymes there. Glycogenolysis or glycogenolysis, pronouncing it like that so it's easier to remember, the lysis of glycogen is the splitting or hydrolysis. And again, this happens in the liver, and this is due to the enzymes involved in the second messenger model. Gluconeogenesis is creating glucose from other molecules, so the amino acids and glycerol, and again this happens in the liver due to the enzymes found there. Now, diabetes is when the body cannot produce enough insulin or it's not responding to insulin anymore. Type 1 diabetes is when people are unable to produce insulin or not producing enough. It normally starts in childhood and as a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections of insulin to counteract this. Type 2 diabetes, though, is when the receptors on the target cells lose their responsiveness to insulin. It usually develops in adults as a result of obesity and poor diet, and it's normally therefore controlled by regulating the intake of carbohydrates in your diet, increasing exercise, and sometimes it still involves insulin injections also. Osmoregulation is how the water potential of the blood is controlled. Blood with too low a water potential would be described as hypertonic. Blood with too high a water potential would be hypotonic. And if you had hypertonic blood, that means that too much water will leave the surrounding cells and move into the blood by osmosis, and that would cause your cells to shrivel or crenate. If you had hypertonic blood, that means that the excess water from the blood would move into the cells by osmosis, and it could actually cause the cells to burst. Now, neither of these are good, which is why we have to regulate the water potential. What could cause this is if you had hypertonic blood, that could be from excess sweating, not drinking enough, having a diet that's containing lots of ions, so salty diets. If you had hypotonic blood, then maybe you've drunk too much water or you don't have enough salt in your diet. So the corrective mechanisms for both. If you have hypertonic blood, more water has to be reabsorbed by osmosis into the blood from the tubules in the nephron, which is in the kidney. Therefore, you'll have urine which is lower in volume and more concentrated. If you have too much water in your blood, so hypertonic, that means less water will be reabsorbed by osmosis into the blood from the tubules in the nephrons in the kidneys, and as a result, you'll be producing larger volumes of urine which are more dilute. Now, all of this happens in the nephron, and you need to know the structure of the nephron, which you find in the kidneys. The nephrons are these long tubules. They're surrounded by capillaries, and you have around 1 million of these in both of your kidneys. It's where the blood is filtered to remove the waste, but also selective reabsorption happens to reabsorb any useful substances back into the blood. So the way this happens is, step 1, as the blood is going to flow into the glomerulus, the capillaries there, because there's a really high hydrostatic pressure, water and small molecules are forced out of the capillaries and into the renal capsule, which is this yellow part here, and the glomerulus is all of the red bit there where you have the capillaries. You've then created this filtrate, and that filtrate will then start to pass through to the proximal convoluted tubule. And this is where selective reabsorption occurs. So glucose is reabsorbed in the proximal convoluted tubule. The loop of Henle is where we maintain a sodium ion gradient, and that is to make sure that water can then be reabsorbed back into the blood. So at this point, the filtrate is going to be losing lots of the ions in the ascending limb, and that will then lower the water potential of the surrounding area around the loop of Henle, and then water will be able to move out by osmosis as it goes down the descending limb. So as the filtrate reaches the distal convoluted tubule, it's lost a lot of water, but also quite a lot of ions. And because it's lost a lot of ions, it is actually quite dilute by this stage. So more water will move out by osmosis, and at the collecting duct, even more water can move out to go back into the blood. The collecting duct carries the remaining liquid away to form the urine. Now that is a very basic overview. To have the full details, you will need to watch my full video where I actually go through every one of those steps in a lot more detail. This is just the basic summary video. That would not be enough detail though, so do check that one out up here. So how then this is regulated is using the hypothalamus and the posterior pituitary gland in the brain. The hypothalamus is where the changes in water potential are detected by receptors called osmoreceptors. If the water potential of the blood is too low, water leaves the osmoreceptor cells by osmosis, and that causes them to shrivel. And this stimulates the hypothalamus to produce more of a hormone called ADH, or antidiuretic hormone. If the water potential of the blood is too high, water enters the osmoreceptors by osmosis. This stimulates the hypothalamus to produce less antidiuretic hormone. So the hypothalamus is where the ADH is produced, but it then moves to the posterior pituitary, and from here it is released into the capillaries and into the blood. ADH travels through the blood and to its target organ, which is the kidney. Now what that hormone does is it causes an increase in the permeability of the walls of the collecting duct and the distal convoluted tubule, so they're more permeable to water. And as a result, more water will move out of the nephron by osmosis and it's reabsorbed into the blood. So you'll have more water returning to the blood and more concentrated urine.
So if we have a look then at this overall view of the homeostasis, negative feedback, we've got our normal water potential of the blood. Now the water potential of the blood could increase, so maybe you've drunk a lot of water in a short space of time. And that increase in the water potential of the blood gets detected by the osmoreceptors, which are within the hypothalamus in the brain. The hypothalamus will then produce less ADH, and therefore the posterior pituitary gland will release that less ADH into the blood. That transports around in the blood until it binds to the target cells, which are the cells lining the distal convoluted tubule and the collecting duct walls, and they then become less permeable to water. That means less water is reabsorbed into the blood and more stays in the nephron, which is then within the collecting duct, which goes on towards to make urine. So it results in a larger volume of dilute urine. And as a result, the water potential of the blood returns back to its normal limit. Now, the opposite of that would be if the water potential of the blood decreases, meaning you don't have enough water in the blood. And that could be you've not drunk enough water, or maybe you've been sweating a lot. And this again is detected by the osmoreceptors in the hypothalamus. And this time the hypothalamus will produce more ADH in response, which is then released into the blood by the posterior pituitary gland. That will then transport in the blood, bind to the receptors on the cells lining the distal convoluted tubule and the collecting duct, and that makes the walls more permeable to water. Therefore, more water is reabsorbed back into the blood, and as a result, there are lower or smaller volumes of urine, and it's more concentrated urine, and this results in the water potential of the blood returning into that normal limit.
So the first thing with inheritance is there are a lot of key terms that come up quite often as a 1-mark definition just to start off an inheritance question. So make sure you know all of these definitions as they've all come up at some point in the past. So genotype is the genetic constitution of an organism. Phenotype is the expression of the genes, but also the interaction with the environment, so don't forget that bit. Homozygous is when you have a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you've got the homologous chromosomes carrying 2 different alleles for a single gene. Recessive allele is only expressed if there's no dominant allele present, whereas the dominant will always be expressed. Codominant is when both alleles are equally dominant and expressed in the phenotype. Multiple alleles is when you have more than 2 alleles for a single gene. Sex linkage is a gene whose locus is on the X chromosome. Genes are located on the same chromosome, not the sex chromosomes though, for autosomal linkage. Epistasis is when one gene modifies or masks the expression of a different gene at a locus. And then monohybrid and dihybrid is referring to types of genetic crosses. So monohybrid is when you're looking at the inheritance of just one gene. Dihybrid is when you're looking at the inheritance of 2 genes at a time. And we're going to go through the types of inheritance in more detail and have a look at examples of genetic crosses. Now, a big part of the inheritance topic is being able to determine the outcome of genetic crosses. So to help you with that all in one place, I've got you this genetic coding table. So for the different types of inheritance, this is how you would represent it in a genetic cross. A monohybrid is what you would have done at GCSE. So you pick a letter to represent the gene, capital is the dominant allele, lowercase is the recessive. For codominant, because they're both dominant, you can't go for a capital and a lowercase. So instead, the base letter is representing the gene and the superscript letter is representing the allele. So for example, blood group is IB for blood group B, IA for blood group A, but if you have this here, IBA, that would mean that it's someone that's blood group AB because both dominant alleles will be expressed. Multiple alleles, again, you can't use the upper and lower case because you have more than 2 alleles, and you might have this linked with the idea of codominance as well, as we see in the example of blood groups. The allele that codes for blood group B is dominant, the allele that codes for blood group A is dominant, but there are multiple alleles because there is a third one for blood group O, but that is recessive. For sex linkage, you have to represent this by showing, first of all, the X and Y chromosome. And in sex linkage, the gene is only found on the X chromosome. So you'll only be drawing the allele on the X chromosome, not on the Y chromosome. But we still stick to capital for dominant, lowercase for recessive. Autosomal linkage is always linked to dihybrid because it's looking at the inheritance of 2 genes found on the same chromosome. So we can still use the capital and lowercase, but we'd have 2 different letters representing the 2 different genes. Epistasis would be the same idea as well, because they often don't link it to any of the other types of inheritance because epistasis is complicated enough. So now for the examples. So we're starting off with a simple monohybrid example where we're looking at the inheritance of cystic fibrosis, which is caused by a recessive allele. And we're told in this question that we have 2 parents that are heterozygous, so we can see that represented here. And we're being asked what is the probability they will have a girl with cystic fibrosis. Now don't be thrown, this is not a sex-linked gene, but they still want you to work out the probability of, first of all, whether the child will have cystic fibrosis. And we can see here we have 25% probability that a child would have cystic fibrosis. But because they want you to say the probability of a girl having cystic fibrosis, there's 50% probability they'd have a girl. So we'd then do 25% times 50%, or half of it basically. So there's 12.5% probability that they'll have a child who has cystic fibrosis who is also a girl. Now if we have a look at a codominant example, cows can be red, white or roan in colour, and this is roan here where it's blotchy with both colours expressed. And in this example we have 2 roan cows reproducing, and they want to know what's the probability they'll produce red offspring. So 2 roan cows, because this is codominance, both the red and the white are dominant. That's how it's possible to get roan, where they're both expressed. So here's how we'd represent it for the 2 parents. And then if we do our Punnett square, we can then see we'd have 1 roan, 1 red, actually a second roan down here, and 1 white. So that would mean we have 25% probability of getting a red cow from those parents. Looking at the inheritance of blood groups is an example of multiple alleles and codominance. So here we have the phenotypes that are possible of the different blood groups. Now you don't need to know this. If they wanted you to work out anything to do with blood groups, they would have to give you some information so you could work it out. It's not on the spec, you have to know the blood groups. It's just a really common example to test your understanding of codominance and multiple alleles. So blood group A, you could either have 2 copies of the dominant A or 1 copy of the dominant A and 1 recessive O allele. Same idea for blood group B, but with the B alleles. To be blood group AB, this is our codominance where we have 1 copy of both of those dominant alleles, so both are expressed. Blood group O is recessive, so it's only possible if you have 2 copies of IO, which is our recessive allele. So if we have a look at an example, we have parents with blood group AB and O reproduce, and we're told that A and B are codominant, O is recessive, because as I said, you're not expected to know that. It would be application of your knowledge. And they want to know what's the probability they'll have an offspring with blood group A. So the first thing is you have to state the parental genotypes. Then we can work out the gametes, which I've put here on the outside of the Punnett square. Then we have the offspring genotype and the offspring phenotype. So we can see the probability of having blood group A is 50%. So we have 2 individuals down here that have both got a genotype that codes for blood group A. So now our example of sex linkage, it's looking at colour blindness, which again you don't need to know, it's an application of your knowledge, and you're told that colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be blind? So first of all, we need to work out the parents' genotypes. So the mother is a carrier, so that means they'll have 1 dominant allele and 1 of the recessive alleles that causes colour blindness. And then the male, XY, they do not have colour blindness, so that means they have to have just the dominant allele on their X chromosome. So here we have our gametes. We then have the offspring's genotypes and phenotypes. So the probability that they would have a child that is colourblind is 25%. So epistasis, we said, is when one gene influences or affects, masks the expression of another. So some examples are the coat colour in mice, or in Labradors, fruit colour of vegetables as well, such as squash. And we'll have a look at the Labrador example. So gene 1 controls whether pigment will or will not be expressed, and the dominant allele codes for pigment will be produced, whereas the recessive codes for no pigment, and therefore the dog won't be producing any of the darker pigment and it'll be that yellow colour. Gene 2 controls what colour the pigment will be if it is expressed. So dominant is black, and recessive is brown. So that is how you get the 3 different colours in Labradors. So we have a look at an example. We're going to cross these 2 parents. So the first thing, we've been given the genotypes, but we have to work out the phenotypes. Now both of them have a dominant E, which means pigment can be produced, and they have a dominant B, which means they'll be black. After that, it's working out all the possible gametes that we could have. Then we'd need to do a Punnett square to work out all the possible genotypes of the offspring, and then the phenotypes of the offspring, which I've done for you just here. So we can see we end up with 4 yellow dogs because they have homozygous recessive for that E gene. So even though they might code for black and brown, we won't see that colour because it's been masked by the first gene. Now dihybrid crosses are when we have 2 genes considered at the same time in a Punnett square. So a common example is Mendel's peas. So we've got round and yellow, wrinkled and green as our 2 different traits, the shape versus the colour. And in all of the genetic crosses that we've gone through, it's these 6 features you have to have for all of the marks. We need the parental phenotype, genotype, the gametes, offspring genotype, and then you match the offspring phenotype to the genotypes, and finally give the proportions. So if we have a look at this example, with our round and yellow, round is dominant and yellow is dominant, wrinkled and green are recessive. And in this example, although we've got the two dominant traits, they are homozygous dominant. And they would have to tell you that in the question, or they would have to give you information so you could work it out. So then we'd have a look at our gametes, and each gamete should have one copy of each gene. So you need one of the letter R's for the shape, one of the letter Y's for the colour. Now in this instance, that would mean that all of the offspring would be heterozygous, and you'll always see this pattern. When a homozygous dominant parent is reproduced with a homozygous recessive parent, you will always get 100% of the offspring being heterozygous and therefore showing the dominant trait for both of those genes. Now if we cross the F1 generation though, where both parents are heterozygous, there are now 4 possible different gametes for both parents. And this would be where we get these large Punnett squares, where we put every possible gamete for both parents in the outside box. Then we work out the offspring genotypes, and then we work out all of those phenotypes, and I tend to put it in the box, so you are then definitely getting that mark of matching the phenotype to the genotype. So our ratio here is 9:3:3:1. And if you have a dihybrid cross where both parents are heterozygous for both genes, then it will always be 9:3:3:1, unless there is autosomal linkage or there is crossing over. So let's have a look then at crossing over and how this could affect the ratio. So crossing over results in new combinations of alleles in the gametes. So that means the predicted gametes that we had in our Punnett square may actually differ. So autosomal linkage is when 2 genes are located on the same chromosome, but it won't be the X or Y chromosome. So we can see here we have gene 1 is found on the same chromosome as gene 2. And this is showing us the homologous pair of that particular chromosome. So what this means is that the alleles for each gene are linked on the same chromosome. So they have to be inherited together, because in meiosis, when gametes are made, that whole chromosome will be pulled to create one particular gamete, and this chromosome will then be pulled to the other pole to create the other gamete. So that would mean you would have to have the 2 dominant alleles for the Y and R gene inherited together, and the 2 recessive alleles for the Y and R gene inherited together, as those would be the only 2 types of gametes that could be made. So if we go back to the dihybrid example where we said we have 2 parents that are both heterozygous, we said these were all the possible gametes that could be made. But if there was autosomal linkage, even though we have 2 heterozygous parents, because the R and Y gene are linked on the same chromosome, and we can see we have the 2 dominants and the 2 recessive alleles on individual chromosomes, that actually means the only possible gamete would be one that has the dominant R and Y and one that has the recessive R and Y. So our possible genotypes of the offspring and the phenotypes are here, and we get a very different ratio, a ratio of 3:1, and that then does not fit the typical 9:3 to 3:1 ratio. So that is how autosomal linkage could prevent you from getting that ratio. But we also need to consider crossing over and autosomal linkage impact as well. So we've just talked through this example where, because of the autosomal linkage, we have a 3:1 ratio, but you could actually have this scenario here. Scientists actually observed the following results. So we have 4 different phenotypes, not just 2. And we can see we mainly had yellow and round, and the least common was the green and round. And this is only possible because crossing over has occurred to create new combinations of gametes. So not just these 2 gametes that we said would be possible, new combinations have been made, so there's now actually more gametes. So this is how it would have happened. We can see that we said we'd have the 2 recessive alleles linked and the 2 dominant alleles linked. If we then have crossing over, we can see that the recessive R and the dominant R cross over, that bit of the chromatid will break and exchange, and we now have 4 possible gametes instead. Now you also need to be able to see how chi-squared, the statistic, can link to inheritance. So chi-squared is used when you're investigating differences, but it's when your data is based on categories and frequencies. So if we go back to the very first example we had in this video, this is what we were looking at, the probability that 2 carriers would have a girl with cystic fibrosis. And we said we would expect the probability to be 12.5%. Now, chi-squared is the statistic used to investigate the differences between frequencies, but more specifically to determine whether there is a significant difference between the frequency we expect from our Punnett square and the frequency you actually observe. So let's go through an example. Corn can be purple or yellow and smooth or wrinkled. It's not actually just yellow like you see in the supermarkets, and this is determined by genetics. So using a Punnett square and by counting the actual number of each type of kernel, we can then use chi-squared to see, is there a difference between the expected frequency and the observed frequency? So does the frequency of the purple and yellow corn match the Mendelian genetics you would expect, which basically means the expected value you get from a Punnett square. So purple is dominant, yellow is recessive. And we've got this particular cross from 2 parents, and we would expect from this that we would get a 3-to-1 ratio of purple to yellow. A student then counted the actual number of each coloured kernel, and they recorded 21 purple and 13 yellow. So we can then use the chi-squared to see, do we have a difference between the expected ratio and the observed ratio? So we always state that there is no significant difference between the expected and the observed frequencies in the null hypothesis. And what we'd first have to do is convert the ratio, which is what we use in the Punnett square, into an actual expected frequency. So this is what was observed. So the total number of corn was 34. So if we have a 3-to-1 ratio is what we expect, the frequency that we would expect from that value would be 25.5 and 8.5. We would then do our observed minus expected. We square that, divide that by the expected, and we get our chi-squared value. In this example, the number of degrees of freedom is 1 because it's the number of categories minus 1. And we have 2 categories, purple or yellow. So the last thing you would need to do would be compare your chi-squared value to a critical value. And in this case, the calculated value of chi-squared we said was 3.176, and our critical value, we have 1 degree of freedom, so we need to look across at the row 1 and we go to 0.05, and that would be telling us that there'd be 5% probability, the results are due to chance. We always compare at this point to be able to say if we have a significant difference or not. And in this case, we can see that our calculated is less than the critical value. So that means there's more than 5% probability the results or the difference between the observed and the expected is due to chance. So that means we have to accept the null hypothesis, which was that there will be no difference between the expected and the observed frequency. And what that means is the expected frequency that we calculated using our Punnett square was the same as the frequency we observed. Or in other words, it did follow the Mendelian genetics that we expected.
So you also need to know about the Hardy-Weinberg principle, which is essentially a mathematical model which can be used to predict the allele frequencies within a population, which means how common one particular allele is within that population's gene pool. So just to review some of these key terms then, gene pool, population and allele frequency. So the gene pool is all the alleles of all the genes within a population at one time. Population is all the individuals of one species in one area at one time, and they can interbreed to make fertile offspring. And lastly, the allele frequency, that's the proportion of an allele within the gene pool. So these are key definitions to learn for AQA, but it's also to help get your head around the calculation, which we're going to go on to next.
Now the Hardy-Weinberg equation is this one here at the top, but you actually use both of these equations simultaneously. And the main thing you need to be confident with is, first of all, knowing those equations, but secondly, knowing what each component means. So a single letter by itself is an allele, and the P is representing the dominant allele, the Q is the recessive allele. These ones here in blue are the genotypes. So P squared is homozygous dominant genotype, meaning 2 copies of the dominant allele. 2pq would be heterozygous genotype because we've got 1 of each letter, and q squared is the frequency of the homozygous recessive genotype. So we use these 2 equations to be able to work out any one of those components using the data you're given. So in this example, we're told that cystic fibrosis is caused by a recessive allele, so that would be the letter Q. 0.02% of the population suffer from cystic fibrosis, and you then need to use that information to work out what proportion are carriers. Now, the first thing you should always do is identify which parts of the formula you have values for. So if we've been told that that percentage of the population are sufferers, that means that is our genotype Q squared, because they must have 2 copies of the recessive allele to be sufferers. So we've been told that Q squared is 0.02%, but we can't leave it as a percentage because in our Hardy-Weinberg equation, we say that the total equals 1, not 100. So it's not as a percentage, we need it as a decimal place. So we divide that by 100 to turn it into a decimal. Next then, we work out P and Q. So if we have Q squared, that means we can work out Q by square rooting that value. So there is our Q value. And if you have Q, we can then rearrange that second equation, making P the subject, and it would be 1 minus Q. So P is 0.986. And once you know what P and Q are, you can work out any other part of the equation. Now, they've said they want you to work out what proportion are carriers. And if they're carriers, that must mean that they are heterozygous in this example. So they're asking you to work out 2pq. So 2pq would be 2 times p, so 2 times 0.86, times q, which is 0.014, so 2pq is 0.03. And you need to look carefully to see if they specify your answer has to be given as a percentage or not. If it doesn't say anything, then you can leave it as 0.03. If it says a percentage, then times it by 100. So next we're considering variation. And individuals within a population of the same species may show a wide range in variation of phenotype. And this is due to both genetic differences but also environmental factors. The main source of genetic variation is mutations, but there will also be some genetic variation introduced by random fertilization of gametes and also in meiosis, crossing over and independent segregation. Now that variation is what leads to natural selection. Predation, disease, competition are all things that are going to be driving this natural selection. And as a result, those organisms with the phenotypes providing the selective advantages are more likely to survive and therefore pass on their favourable alleles to the next generation. This changes the allele frequencies within the gene pool, because whichever allele gave the selective advantage will become more frequent. So that takes us on to this idea of types of selection. Now, in topic 4, you learn about stabilizing and directional. What we're going to be covering is just disruptive, because that's the only one that comes up in topic 7. And disruptive selection, which we can see up here, is when individuals which contain the alleles coding for the extreme traits are more likely to survive and pass on their alleles. So as a result, the allele frequency changes over many generations, and more individuals end up having those 2 extreme traits. And the middling trait is lost because having that trait is not an advantage to survival, so it won't be passed on to the next generations. Now that disruptive selection ultimately can result in speciation, which is the creation of a new species. And this occurs when one original population of the same species becomes reproductively isolated. And that isolation means that there are now 2 populations of the same species, but they will not breed together. So that can result in the accumulation of differences in their gene pools to the extent that those 2 populations would be unable to interbreed and make fertile offspring if they were then able to reproduce. So there has to be something that is preventing them from reproducing with individuals from those separated populations, and therefore over many, many generations of those two populations accumulating different mutations, we end up with them having such different DNA that they cannot make fertile offspring. Now that could be allopatric speciation or sympatric. So allopatric is when there is a geographical barrier, so that could be a new mountain range, for example, whereas sympatric is when there are reproductive mechanisms preventing it. So in allopatric speciation, a population can become geographically isolated over time. It could be a new mountain range or new bodies of water separating the land masses, and that separates the original population into two. So they are now unable to reproduce due to that geographical barrier. Both of those populations will be accumulating different beneficial mutations, and over many, many generations, that will then result in their DNA becoming so different that they cannot interbreed to create fertile offspring. And therefore we class them as two new species. Sympatric, we said, is when the populations become reproductively isolated, and this is due to differences in their behaviour. So they are not geographically isolated, they are in the same area, but they still won't reproduce. This could be because a random mutation has impacted the reproductive behaviour. So for example, it might cause individuals to perform a different courtship ritual, or maybe they now become fertile at different times of the year. So even if they did mate, they wouldn't become pregnant, and therefore they would not be mixing genes within that particular gene pool. Due to this, these individuals will not produce together and there'll be no gene flow between the populations. Over time, these reproductively isolated populations, just like in the other example, accumulate different mutations to the extent that their DNA is so different they are no longer classed as the same species. Genetic drift is the change in the allele frequency within a population between generations, and there will always be genetic drift from one generation to the next, but continual, substantial genetic drift is what results in evolution. And the smaller a population is, the bigger the impacts allele frequency changes have proportionally, and this is why evolution occurs more rapidly in smaller populations.
For this part of topic 7, the populations, there's a lot of key terms that can come up as just 1 or 2 mark questions in the exam. So these would be great to turn into flashcards or just to test yourself. You can screenshot and take a copy of this table. So populations is a 2 mark definition, and your definition is it's a group of organisms of the same species living in the same area at the same time. That first part is 1 mark. The second mark is that they can interbreed to make fertile offspring. Habitat would just be 1 mark, so it's the part of an ecosystem in which particular organisms live. Community, so this is all populations or all the populations of different species in the same area at the same time. And it is essentially say in the same area at the same time. The ecosystem, so that is the community and the non-living components of the environment, so essentially the biotic and the abiotic factors. And you need to have an awareness that the ecosystems can range in size from being very small to very large, like global. Niche, this is often a 2 mark definition as well. So it's an organism's role within an ecosystem, including its position in the food web, but also its habitat. And it's normally split as it's an organism's role within an ecosystem, and then the second mark is also the habitat also. And each species occupies its own niche, and no 2 species can occupy the same. And if they do try, then that's when you get competition, and one species will eventually be outcompeted. Carrying capacity is the maximum population size that an ecosystem can support. And if you exceed that carrying capacity, there then won't be enough resources for all of the individuals, and they'll start to die, and that will then bring it back to the carrying capacity level. Abiotic factors are the non-living conditions of an ecosystem that affect the survival of individuals. And the biotic factors are the impacts of the interactions between organisms.
Have a look then at the abiotic factors and the biotic factors. Abiotic factors, as we said, are the non-living components of the ecosystem, and these can be factors like the temperature, oxygen and carbon dioxide concentration, light intensity, pH and soil conditions. Plants and animals are adapted to the abiotic factors within their ecosystem. These adaptations develop through the process of natural selection over many generations. The less harsh the abiotic factors, so for example if there's plenty of water and light, the larger the range of species and the larger the population size. So for example, in the desert, because it's very harsh conditions, you'll have a smaller range of species and the population size is smaller. Compared to down here where we can see there's plenty of light and water, we have a bigger range of species and there's more of each species. Biotic factors we said are the living components that are interacting together. So for example, we could be looking at competition, whether it's interspecific or intraspecific, and also the effect of predation. So our two types of competition. Interspecific competition is when members of different species are competing for the same resources that are in limited supply. Whereas intraspecific competition is when members of the same species are competing for the same resources and also a mate. And competition for a mate links to the courtship rituals. The predator-prey relationships you will see presented in graphs like this, which are called predator-prey graphs. And regardless of the species, the graph always follows a very similar pattern. The size of the predator and prey populations will both fluctuate. There will typically be more prey than predators because there has to be more prey to sustain the predators because they are eating them. And the size of the population change will always have a slight lag. So whatever happens to the prey, that will then happen to the predators shortly after. So the prey will increase first, then the predators will. The prey will then decrease because they're being eaten by the predators. But then because their population decreases, so will the predators. So if we think about sampling, first of all, why do we have to sample? Well, if you were asked to work out how many daisies there were in this field, you would have to do sampling rather than count every individual one. It's more time efficient and you're actually more likely to get an accurate answer because you'd probably miscount if you had to count all of those. But to make sure sampling is actually going to accurately represent that population size, you have to make sure that you are randomly sampling if it is a uniform area, and that's to eliminate bias. You also should have a large sample, so take at least 30 samples. Now I've got here a sampling summary just to help you see how it all links together. So if you have a slow moving or non-motile organism like plants, you would sample using a quadrat. If it was uniform distribution, you would do random sampling. If it wasn't evenly distributed, so for example a river in the middle of your field, then you would do a line transect. If you have a motile organism, meaning they can move, then you would need to use mark, release, recapture. So let's have a look at these three types of sampling. So first of all, random sampling. This is when you'd have two tape measures pulled out at right angles to create a gridded area. You would then need to use a random number generator to get two coordinates, and those coordinates indicate where you would place your quadrats. Once you place your quadrat, you then collect the data, and you would then repeat this at least 30 times and calculate a mean. Now if you didn't have an even distribution, we would use a line transect. So common examples of when you need a line transect are rocky shores or any kind of beachfront, as well as maybe going across a path or a river. So for example, if we wanted to have a look at the impact of the river, you would put your line transect going across it. If you wanted to know the impact of the rocky shore, you'd go from the rocky shore going further away into the land. You can either do a belt transect or an interrupted belt transect. A belt transect is when the quadrat is placed at every single position along the tape measure. An interrupted belt transect is when you place it every set distance. So it might be every 2 meters, every 5 meters, whatever seems appropriate for the area you are sampling. So for this method, you would place the tape measure at a right angle to the shoreline, just like we see here. You would then place your quadrat every 5 meters or whatever position distance you're going to state. Collect the data that you can see in the quadrat, but you still have to repeat this multiple times. But you would be repeating this by placing another 30 transects along the beach at right angles to the shoreline. And you have to be really specific with how you word number 4. If you just said repeat 30 times, you won't get the mark. You have to describe the position. Now, mark, release, recapture you'd use for moving animals. So first of all, we have the method here. An initial sample of the population in question is captured. These individuals are then marked and the number caught is recorded. And the mark has to be all weather resistant, so it doesn't come off, but it also has to cause no harm to the organism. The marked individuals are then released, but you have to leave them for long enough to evenly distribute across their habitats before you then take your second sample. And when you take your second sample, the total number captured in that sample is recorded, as well as how many you recaptured that had the marking on them. And the size of the population can then be calculated using that information. But as with all sampling, the more times you repeat it, the more reliable your results should be. So here's our calculation. To estimate the total population, you would do the number of organisms initially caught times by the number of organisms in the second sample. Divide that by the number of organisms that you recaptured. So here we have an example where we caught 20 butterflies. In the second sample we caught 22 and 9 of them were from the original 20 that were marked. So that would mean we had 48.89, but we have to round up to have a whole number. So 49 butterflies. Now you have to consider the ethics here of how you capture them. And you aren't meant to be an expert and know exact suggestions. What you need to know is how you capture them and how you mark them must cause no permanent damage. So don't use anything that's going to be toxic, and it can't increase the chances of predation, and it mustn't interfere with their reproduction either. There are some assumptions with this as well. So the estimate may not actually be very accurate because of these assumptions. Because we assume that the population size is constant, meaning there's no birth and death and no migration, which is very unrealistic. We also assume that the animals do redistribute evenly, but that might not be true. They might all huddle together near a food source. So next we have a look at succession, which is the change in an ecological community over time. A primary succession starts with a pioneer species colonising bare rock or sand. And the pioneer species, such as lichen, are adapted to survive in harsh abiotic factors, and through their death and decomposition they change the abiotic factors to become less harsh, and this forms a thin layer of soil which we call humus. Mosses and smaller plants are now able to survive in that area, and they further increase the depth and nutrient content of the soil. And this pattern continues, and as the abiotic factors continue to be less harsh, larger plants can survive and they continue to change the environment further. Each new species may change the environment in such a way that it becomes less suitable for the previous species, and therefore those previous species become outcompeted. Changes that organisms produce in the abiotic environment can result in a less hostile environment and increases the biodiversity. The final stage of a succession is known as the climax community, and this is dominated by trees typically. Now, a secondary succession is when the succession is disrupted and the plants are destroyed. Therefore, the succession has to start again, but we don't start again from bare rock because the soil is already there, it's just the plants that have been destroyed. So overall, from succession, as we have that change over time, the species richness will increase, as well as the number of organisms, or in other words, the biodiversity increases over time. As succession occurs, larger plant species and animals will also start to colonise the area. Now, often conservation of habitats involves controlling succession. The destruction of habitats, usually due to human activity, results in a loss of food and a loss of space or habitat for organisms, and it can lead to extinction. So that is why we conserve the habitats, and succession is often managed to do this. So by maintaining an earlier stage in succession and preventing the climax community, a greater variety of habitats are conserved, and therefore we get a greater range of species. There needs to be management though between the conflict between human needs, such as needing the wood for timber, and conservation in order to maintain the sustainability of the resources. So a compromise could be that forests are coppiced to provide timber for fuel, while still allowing the trees to survive. Now this picture isn't actually showing coppicing, this is showing that all the trees have been cut down. Coppicing is when you cut down just part of the tree so they can still then grow. So we begin with mutations, specifically gene mutations, which are a change in the DNA base sequence of a gene. And this mainly occurs during DNA replication, which is within interphase of the cell cycle. They occur randomly and spontaneously, but the exposure to mutagenic agents, so things like ionizing radiation, do increase the frequency at which they can occur. If a gene mutation does occur, then it could result in a different amino acid being coded for, and therefore a different sequence of amino acids in the primary structure of a protein. This could cause the hydrogen and ionic bonds in the tertiary structure to form in different locations, and therefore it results in a different tertiary structure 3D shape. And it's the shape of a protein that determines its function. So that is why a mutation could result in a different function or a non-functioning protein. Now, if that mutation happens in a gene that controls the cell cycle, it could then result in cancer. So there's 6 types of gene mutations that you need to know about, and we'll go through each of these 6 just to demonstrate what they actually mean. So first of all, if we have a look at addition mutations, this is when one extra nucleotide and therefore base is added to the sequence. So here we have our original example of a DNA sequence, and we've added in an extra base. Now the result of that we can see is that all of the subsequent bases are shifted along one position, and therefore after the mutation all of the codons are changed. And we also have one extra base just there that won't even code for a whole amino acid. So it's very likely that this will code for a completely different tertiary structure, and therefore either a non-functioning protein or a protein with a very, very different function. Deletion's a similar idea. So you delete a base this time and you still get what's called the frame shift, but all of the bases after the mutation shift back one position. So every single codon after the mutation is changed, and also you'll have one fewer codons because we now only have 2 bases in this final codon because one of the earlier bases was deleted. Substitution is where one of the bases is swapped for another one. So again, we can see we've got our original sequence, and here's the new sequence where we've had a mutation occur. So thymine has been swapped for adenine. Now this could result in a different amino acid being coded for, or it might actually result in the same amino acid being coded for because the genetic code is degenerate. It could also have no impact on the protein being coded for if this substitution happens within the introns, because introns do not code for amino acids. An inversion mutation is when a section of the bases detaches from the DNA sequence, and when it rejoins, they are the other way round, so they're inverted. And that's what we can see here. This section, TTC-AGG, that has been removed, but then when it was replaced, it's back to front, it's inverted. So that particular section of the DNA will now probably code for different amino acids, and therefore you'll have a very different primary structure. Duplication is when one particular base is duplicated at least once in the sequence. So again, you'll have a frame shift, just like with the addition, but it's going to have even more of an impact because you're adding in even more bases. Translocation is when a section of bases on one chromosome detaches and attaches onto a different chromosome altogether. Now this is a substantial alteration and can cause significant impacts on gene expression, and it can cause therefore a very, very different phenotype. Now multicellular organisms have a range of specialised cells, and those all originated from stem cells. And stem cells are undifferentiated cells that have the ability to continually divide and also become specialised. There are different types of stem cells as well, and this is determined by the differentiation abilities that they have. So you can have totipotent, pluripotent, multipotent and unipotent cells. So if we have a look at the totipotent first, these stem cells can divide and produce any type of body cell. So they have the ability to differentiate into any cell. And the totipotent cells occur only for a very, very limited time in the early mammalian embryos. Pluripotent stem cells are also found in embryos, and they can divide into almost every cell, but not the placenta cells. So they can divide an unlimited number of times as well, and they're used in treating human disorders. The multipotent and unipotent stem cells are found in mature mammals, so adult cells, and can divide to form only a limited number of different cells. Unipotent can only become one type of cell. Multipotent is a very limited number. So the bone marrow is multipotent stem cells. It can only make blood cells, for example. And unipotent stem cells, an example is making cardiomyocytes. Now, there are quite a few ethical issues with using pluripotent stem cells from embryos, and that is why induced pluripotent stem cells are now used. So these can be produced from adult body cells, that's what somatic means, using protein transcription factors to overcome the ethical issues. So what you would do is take a sample of the adult cells, which we can see here, a sample from the liver, and if you switch the genes off that made those cells specialised, then you can turn those cells back into their pluripotent state. So this links to the theory of transcription factors. And that leads us into transcription factors. So this is how transcription is controlled. We'll be looking at how translation is controlled later. So in eukaryotes, the transcription of target genes is stimulated or inhibited when specific transcriptional factors are moving from the cytoplasm into the nucleus. And when they do that, they can bind to receptors into the DNA to either turn on or off genes. And what that means is that gene will either be transcribed or it won't, and therefore the protein will either be coded for and created or not. And it's this turning on and off genes which makes cells specialised. So how this works then? Transcription of a gene only occurs when a molecule from the cytoplasm enters the nucleus, because it has to bind to the DNA, and the DNA is only found in the nucleus. These molecules are proteins, and each one can bind to a different base sequence on DNA, and that will be how it initiates the transcription of a gene. Once bound, that is when transcription can then begin, and mRNA is created. That is then used in translation to create the polypeptide chain. So if the transcription factor does not bind, the gene is inactive, meaning transcription won't occur and the protein won't be made. So what we can see here is a transcription factor. So we have our DNA binding site. So this is the part that will bind onto the DNA. And we have a receptor here because often another molecule has to bind to the transcription factor before it can work. And that is where estrogen comes in. Estrogen is a steroid hormone that can initiate transcription. The way it does this is, first of all, because it's steroid based, which is a lipid, it can diffuse through the cell surface membrane into a cell. It will then bind to the receptor. So estrogen is complementary in shape to the binding site of the receptor on the transcription factor. When it binds, it causes the DNA binding site to slightly change. That transcription factor will then move into the nucleus, and because the DNA binding site has changed shape, it's now complementary to the DNA so it can attach and initiate transcription. mRNA is then created, which can then leave the nucleus and be used in translation. Now, another way that protein synthesis is controlled is through something called epigenetics. And epigenetics is the heritable change in a gene function, but the DNA base sequence itself is not changed at all. Now, these changes can be caused by the environment, so things like your diet, stress levels, exposure to certain chemicals, and this can inhibit transcription. So one type of impact is increased methylation of DNA. Now, what this means is if you have lots of this chemical tag, methyl groups, binding to the DNA, it actually inhibits transcription. And the reason for that is when the methyl groups are added to DNA, they attach to the cytosine base and it prevents transcription factors from binding. It will attract the proteins that condense the DNA histone complex, and in this way it prevents sections of DNA from being transcribed. So that's what we can see happening here. The methyl groups are added, and because they are an opposite charge to the DNA, which is slightly negative in charge, it causes it to attract and to coil and bind up more tightly. And because that DNA is coiled up more tightly, there is no space for the transcription factors to be able to bind onto. And that is how it will inhibit transcription. Now, the opposite is acetylation, and that is when acetyl groups are added. But they don't bind to DNA, they actually bind to the histone protein, which is what the DNA wraps around. And decreased acetylation, so if you don't have very many acetyl groups bound to the histones, that will also inhibit transcription. If acetyl groups are removed from the DNA, then the histones become more positive. Now, just to emphasize, where it says here from the DNA, it does mean it's still bound to the histone groups, but they're within that DNA complex. But if you take away the acetyl groups, which are negative in charge, that means the histones themselves are more positive, and they'll then be able to attract the phosphate group on the DNA more, which is negative. And that makes the DNA and the histones more strongly attracted and associated, and again it coils up more tightly, preventing the transcription factors from being able to bind. So decreased, if you don't have very many acetyl groups bound, you also have transcription being inhibited. So in summary, if we think about just the methyl groups, if you don't have very many methyl groups bound, then that means the DNA is less tightly coiled, so it can be transcribed. And we call it euchromatin at that stage when it isn't very tightly coiled. If you had lots of methyl groups bound, so increased methylation of DNA, that results in the DNA and the histones tightly coiling up and the transcription factors can't bind, so transcription is inhibited. If you have increased acetylation of associated histone proteins, again, that would result in the less tightly coiled active euchromatin, so transcription can occur. But if you have decreased acetylation, you have heterochromatin, which is when all of the DNA and histones are tightly coiled, so the transcription factors cannot bind, and therefore transcription is inhibited. The final way that gene expression can be controlled is through RNA interference, or RNAi. And that happens at this stage here. So the DNA has been transcribed to make mRNA. The mRNA leaves the nucleus, but we're going to be looking at how translation can be controlled or interfered with using RNAi. So in eukaryotes and some prokaryotes as well, translation of the mRNA produced from target genes can be inhibited through RNAi. And this is when an mRNA molecule that's already been transcribed gets destroyed before it's translated, and therefore it won't create the polypeptide chain. And this is done using small interfering RNA, or siRNA. So this is the molecule that is going to destroy the mRNA, and that's why this is called RNA interference, because our mRNA has been interfered with. So let's have a look at how that happens. Transcription results in the production of mRNA, and mRNA is then used in translation by attaching to a ribosome, and that creates the primary structure of a protein, or the polypeptide chain. However, if there is small interfering RNA, that can disrupt the mRNA that's been created and destroy it, and therefore it prevents translation from occurring. And this is what we mean by RNA interference. So small interfering RNA results in this interference. So that's just a concept map just to show you how this all links together. Now, how this actually destroys the mRNA is you have some double-stranded RNA, but there's an enzyme that can cut that and make it single-stranded, but also into small pieces. So it's small and it's going to be interfering, so siRNA. That will then bind to an enzyme, and that enzyme's called RISC, but you don't need to know that's the name. Because it's single-stranded, this complex of the siRNA and the enzyme can bind to the mRNA molecule that was made in transcription. And that then means that the enzyme is able to cut up that mRNA into small pieces, and therefore it cannot be used in translation. It won't be able to bind to the ribosome, and because it's cut up, you can't translate that entire sequence. The next thing you need to know about is cancer, which we briefly mentioned at the start. So it can result from mutations in genes that regulate mitosis. And if these genes do mutate and a non-functioning protein is created, then that means mitosis cannot be controlled. And that is why you end up with this uncontrollable cell division, and that is when a mass of cells or a tumour is created. Now, there are different types of tumours. You can have what's called a benign tumour or a malignant tumour, and you do need to know the differences in the properties of these. So a benign tumour can grow very large still, but it will be dividing at a slower rate, so it will take longer for there to be a larger tumour. Benign is non-cancerous, and one of the reasons it's classed as non-cancerous is they produce this adhesive sticky molecule, and that will then surround it so it cannot move. It's also in a capsule as well, which means that these cells that are having this uncontrolled mitosis, they're not able to break off and spread to other parts of the body. And therefore, if surgery can be conducted on the part of the body that the tumour is located, you may be able to cut out and remove that entire encapsulated tumour, and then it'd be very rare that the tumour would regrow. So the impact is localised, and it's often non-life-threatening because you can cut it out and remove it. But it does, as I said, depend on the location of the tumour. So, for example, a benign brain tumour is still very dangerous because any type of brain surgery is very high risk because you have to cut through the skull, and in removing that tumour you could actually damage some of the other brain tissue. Now, in contrast, malignant tumours, these are the ones that are classed as cancerous, and they do grow much quicker. The cell nucleus becomes very large and it returns back to its unspecialised state again. They don't produce that adhesive sticky layer or the capsule, and therefore they can metastasise. And what that means is cells of the tumour can break off, spread through the blood, and therefore lodge in other tissues in the body, and then you get secondary tumours in other organs. The other thing that we can see here is because it's not encapsulated, yes, it also links the idea of metastasis, but also it can grow projections and reach the blood supply. And once that tumour has its own blood supply, it's going to be receiving lots of oxygen and glucose for respiration, and that is one of the reasons why those cancerous tumours can grow so rapidly. It can be life-threatening, and the removal of the tumour needs often supplementary treatment like radiotherapy and chemotherapy, and the reoccurrence is more likely than with a benign tumour. So the tumour development then, as we said, it could be due to a gene mutation, and if it is a gene mutation, the mutation would have had to have occurred either in the tumour suppressor gene and or the oncogene. It could also be linked to epigenetics though. So if you have abnormal methylation of a tumour suppressor gene or an oncogene, or if you have increased estrogen concentrations affecting transcription. So those are all of the possible causes of these tumour developments. Now, we've used this term tumour suppressor gene and oncogene, but let's just go through what those genes code for. So oncogenes are the mutated version of a proto-oncogene. And proto-oncogenes code for proteins which are involved in initiating DNA replication in the cell cycle. An oncogene, though, can result in the process being permanently activated to make cells continually divide even if new cells aren't needed in that part of the body currently. Tumour suppressor genes code for proteins which are involved in controlling the cell cycle, and they're involved in slowing down the cell cycle or to cause cell death. Now, if a mutation results in the tumour suppressor gene, that means that protein isn't produced and cell division isn't slowed down, so it'd continue. Mutated cells would not be identified and destroyed. Now, we said that epigenetics can also link to the cause of tumours, and that was because of abnormal methylation. So we looked at how methylation can cause genes to be turned on or off, and if a tumour suppressor gene becomes hypermethylated, that would mean there's an increased number of methyl groups bound to it, and that results in the gene being inactive and it is switched off. So if that happened to a tumour suppressor gene, that means that protein won't be produced that slows down the cell division in the cell cycle. Now the opposite could occur in an oncogene. So as they may be hypomethylated, meaning you don't have very many methyl groups attached, that could result in the gene being permanently switched on, and therefore lots of the protein is being produced to initiate that cell division constantly. Now the final thing we said is estrogen also has an impact in increasing the risk of cancer, and it's linked to this specific example. So estrogen is produced by the ovaries to regulate the menstrual cycle. However, after the menopause, the ovaries stop producing estrogen, and instead the fat cells in the breast tissue produce estrogen instead. And this is what's been linked to breast cancer in women post-menopause, and that is why age is one of the risk factors of developing breast cancer. So we looked at earlier how estrogen is a molecule that can bind to receptors on transcription factors. So if that is going to happen to a transcription factor involved in the cell division, so for example, it's a transcription factor that can bind to the DNA of a proto-oncogene, that would mean that that particular gene is permanently turned on, activating cell division within the breast tissue. Now, it has a knock-on effect as well, because as the tumour grows, because those tumour cells are breast tissue cells, that will result in even more estrogen being produced, and therefore even more of those proto-oncogenes could be switched on, activating more cell division. So this is an example of positive feedback. The more that we have of the tumour, the more estrogen, but that results in even more tumour cells occurring. The next bit of the spec is looking at the genome, and the genome is the entire genetic material of an organism in the nucleus of a cell. And sequencing of this genome, what that means is identifying the exact DNA-based sequence for all of the DNA in a cell. And many organisms' genomes have now been sequenced by scientists, including the human genome, which took about 13 years, but it was finally completed in 2003. And that's the timeline we can see here. Now, sequencing methods being used to identify this DNA-based sequence are continuously being improved and now automated. And that's why AQA have taken it off the spec. You don't need to know any exact methods because they are so frequently improving that if you did learn one, it would probably be outdated within a year. Now, there have been methods such as the Sanger method, but as I said, you don't need to know that because the science of how this is done is so fast changing. All AQA want you to be aware of is that fact that sequencing methods are now automated and they're continuously being improved. Now, simpler organisms like prokaryotic cells, you learn earlier on in the course that those DNA do not contain introns. And that means that the genome can be used to directly sequence the proteins that derive from the genetic code, or in other words, the proteome. And that's really useful for many reasons. For example, identifying potential antigens on a microorganism like a bacteria to then create an antigen for a vaccine. More complex organisms like humans and eukaryotic cells, other eukaryotic organisms, have introns. And we also have regulatory genes in our DNA. And due to this, the genome can't easily be used to translate to the proteome. And the proteome is the entire set of proteins within a particular cell. Now, the next bit of topic 8 is looking at gene technologies. For example, recombinant DNA technologies. And I've put all of the rest of topic 8 onto one flow diagram here, and we're going to go through it a section at a time. So the three key concepts are creating DNA fragments, the idea of genetic fingerprinting, and then the uses in genetic screening, counselling and locating of genes. So gel electrophoresis is actually used in genetic screening, but also genetic fingerprinting. Genetic screening, you're screening for genes and personalised medicines. Genetic fingerprinting is used in forensic medicine, forensic science, medical diagnosis, plant and animal breeding and paternity tests. But what we're going to be focusing on first is this left-hand side. So creating DNA fragments, we're going to look at what are the three methods for how DNA fragments can be created, how those fragments can then be cloned either in vitro using PCR or in vivo looking at the use of sticky ends, plasmids and gene markers. So we're going to look at creating DNA fragments first. So recombinant DNA technology, what this means is you're recombining the DNA of two different species. And this is what enables scientists to manipulate and alter genes to improve industrial processes, but also medical treatments. So, for example, manipulating the DNA in bacteria so it can produce human insulin. And the first step in these technologies is you have to isolate the fragment of DNA that you want to recombine or insert into another organism's DNA. And there's three methods: reverse transcription, restriction endonucleases, and the gene machine. So reverse transcription, as the name suggests, we are doing the opposite of transcription. So we're using an mRNA molecule to create a DNA copy. So the enzyme that is used is going to make copies of DNA from mRNA. And the enzyme is reverse transcriptase. And reverse transcriptase naturally occurs in viruses like HIV. What you would then do is you would find a cell that produces lots of the protein of interest. Then from that cell, you should be able to find large amounts of the mRNA that will code for that protein. You can then isolate that mRNA and add the enzyme reverse transcriptase. And that will join the DNA nucleotides with complementary bases to the mRNA sequence. You will then have created a DNA sequence, but it will only be single-stranded. To make the DNA fragment double-stranded, the enzyme DNA polymerase is then used to create that second strand. So this is what we can see here. We've got our mRNA molecule, which is single-stranded. You would then add the enzyme reverse transcriptase, and free-floating DNA nucleotides would align opposite their complementary base pairs, and we've then created a single strand of DNA. We'd then add DNA polymerase to make it double-stranded. And the advantage of this option is cDNA, which is what we call the molecule that's made, is intron-free. And that's because it was copied from mRNA, and mRNA already has the introns removed. The second option is using restriction endonucleases, and these are enzymes that cut DNA. They naturally occur in bacteria as a defense mechanism to cut up the DNA of any invading organisms. And there's many different types of restriction enzymes that have an active site complementary in shape to a particular DNA base sequence. And we call that the recognition sequence or recognition site. And therefore each enzyme will cut the DNA at a particular location. Some enzymes cut at the same location in the double strand, and that would result in a blunt end. Other enzymes cut to create staggered ends and therefore exposed bases, and we call those sticky ends. Now that's what the diagram is representing here. This enzyme, HIND3, cuts the DNA, but it doesn't cut straight down, creating two straight edges, which is what we call a blunt end. It cuts in this staggered way to create exposed bases on both of those cut chains, and we call it sticky because there's the potential for complementary bases to align and then they join together. Now the palindromic sequence, that is referring to where it cuts. The top part of our DNA sequence is the same as the bottom part backwards. So palindromic means it's the same forwards as it is backwards. The third option is the gene machine, and this is when DNA fragments are created using a computerized machine in the lab. So the first part of the process is you'd have to examine the protein that you want to create lots of, identify what the amino acid sequence is, from that work backwards to sequence what the mRNA would be, the mRNA sequence, and therefore the DNA sequence. You would then enter that DNA sequence into the computer. There'd be checks for biosafety and biosecurity, that the DNA being created is safe and ethical to produce. The computer can then create these small sections of overlapping single strands of nucleotides that make up the gene, and we call those oligonucleotides. The oligonucleotides are then joined to create the DNA for the entire gene. PCR, which we'll be looking at shortly, can be used to amplify the quantity that you have, and it makes it double-stranded. So this process is very quick, it's accurate, you can design it to be intron-free, so that means the DNA can be transcribed in prokaryotic cells. So that's creating our DNA fragments. The next thing is we're going to look at how those DNA fragments can be amplified or cloned. And the first option is in vivo, which means cloning these DNA fragments inside of a living organism. So here are our stages of in vivo cloning. We've already looked at creating the DNA fragment, so the next step is how that fragment is inserted into a vector and how the host cell will then incorporate that vector. So those are the two bits we're going to look at first. Now, restriction endonucleases are used in this particular method. So in vivo cloning, you have to create your DNA fragment using a restriction endonuclease. And we can see here that enzyme has cut the DNA at the recognition site, and we have these sticky ends. Now, the DNA fragments need to be modified before they are used to make sure that transcription definitely occurs. One modification is a promoter region is added, and this is added at the start of the DNA fragment, and a promoter region is a sequence of DNA bases, which is the binding site for DNA polymerase. So that is to make sure the enzyme can definitely attach and transcription occurs. The second modification is a terminator region is added, and this is put at the end of a gene, and it causes RNA polymerase to detach and therefore stop transcription So only one gene at a time is copied. So then we need to see how is the DNA fragment inserted into a vector. But in case you're not sure what a vector is, first of all, a vector is something that will carry the DNA fragment into the host cell. So whatever organism it is that you want to now contain that DNA fragment, the vector will transport it into it. And often plasmids are the vectors that are used. And plasmids are just loops of DNA that are sometimes found in bacteria. So the way that we insert the DNA fragment into the plasmid, which is our vector, is we cut open the plasmid using the same restriction endonuclease that was used to cut the DNA fragment of interest. That means that the same sticky ends are going to be created on the plasmid as the sticky ends that you have on the DNA fragment. So then when you mix your DNA fragments and the plasmid together, you should have complementary base sequences opposite each other so that the two pieces of DNA can align, and then you add the enzyme DNA ligase to join those nucleotides together. And that's what we're just seeing here, that the enzyme DNA ligase is going to be joining those nucleotides together by catalyzing the condensation reaction to create those phosphodiester bonds. So we now have, hopefully, the DNA fragment inserted into the vector. The next step then is getting that vector into the host cell where you want the gene to be expressed. So to do this, the cell membrane of the host cell must be made more permeable so that we increase the likelihood of these plasmids being able to move into the cell. Now to do that, you can mix calcium ions with the host cell and heat shock the cell. And what that means is you have to have this sudden increase and then decrease in temperature. That affects the permeability, and therefore the vector is more likely to enter the host's cytoplasm. Now the final thing is, in this in vivo process, is how you can actually check to make sure that the plasmid definitely took up the DNA fragment, and how you can check that the host cell definitely took up that plasmid. And that is where these gene markers come in. And that's what we're going to look at next. How you can identify whether the cells were transformed, meaning they took up the modified plasmid. And then if you do have cells that have, the final step will be growing lots and lots of those transformed cells. But we're just going to focus on how to identify transformed cells. Now there's three issues that might have happened, which is why we have to check that we do have transformed cells that contain the recombinant plasmid. Now issue number one is that plasmid that you've created, which is our vector, maybe it didn't actually get inside of the host cell, which might be a bacteria cell. The second issue could be that maybe the plasmid, it had sticky ends, it might have just rejoined with itself and sealed back up. So you might have plasmids that did get in, but they don't contain the DNA fragment, so that's not going to be useful. The final thing that could happen is that DNA fragment that you created, it could actually loop around and join together on itself, creating a mini plasmid. And therefore it's not attached to the plasmid, it's not going to be able to enter into the cell, and it won't work. So that's why we have to try and examine and identify which cells do contain the plasmid with the DNA fragment inserted into it. So one method is using marker genes. And marker genes are genes that are occurring on the plasmid, and we use them to identify whether the host cell, which in this case we're thinking about bacteria, successfully took up the recombinant plasmid. And the three types of marker genes which are commonly used are genes for antibiotic resistance, genes that code for fluorescent proteins, and genes that code for certain enzymes. So we're going to go through the antibiotic resistant marker genes option first. So we have here a plasmid which contains two marker genes. One gene is one that makes the bacteria resistant to the antibiotic tetracycline, and the other one is a gene that produces a protein to make the bacteria resistant to the antibiotic ampicillin. And here is our DNA fragment. Now that DNA fragment is deliberately inserted in the middle of the tetracycline gene. And in that way, it disrupts the tetracycline gene, and the bacteria would therefore no longer be resistant to the antibiotic tetracycline. So if that bacteria was exposed to tetracycline, the bacteria would die. So we then grow the bacteria on agar, and we can see these different colonies have grown. We then transfer a copy of those colonies in the exact position using this velvet block. So you basically stamp it on top of your agar plate, and then you place that on top of a new agar plate. And this time it's an agar plate with the antibiotic ampicillin dissolved within it. And what we can see is which colonies now are killed off because they are not resistant to ampicillin. We then do the same thing again. Now we transfer those colonies onto a second plate which contains the antibiotic tetracycline within the agar. And we can see we now only have 3 colonies left. So we can analyse these 3 agar plates and the different colonies that are present to work out from these bacterial colonies which of those bacteria didn't take up a plasmid at all, or which took up the plasmid but it didn't have the DNA fragment inserted in it, or which took up the plasmid and it did have the DNA fragment inserted in it, because that is the one that we're interested in, because we want that DNA fragment inserted in. So we can see here in this first one, this first copy plate, that if there is ampicillin in this agar, that means only bacteria which contain the plasmid at all would be able to grow. Because if it didn't contain the plasmid, it wouldn't be resistant to ampicillin and the bacteria would die. So that means only these remaining colonies definitely contain a plasmid. But what we don't know is whether it's the original plasmid or the plasmid of interest which has the DNA fragment inserted into it. And that's why we needed to do another copy plate where we can see we have tetracycline this time in the agar. And the plasmid of interest has the tetracycline resistance gene interrupted. So that means the bacteria that have the plasmid with the DNA fragment in should not grow on this plate because that gene has been interrupted. So that means colony A, D and I do have a plasmid, but because they can grow on tetracycline, they must contain the original plasmid because the gene is still intact and therefore they're resistant to tetracycline. Now colonies E and G survived on ampicillin but were killed on tetracycline. So that means colony E and G must contain the plasmid of interest because they are resistant to ampicillin but they're not resistant to tetracycline. And that is how we use those antibiotic resistant marker genes. Now a similar idea is using the fluorescent gene markers, and GFP, which is green fluorescent protein, is a gene that naturally occurs in jellyfish and it can create this protein that fluoresces. So the same idea again, you can have a plasmid which has the gene for GFP and we then have our DNA fragment. We deliberately insert the DNA fragment in the middle of the GFP gene. So that means any bacteria that take up the plasmid with the DNA fragment in will no longer fluoresce. So that's what we're looking for, is when we grow the bacteria, all of these colonies which are not bright green, they must have the gene disrupted by the DNA fragment. So the ones that are not glowing are the bacteria of interest. The ones that are glowing have the original plasmid with the gene still intact, so the DNA fragment didn't successfully get taken up. The last one was enzyme markers, and the enzyme lactase is often used because it can turn a certain substance blue to colourless. So the gene for this enzyme is inserted into the plasmid. Again, it's inserted deliberately in the middle of the gene for that enzyme to disrupt it. All of the bacteria are then grown on an agar plate with this colourless substance, and any colonies that can turn the colourless substance blue must be the ones that contain the original plasmid without the DNA fragment. However, any that cannot catalyse that reaction of going from colourless to blue, so the colonies remain colourless, must contain the DNA fragment because the enzyme gene was interrupted. So those would be the bacteria that you would then remove and grow large quantities of. Now, alternatively, the DNA fragments can be amplified in vitro, and that means not in a living thing, and that is where PCR is used, which is the polymerase chain reaction. So once you have your DNA fragment, what you would then do is amplify them using an automated machine, and this is the equipment that you would need. The machine is called a thermocycler. You would add the DNA fragments that you isolated that you want to clone. You need to add the enzyme DNA polymerase to catalyse the creation of new DNA polymer chains. You would add primers, which are short single-stranded sequences of DNA to help initiate the DNA replication. And you need lots of DNA nucleotides so that you can create these new DNA polymers. Now, this bit here where it says Taq polymerase, this is referring to the fact that this DNA polymerase is actually taken from bacteria which can survive in extreme temperatures. And that is because this machine is run at very high temperatures. So we need to have a DNA polymerase version which isn't going to denature at high temperatures. So here's our method. The first step is the temperature is increased to 95 degrees C, and what that does is it breaks the hydrogen bonds between the DNA fragment that you added. So we now have single-stranded DNA molecules. The temperature is then decreased to 55 degrees C, and that is to allow these primers to attach, and we call that annealing. The enzyme DNA polymerase can then attach and join any of those complementary DNA nucleotides that align. So it's going to join those adjacent nucleotides, creating the second chain. So we have now created a copy of that DNA fragment of interest. The temperature is increased to 72 degrees at this stage because that is actually the optimum temperature for the Taq DNA polymerase. Now that is one cycle. This machine will be left to cycle over and over, so you're making thousands of copies of that DNA fragment of interest. So the advantages of PCR is it's automated. Once you've added all of those ingredients, you turn the machine on and therefore it will just start working and create the fragments. It's very, very rapid. So you can make 100 billion copies of DNA within hours. And it doesn't require living cells. So it doesn't require bacteria like you needed in the in vivo methods, which makes it quicker and it's less complex. Now, DNA probes are used in lots of the gene technology applications. So what a DNA probe is a short single-stranded piece of DNA, and they are labelled so you can identify where they are. And they're labelled usually either using a radioactive molecule or a molecule that will fluoresce. So that is how we can then visualise where that DNA probe is. And that's what it's used for, locating specific alleles of genes. And this could be used to screen patients to see if they contain a gene of interest linked to a heritable condition. It could be used to identify whether you have a gene that might indicate a particular response you'll have to a drug. So the way that this would be done is a sample of the patient's DNA is removed, and we heat it up to make that then single-stranded. You would mix that patient's single-stranded DNA with these single-stranded DNA probes, and if there is a DNA probe which is complementary to the sequence of a patient's DNA, then they will bind together. And these DNA probes can be designed to be the exact complementary sequence to a particular allele, and therefore, if they do align, that means that this patient does have the allele that is known to cause a particular disease. So the final step would be you'd have to visualize this DNA probe, and if you used a radioactive label, you'd use an X-ray machine, and that would make that radioactive label light up. If you used a fluorescence, then you'd need to use a UV light. DNA hybridization is when the DNA is heated to separate the double helix and single strands, and this is then mixed with a complementary sequence of single-stranded DNA. So much like we just looked at there in the DNA probes. Once it's cooled, the complementary strands will join together, which is what annealing is. And this method is used with those DNA probes that we just saw in medical diagnosis to see if someone does have a particular allele in their DNA, which is known to cause a particular disease. Now, alternatively, it could be used linked to personalised medicines and genetic counselling. So as well as screening for the presence of alleles, there are other uses also. Now, if you have used it to screen for a particular allele, that will enable the doctors to then select medicines that are known to work better with that particular genotype. Because there are some drugs that will have different effects on you depending on what your genotype is. So some drugs are more or less effective depending on the alleles you have. So it means that number one, the drugs that are selected will be more effective at treating your condition, but also it's more cost effective. You're not taking drugs that aren't going to have any effect, which would therefore have cost just to have drugs that are doing nothing. Genetic counselling is a type of social work where people are given advice based on their results of the top experiments. So if you are screening to see, do you have a particular allele which is known to cause a disease, then genetic counsellors would talk you through, first of all, before you screen, whether you would actually like to screen and what the pros and cons are of finding out if you have that disease-causing allele. And then afterwards, if you do have results that show you have a disease-causing allele, they'll talk you through what your options are now. So it could be they might talk you through options of changing your lifestyle to reduce the likelihood of that disease having any impact, or how you can monitor and check to see if you do have that disease, you detect it earlier, and therefore you're more likely to have successful treatments. So next we go on to this concept of genetic fingerprinting, which is a way to examine DNA. And this uses VNTRs in your DNA. And 95% of human DNA, or sometimes the estimate is actually around 98%, is made up of introns. Introns do not code for amino acids, and they contain VNTRs, variable number tandem repeats. And what this means is you have sequences of DNA just repeated over and over and over. And the probability of two different individuals having the same VNTRs is very, very low. However, the more closely related you are, the more similar your VNTRs are. And therefore, genetic fingerprinting analyzes the VNTRs in your DNA, and it can use this to identify genetic relationships and also variability within a population. Now, genetic fingerprinting is split into these stages. So we'll go through collection, extraction, digestion, separation, hybridization, development, and analysis. Now, collection just means collecting the DNA sample. And if this was a crime scene, well, you have to collect whatever you happen to have. If this is a collection to do a paternity test, you might take the DNA from white blood cells, or it could be DNA from a cheek swab sample, for example. Now, the smallest sample of DNA can be collected, and it could be from blood, it could be from body cells or hair follicles. Depends if it's a crime scene, what you have available. If the sample of DNA is small, which most of the time it is, then PCR would be used first of all to create a large sample of that DNA. Then we would have to digest this sample, and this is where we use restriction endonuclease enzymes. So they will cut at the recognition sites and we're left with these sticky ends. The DNA samples are then loaded into these holes, which we call the wells, on a gel plate. And the gel is placed in a buffer liquid and it has an electrical voltage applied to it. Now, DNA is negatively charged because of these negative charges on the phosphate groups of DNA. So the DNA that's been injected into these wells will actually start to travel through the gel towards the positive end of this gel because it's attracted to it because DNA is negative and we've added this positive electrical charge. So this is the stage of how we separate out the DNA fragments that have been injected into the wells. And this is the gel electrophoresis step. So the agar gel creates a resistance for the DNA moving through, and the smaller pieces of DNA can therefore move faster, so they'll move further along the gel. This is how the different lengths of the DNA VNTRs are separated. An alkaline is also added to separate the DNA so it becomes single-stranded instead of being double-stranded, and that is then useful for the next step, hybridization. So we add in DNA probes, which we said are short, single-stranded pieces of DNA that are either radioactively or fluorescently labelled. They have been designed to be complementary in sequence to the VNTRs. So you add these DNA probes, they'll align next to the different VNTRs, and they will then hybridize, meaning join together. The development stage is how we can then visualize where those DNA probes are. Now the agar gel, as it starts to dry out, it does shrink and it cracks. So the VNTRs and the DNA probes have to be transferred to a nylon sheet. And the nylon sheet can then be exposed to X-rays to visualize the DNA probes if they were radioactively labeled, or if they were fluorescently labeled, you would apply a UV light. And what that will then give you is one of these patterns that you might be more familiar with, where it shows you all of the VNTR bands and you can see the positions of them, and then you can identify or do your analysis. So in this case, this was our unknown sample of DNA, and we then had 5 options of organisms that may be our unknown. And you compare the position of the bands, and if they have the same positions, then it is the same DNA. So our unknown, we can see in this example, was the same DNA as organism 3. So that enabled us to identify that the unknown sample must have come from organism 3, because all of the other bands, they don't match up. So this can be used in paternity tests or at a crime scene. It can be used to identify whether the DNA found at the crime scene is matched to a potential suspect, or it could be the victim. It doesn't prove any crime has happened or that the person is responsible. It just proves that a person was present at the crime scene because their DNA was there. So here's another example just showing you these results. This time it's for a paternity test. So we have the mother's gel electrophoresis results. We have the child, and then we've got father 1, 2, 3, and we have to identify which one is the father. Now, any of the bands that didn't come from the mother, which you can see here have been highlighted in orange, must have come from the father instead. Now, in this case, actually, none of these would be the father because none of the bands exactly line up. Number 1 is very, very similar, but they don't actually have that band there. So this paternity test actually shows that none of them were the father. Now, another set of uses are, we've talked about the forensic science, but it could be used for medical diagnosis. So to identify, do you have a particular sequence of DNA that's linked to a genetic illness, or it could be used to identify how closely related individual animals or plants are before you breed them together, to make sure that you're not going to be doing any inbreeding and therefore potentially passing on harmful recessive alleles. So that is it, topic 8 in a nutshell. I hope you found it helpful. If you have, please give this video a thumbs up and make sure you subscribe so you don't miss out on any of the other whole topic videos.